Well, welcome everyone to day 3 of the World Stem Cell Summit. This is a really packed day. We're honored to have so many guests. This is record attendance this year from the beautiful Palm Beach County Convention Center. My name is Bernard Siegel. I'm Executive Director of the Regenerative Medicine Foundation, which is the nonprofit organization that is the producer of this global event. And this is our 12th World Stem Cell Summit and RegMed Capital Conference. To navigate what sessions to go to, I want to point out at noon today, or at lunchtime today, there's a special Japan Symposium. I think it's the 5th year that we've done this, hosted by Dr. Norio Nakatsushi, formerly that headed ISEMS at Kyoto University and now leads the Nakatsushi Foresight Foundation. And if you really want to understand the nature of what is going on in the world, Japan is a leader, so that symposium is very important, and actually you'll have a choice of what to do at lunchtime, and you might want to grab a sandwich or something from downstairs, but certainly spend some time at the hall and budget for it, because we also have the Cure Alliance has a special panel that includes Dr. Arnie Kaplan and Dr. Ricordi related to the changes in regulations that we are dealing with now, that of course is one of the hot topics. One of my great pleasures of the year, of any time I've actually been working in the field, is the opportunity to see laboratories, visit sites, and try to understand the breadth of this amazing field of regenerative medicine. So this year I had the opportunity with a fellow board member of my organization, Tim Choate, to get the backstage tour of the Kennedy Space Center in a meeting that I had with the Center for Advancement of Science and Space, CASIS, which is a co-organizer of this event, to better understand the work in cell biology that's going on the International Space Station. At this point, I've been doing this 14 years. It's a little hard to be impressed, but I was enthralled about what I have seen and delighted that CASIS has come on board. What is happening in low Earth orbit right now and in microgravity will change this field positively. There's even a company presentation later today from Zero Gravity Solutions, which is going to be a gigantic commercial success from technology and study of cells on the International Space Station. It's absolutely amazing. So I want to welcome this plenary panel and Dr. Michael Roberts, who's going to lead the panel, so that we can all learn about the exciting things that are happening in space. Michael?
Good morning, everyone. I wanted to echo the comments of Bernie and welcome everyone on the 3rd day of the conference. It's exciting to see so many people have awakened on time to make it in this morning. In honor of Japan Day, Ohayou gozaimasu. Genki desu ka? No. This morning's panel is an opportunity for CASIS to present to this audience the opportunities that are available for research in a new environment. I'd like to take just a few minutes to reintroduce what those possibilities are. And what you're going to hear this morning are presentations from investigators who have had opportunities to develop and in several cases actually have already flown their research to a brand new environment. As everyone in this audience I'm sure understands, stem cells are exquisitely sensitive to their environment, and their physical environment drives their ability to differentiate and do things that are of great benefit to us for a variety of applications. What you're going to hear this morning in this panel are some different aspects of the value of microgravity and access to the space environment and how it impacts our fundamental understanding of how stem cells behave in the space environment, and experiments that go all the way through to the translational and therapeutic end for investigations that are focused on beneficial applications of these stem cells for regenerative medicine. So just take a minute to reintroduce CASIS to you, which you've heard from in other presentations at this Congress. CASIS is a not-for-profit organization that has as a mission the responsibility for enabling access to the International Space Station National Lab. In 2005, the U.S. Congress declared the U.S. operating segment of the International Space Station a national lab, with the intention of opening it up as a research platform and orbiting laboratory that could enable investigators from a variety of disciplines, from a variety of institutional constructs within the U.S., to have access to space as a unique environment in which to test hypotheses, to demonstrate technologies that were of interest to their portfolio. CASIS has the responsibility of engaging with these new-to-space communities as well as seasoned microgravity investigators to provide for them access to the International Space Station and the space environment. In thinking about the possibilities of utilizing space as a platform, I'm reminded of this iconic picture taken in the late 60s. For those of you who were born in the 60s or before, will remember that we as a nation had a mission declared by President Kennedy to go to the moon before the end of the decade. So our target was having a human presence on the moon sometime before that end of that decade. The reality of the outcome of that was that for the first time we realized the sanctity of life and the specialness of our planet. So even though our target was the moon, I think what we actually discovered was Earth for the first time and what it looked like from the space environment. We now have a similar opportunity with the International Space Station. The International Space Station was designed as an international collaborative project to support research in low Earth orbit. But I think most of the discoveries and a lot of the value that we've seen for the International Space Station has less to do with the space environment and more about learning about us as humans and ways that we can utilize that space environment to advance our understanding of life on Earth as we know it. CASIS's mission in managing the ISS National Lab is on the far right. It's simple and straightforward. Our mission is to improve life on Earth. And we do that through direct engagement with commercial companies. We have a variety of pharmaceutical companies, large Fortune 500 companies, as well as small entrepreneurial companies that have developed and flown projects to space. We also are working very closely with government agencies, including the National Science Foundation and the National Institutes of Health, NCATS in particular, to offer sponsored programs. So although CASIS can act as a funding organization to provide research dollars, what we're actually seeking to do is leverage additional dollars from other government agencies, from the commercial and from public-private consortia, to enable research that benefits your research interest. So this morning, I'd like to introduce our panel this morning to introduce to you the concept of stem cell research on the International Space Station National Lab and the benefits that have derived from that. So in the second chair to my left, I'd like to introduce Chunhua Xu from Emory University School of Medicine, and she'll give our first presentation this morning.
So how do I get to my slides? Yeah. Green arrow? Okay. Yeah. So thanks, Mike, and thanks for the organizer for inviting me here. It's really an honor for us to present our work here. So I'm gonna first give an overview of cardiomyocyte differentiation from pluripotent stem cells. Then I'll show you how simulated microgravity can improve cardiomyocyte differentiation. As you know, human pluripotent stem cells can self-renew extensively under proper culture conditions. These are very powerful cells. They can differentiate into virtually all types of cells in the body, including functional cardiomyocytes. These derived cells can then be used to treat degenerative diseases. For example, cardiomyocyte derived from human pluripotent stem cells can be used to treat heart failures, to recover the heart function. The other promising application of human PSC-derived cardiomyocyte is disease modeling and drug discovery. For example, my lab's interested in modeling CPVT using iPS cells. CPVT is an inherent cardiac disorder that can lead to fetal arrhythmia. So this is often caused by mutations in genes involving calcium handling. So the fundamental therapy for this disease is beta blocker. But for unknown reason, about 25% of patients do not respond to beta blocker. And we have identified such a patient in our clinic. This boy did not respond to the beta blockers, but another drug called Flecainide can control his arrhythmia. We then generate iPS cells from these patients and then turn them into cardiomyocytes. In the dish, we can see the disease phenotype as shown here by the abnormal calcium waves and calcium sparks. We then treat the cells with Flecainide and beta blockers. So similar to what we see in clinic, the cells did not respond to the beta blocker, but Flecainide can substantially improve the calcium handling of the cells by eliminating those arrhythmogenic calcium waves and calcium sparks. So our experiments provide a proof of principle showing that human PSC-derived cardiomyocyte can recapitulate the clinical drug response. However, there are still many challenges to be overcome for application of human PSC-derived cardiomyocytes. For example, cardiomyocyte differentiation can vary even with the most advanced differentiation protocol. And the cells typically culture on 2D cultures, so that can limit the cell-cell interactions. So the cells are typically immature. The low cell yields can be a bottleneck for all the application of the cells. So we and our collaborator have tried to address some of those issues. So I'm going to show you some related data. So to induce cardiomyocyte differentiation, we can treat the cells with either growth factors or small molecules in the 2D culture. So typically 2 weeks after culture, you can see monolayers of cultures were beating everywhere. In a good differentiation, you can get more than 70%, even to 90% of the cells are actually cardiomyocytes. But in practice, we do see variation between cell line to cell line. To overcome this problem, we have established a very simple method to enrich cardiomyocyte through formation of this 3D cardiac construct. We call them cardiac spheres. So this was done through a microscale technology. What we've seen here is that the 3D culture can generate more than 90% of cardiomyocyte from a 2D culture that has only less than 40% of cardiomyocytes. We also, so this is how the spheres look like. You can see all of them are beating nicely. We also found the 3D culture can improve the structural maturation of cardiomyocytes. As you see in here, the cardiomyocytes from the 3D culture, almost all the cells have this nice clear pattern of muscle striations. However, the 2D culture seem to have mixed populations. So compared with the 2D culture, the 3D culture definitely have more cells with a higher level of structural maturation. since there's many advantages of using 3D cultures, and similarly, the microgravity can have profound effect on cell behavior. So we decided to combine these two technologies and see this can improve cardiomyocyte differentiation. So to turn stem cells into cardiomyocyte, the cells have to first go through cardiopregenter cell stages. So we speculate if we simulate proliferation and survival of progenitor cells, we should be able to get more cardiomyocyte at the end. So to do this, we generate cardiospheres from early stage of cardiopregenter cells, and then we put them into, expose the cells to simulate microgravity using a random position machine. So what we've seen here is the simulate microgravity culture generate almost up to 99% of cardiomyocytes with about 90% of viability. So both the cell purity and viability were higher than the parallel cultures of both 2D and 3D cultures under standard gravity. So the cardiomyocyte yield from the simulate microgravity culture was about 5-fold higher than the 2D culture and about 1.5-fold higher than the 3D culture under standard gravity. These cells express appropriate cardiomyocyte markers. The similar microgravity also increase the expression of several genes associated with cardiostructure proteins and calcium handling protein. We also found the simulate microgravity culture have improved calcium handling capacity, as shown here, the fast calcium transit, which is a feature for more mature cardiomyocytes. And these cells have expected electrophysiological properties. We then asking why the simulate microgravity can give us more cardiomyocytes. And here we're looking at the cardiac inductions by a time course experiment, looking into the expression of many markers associated with the cardiogenesis event. We've seen the expression of these markers follow the pattern of what we expected for cardiogenesis. So interestingly, simulated microgravity increased expressions of markers associated with both cardiopregenter cells as well as the later stage of cardiomyocytes, suggesting the simulated microgravity actually increased the induction of cardiomyocytes at early stages. We also found simulated microgravity in 3D culture increased the proliferation of cardiopregenter cells. So we named the NKX positive cardiopregenter cells. There are more cells express Ki67, which is a marker for cell proliferation. We also seen higher level of expressions for other markers associated with cell cycles and cell proliferations. And they also have higher capacity to incorporate EDU, which is another indicator for cell proliferation. Interestingly, this cell population has more viable cells. The simulated microgravity have more than 90% viable cells, which at the end give us more cardiopregenter cells. This population also express higher level of markers associated with cell survival, such as surviving heat shock proteins and phosphatase AKT. So what I have shown you here is simulated microgravity and 3D culture can increase the induction of cardiac differentiation. That at the end, that we got more cardiomyocytes which have higher purity and higher cell viability. And the cells have appropriate molecular and functional characteristics. With that, I would like to thank members from my lab, particularly Rajnish Jha, who have been leading this simulated microgravity project. And I also wanted to thank my collaborators from Emory, Georgia Tech, and Children's Hospital, and my funding sources from CASIS, NIH, and AHA. Thank you. No questions.
We have time for questions after each presentation this morning. Are there any questions for Chinwar, or do you want to hold them till the end? Okay, thank you, Chinwar. So the next presenters will be Dr. Peter Lee and Dr. Arun Sharma. They are co-principal investigators in an investigation that was led out of Dr. Joseph Wu's lab at Stanford University. Peter?
Great. Well, good morning. First of all, Arun and I would like to thank the organizers for giving us the opportunity to speak to you today about our experiment. An experiment that flew aboard the International Space Station this past summer for a month, and the data are still in process, but we'd like to talk about what led up to the project and about how the project was carried out. So we were looking at the effects of microgravity on stem cell-derived heart cells or cardiomyocytes, and also we looked at cells that came from a diverse patient population. As many of you know, spaceflight or microgravity environment affects deleteriously most systems in the body, whether it's the immune system, the musculoskeletal system, neurovestibular system. Fortunately, the cardiovascular system is not immune to these effects. And what we know from animal studies and human studies is that there's an increase in cardiopulmonary volume, alterations in cardiovascular control systems, decrease in cardiac work, and also an increase in cardiac atrophy and arrhythmias. And we expect that these effects and changes would become even more pronounced as you look at going beyond Earth orbit in long-duration missions, such as going to Mars, where a mission could last 2, 3, or even more years, where the radiation exposure is considerably higher, and also the astronauts are increasingly isolated, where there's minimal to no access to advanced medical care and therapies. Probably just as importantly as knowing how these spaceflight affects astronauts in order to help and treat them is also the fact that we know a lot of what happens to astronauts appear to mimic an accelerated aging process. And so that has an impact in the cardiovascular system, as we know that cardiovascular disease is the #1 killer in the United States, and heart disease alone has a higher mortality than all cancers combined. So what we expect and hope is some of the results and the data that we acquire from spaceflight studies will have applications for patients on Earth. As I mentioned earlier, most, if not all, of what we know about the cardiovascular system in space have been derived from human studies or from in vivo animal models. As you may all know, there are certain limitations to such models, including the fact that animal models exhibit interspecies differences in cardiac physiology and gene expression. And in an attempt to try and address these differences, people have looked at using human cells, and in the case of cardiac cells, looking at primary human cardiomyocytes. Unfortunately, these are notoriously difficult to obtain and maintain in culture for extended periods of time, making practical investigations very difficult, if not impossible. Others have attempted to take non-cardiomyocytes and transfect them with cardiac ion channels in order to mimic a cardiomyocyte phenotype. But again, these models are also quite imperfect in terms of mimicking cardiac function. As we all know, with stem cell technology, that has really revolutionized the field and provide a lot of advances and opportunities. Unfortunately, there are limitations with embryonic stem cells, as you all well know, mostly with access to these cells and also the associated ethical issues. iPSC's technology has really revolutionized the field, and now we have access to cells from almost any individual, and we can take these somatic cells, whether they're from adipose cells or fibroblasts from skin samples, and they can be reprogrammed and brought back into a pluripotent state. And then they can be made into whatever cells that we would be interested in. In our case, we're interested in cardiomyocytes. One of the unique features of cardiomyocytes is that they have an ability to spontaneously contract, and they also communicate with each other just as they do in vivo. And with that communication, you can see that they contract synchronously. So with this ability to isolate cells from almost any individual expand them to a large enough size we can do a number population to do practical experiments and then convert them to cardiomyocytes. There are multiple applications, including using them for gene therapy, using them as an experimental in vitro model for disease modeling, and also for screening for drug discovery. For our experiment, we wanted to use these cardiomyocytes as a model for studying how spaceflight affects cardiac cells. So the big picture question is, how does microgravity affect the heart and heart function? And we want to look at it at a cellular level. So we had proposed taking the human-induced pluripotent stem cell-derived cardiomyocytes and sending them to the International Space Station for a period of about a month, bringing them back, some live, some fixed in flight, to see what changes occur, whether it's gene expression, in function, and morphology. Additionally, we also took sources from cells from 3 different patients from a diverse patient population to see what patient-specific effects there might be. So I'm going to pass this on to Arun, who's going to talk to you a little bit more about the experiment design itself and some preliminary data.
Thanks, Peter, and thanks to the organizers for, again, having us here today. I'll talk a little bit about our experimental timeline and the setup that we use to address this question of the effect of microgravity on function of human iPS-derived cardiomyocytes. So we started off taking a tissue sample, either blood or skin biopsy, from 3 individuals representative of a diverse population. This process took about 1 month. From there, we reprogrammed these samples to induce pluripotent stem cells, a process that took approximately 2 months using non-integrative sendiviral reprogramming methods. We differentiated these populations into cardiomyocytes using a chemically defined differentiation protocol that takes about 4 weeks to produce and to purify these myocytes. And hopefully you can see this, but basically we had 2 sets of samples, 1 set of iPS cardiomyocytes that was sent to the International Space Station for culture aboard the space station for 1 month. These cells were grown with a lot of help from Dr. Kate Rubens, who was the astronaut on board the International Space Station, who is helping us with this particular experiment. These cells were imaged using live microscopy, and there was also a sample of cells that was preserved in RNA preservation reagent for downstream analysis after the cells returned to our lab at Stanford University. And in parallel, there was also a set of ground controls that were undergoing the exact same conditions that the space-flown samples were experiencing as well. And so currently we are in the process of conducting a lot of downstream analysis on these space-flown cells and these ground controls. Again, we are in the process of conducting gene expression analysis to see at the genetic level what is happening to these cells in response to extended microgravity exposure. We are also conducting contractility analysis and calcium imaging to better understand the effect of microgravity on cell contractility and the ability to beat. Also, immunofluorescence analysis and structural analysis to see if microgravity has an effect on altering the cell structure. And again, these analyses are being done on both the space-flown and ground control samples. So this is our general experimental setup. We collaborated with Bioserve Space Technologies, which is an engineering company based out of the University of Colorado. They designed for us a custom 6-well biocell cell culture system. You can see here that it's in large part a replicate of the standard 6-well cell culture system that a lot of us use in our laboratories. But the difference is this particular system is fully enclosed, and that way you can control for the microgravity of the environment, so the media does not, leave the cells and float around in space. So it's a fully enclosed system with individual ports allowing for isolated media exchange. And these biocells were cultured in a plate habitat, or PHAB, which allows for the temperature control and carbon dioxide control aboard the station. So during our SpaceX CRS-9 mission, as I mentioned, astronaut Kate Rubins, who is also a cell biologist in her other career, conducted weekly nutrient exchanges, microscopy, and cell preservation on these iPS cardiomyocytes for the 1-month-long duration of this mission. These are just some images of the mission. The first in the top left is the launch that occurred in mid-July. And also the other images are of Kate conducting light microscopy on these iPS cardiomyocytes, conducting nutrient exchanges, and observing the cells for alterations in cell shape, size. So just some preliminary data. This is an image, this is a video of the iPS cardiomyocytes as they were contracting aboard the International Space Station. We were really fortunate to have received continuous feedback and continuous information from Kate as she was examining these cells, including these high-resolution videos. So as you can see, they are able to contract and function normally, relatively normally, aboard the International Space Station. This is at day 20 day 20 in flight. Additionally, as I mentioned, we had a sample of cells that was returned to our laboratory for further analysis, and this is day 5 post-flight. And as you can see, the cells continue to contract. We are conducting additional analyses, but we can say there were some changes in the way the cells contracted aboard the space station, but in large part, these changes reverted back to normal as the cells returned groundside. These are some iPS cardiomyocytes that were stained with a calcium indicator dye, which will allow us to conduct further contractility analyses and further evaluate the role of calcium cycling, or the role of microgravity on calcium cycling of individual iPS cardiomyocytes that were flown to the station. And so, as I mentioned, a number of our analyses are still in progress, including the gene expression of space-flown versus ground-controlled iPS cardiomyocytes, the contractility and the force output of the iPS cardiomyocyte in response to microgravity, immunofluorescence and structural analyses of myocyte proteins on the cellular level. And one thing that we're also very interested in is to see if there would be any patient-specific differences between different iPS cardiomyocytes derived from different patient populations in response to microgravity. And so I would like to thank, again, the organizers for having us here today. I'd like to thank our Stanford team, led by Dr. Joseph Wu, our collaborators at NASA, including Kate Rubins, and Bioserve Space Technologies for helping us design the equipment for this particular experiment, as well as our funding sources, including CASIS, the American Heart Association, and the NIH. Thank you very much.
And we have time for questions for Peter or Arun. Okay, thank you. Thanks, Arun. Next, I'd like to bring up to the podium Mary Kearns-Junker. She's a professor at Loma Linda University, and she's currently in the process of developing her experiment for the International Space Station. Her experiment will be launching to the International Space Station in the next year. Mary?
It's a pleasure to be here today to tell you about some of our ongoing work looking at the effect of microgravity on cardiovascular progenitor cells. Stem cell-based treatments have the potential to be beneficial for patients of all ages with cardiovascular disease. Our laboratory is interested in islet-1 positive, c-KIT positive cardiovascular progenitors that co-express additional markers that are expressed on early cardiovascular mesoderm. These cells reside within the heart itself. Islet-1 expressing cells contribute substantially to cardiac development and may therefore represent an optimal candidate for cardiac therapy. Neonatal cardiac progenitor cells have an enhanced capacity for repair and can be distinguished from comparable adult cardiac progenitor cells by their microRNA and gene expression profile. We isolate the cardiac progenitor cells from human patient samples, from both neonates as well as from adults. We bring the cells into a single cell suspension, as you can see here. We clone them at less than a cell per well. We expand these clones. We select for the clones of interest. And then by about 3 weeks later, we can have enough for transplantation and further studies. And we're stuck. Okay, okay, we're back. Okay, I don't know why it's not working well. That's really strange. Okay. Well, let's see if we can go back. Okay, here we go. The cardiac progenitors, as I mentioned, are positive for c-KIT and ISL1. You can double label them. They express CXCR4. They also express brachyury, MESP1, and PDGF receptor alpha. This puts these cells in the realm of the common myocardial progenitor cells by virtue of what they express, and we're interested in them because they can be differentiated both in vitro and in vivo into cells of all cardiovascular lineages. So in our early studies using simulated microgravity and a clinostat that we obtained from NASA, we've shown that microgravity enhances a stemness in neonatal cardiac progenitor cell population, but we don't see this in the adult population. So after 7 days in simulated microgravity, we see enhanced stemness. We've looked at the microRNAs that are expressed differently in the neonatal versus the adult progenitor cells, but we haven't yet identified the mechanism by which enhanced stemness occurs in the neonatal cardiovascular progenitors. And so I'll tell you a little bit about this today. So the objective of our current work now is to identify the gene expression and signaling pathways that are altered by microgravity in the human neonatal cardiac progenitors. This application could be very beneficial for patients on Earth because if we can perhaps enhance the stemness and associate that with enhanced regenerative capability, that could be very beneficial for patients in need of stem cell-based repair. Additionally, the new information may benefit astronauts returning from long-duration missions that may impact cardiovascular function. So our methods, as I mentioned, were to use a two-dimensional clinostat. We load the cells into an opticell, and we maintain the cells there for 7 days. We analyze immediately for using flow cytometry and PCR, so they are not maintained at normal gravity anytime afterwards. Under simulated microgravity, we've noticed that we get an upregulated expression of transcription factors that control cardiac stem cell fate. There's an upregulation of MESP1 in the neonates, shown here in yellow for microgravity exposed cells versus controls in blue. And there's an MESP1, if you're not aware, is a master regulator of multipotent cardiovascular progenitor cell function. We also have an upregulation of GATA4, which is expressed in early precardiac mesoderm. We do not see this effect in the adult cardiac progenitor cells. Looking at some of the microRNAs, if you'll notice here on the right, we have the cells that were exposed to microgravity, and this is the neonatal progenitors, compared with these cells that were maintained under normal gravity conditions. And there were a number of microRNAs that were significantly impacted. And very interestingly, several of these have been shown to be downregulated, as they are here in microgravity, in situations where we have enhanced regenerative ability. So for example, microRNAs 99 and 100 is reduced during regeneration in zebrafish. The microRNA 15 family modulates neonatal cardiac regeneration in the murine model, where there's a small window where there is enhanced regenerative ability, only in the neonates, it's lost later on. And microRNA LET7 plays a role in regeneration as well when it's downregulated, as it is here. So we're very interested in that. We want to know a little bit more about what these microRNAs might be targeting and what that might tell us. So we used gene targeting software, and we could see that the Rho GTPases and the Wnt genes were the most likely to be targeted, as shown here. And we looked further into that and found that the most likely signaling pathways could be the MAP kinase, BB signaling, and the Wnt signaling pathways, and that's assessed by the number of microRNAs that are impacted and the number of genes affected. ERBB2 signaling, very interestingly, also promotes cardiomyocyte dedifferentiation, proliferation, which we do see in our model. We have enhanced proliferation in the neonates, not in the adults, and it plays a role in neonatal heart regeneration. So we looked at this a little bit more, and we delved a little bit into the Wnt signaling pathways. And so, as you remember, Wnt signaling can be either mediated by the canonical or the non-canonical Wnt pathways. And so initially we looked at the canonical Wnt pathway. We found that beta-catenin is downregulated and GSK3B is simultaneously upregulated, suggesting that it's not the canonical Wnt pathway that's activated. Looking at the planar cell polarity pathway, the small rho GTPase, CDC42, and rho A are downregulated, as would be expected by the activation of the genes that we see here. And you can see this here, verified by gel electrophoresis in the neonate. So the PCP pathway is not being activated. But very interestingly, we have an upregulation of Wnt5A and Wnt9A of the non-canonical Wnt pathway. And you can see this on the gel here for the neonates. Looking further into this, we would expect that if the transcripts in the non-canonical Wnt calcium pathway were activated, we would see an upregulation of phospholipase C and protein kinase C in the NFAT-C3, as you can see here by this pathway. And in fact, we do see upregulation of these transcripts in neonatal cells that are exposed to the microgravity, but not the adult progenitors. If PKC is elevated, then we would also expect to see an increase in the level of GATA4, which I showed you previously. We do see that. And then we were interested in looking at MAP kinases, glycogen synthase kinase, and TGF-beta, which would also then be increased if this pathway was activated. So we did gene expression plates, looking at the signaling pathway, which might tell us whether or not MAP kinases are activated. And so these are the neonates shown here. These are increased in expression, and these are the adults where these genes are not increased in expression. We can show that there is increase in the level of the expression of genes involved in the activation of ERK-MAP kinase and in the transduction of ERK-MAP kinase, as shown here. So the yellow, again, is the cells exposed to simulated microgravity, and the blue is control. We also see an elevation in expression of targets of the ERK-MAP kinase pathway, shown here. And you can see this here on the gel. These are the control, and these are the simulated microgravity exposed cells. So if the non-canonical Wnt pathway is activated, as we see, then we would show KC elevated, ERK-MAP kinase is elevated, JUN-FOS and ELK are elevated, as I showed you, and we would see an increase in early transcription factors, which we in fact see. 3 of them are shown here, OCT4, brachyuria, and MESP1. We also see a downregulation of NKX 2.5, which is later on in differentiation. And so we're looking at the very first stages, the initial steps. And so what we see is there's some gene expression changes here that are associated with dedifferentiation, and then enhanced proliferation after that. And interestingly, in the other models, again, where there's an induced ability to repair, Wnt5A and 9A are induced in neonatal cardiac response to cryoinjury, and TGF-beta is involved in the epithelial to mesenchymal transition, which is also facilitatory for this interaction. We sought to look at then FSTL1, and FSTL1 expression promotes cell cycle entry and partial dedifferentiation of cardiomyocytes during cardiac repair in other models. And very interestingly, transcripts for FSTL1 are induced under simulated microgravity in the neonates. And similarly, polo-like kinase 1, which is a transcript that's essential for the cardiovascular cardiomyocyte regeneration in the zebrafish model, is also induced quite high in the neonatal cardiac progenitor cells. And so what we see is a partial dedifferentiation. So if you look here, these are our control cells. These are the cells exposed to microgravity. And if this is a heat map from low to high level expression, and if these are the genes that we're looking at, these are HES-derived cardiomyocytes, we just back up a couple of days from something like this, shown here, to something like this, shown here. And in many cases where there is enhanced differentiation, you see the dedifferentiation, you see enhanced proliferation, and then you see improved regeneration in other models. At this point, what we know is we just can tell you that there's a activation of the non-canonical Wnt pathway in these neonatal cardiac progenitors. I can say that the gene expression profile suggests a partial differentiation early on in the neonatal cardiac progenitor cells exposed to simulated microgravity. And whether or not that's associated then with a enhanced redifferentiation as in shown in the other models and more efficient regenerative ability has yet to be seen. So they back up a little bit to go forward more efficiently. And that's what it appears at this moment. Well, we're looking forward to our future studies on the International Space Station, and what that will do will allow us to compare the effect of microgravity in the space environment on these cells, and to tell us a little bit more about what we might expect in that environment. And so I'd like to acknowledge the help of the people in my laboratory, the cardiothoracic surgeons who provide our samples, radiation biology, Mike Picot. We're working with BioServe for our upcoming flight on the ISS, and our funding for this project has been entirely from CASIS. And thank you.
Any specific questions for Mary? Okay, thanks, Mary. I'd like to next bring to the podium Dr. Abba Zubair from the Mayo Clinic in Jacksonville, just up the road. And in Dr. Zubair's talk, you're going to hear that the effects of microgravity on stem cells is not just limited to cardiogenic progenitors. There are effects on cells of all different types and all different sizes that are responding to changes in their physical environment. And in some cases, those changes lead to an increase of stemness, as you've already heard, and that, of course, enables the potential for utilizing microgravity or simulated microgravity to not only better understand the pathways that are involved and the biomarkers that you can use to track that maintenance of stemness, but actually leads to the possibility of being able to utilize what you understand from those changes to actually proliferate cells that have higher densities and better therapeutic effect at the end. And I think Dr. Zubair's talk is going to touch on that potential a little bit. Dr.
Zubair. Thank you very much for the opportunity. So my talk is, or our project really focused on application. So how can we take stem cells that are grown in microgravity environment and treat patients? Are the cells safe? Can we use it without any concern? So my lab focused on stroke. Some of you know there are two types of stroke. Hemorrhagic stroke, which is usually when you have bleed in your brain, and it's associated with hypertension as well as other conditions. And then there is the ischemic stroke, which is associated with a clot in your brain. And there's a lot of studies looking into the ischemic stroke and seeing how stem cell can be used to treat patients with ischemic stroke. So we focus on the hemorrhagic stroke. We use a model that has been used by other investigators to study whether actually stem cell, specifically mesenchymal stem cell, can have an impact in alleviating some of the hallmarks of stroke pathology. That is ischemia, which is lack of blood supply, lack of oxygen, and glucose, which is what the brain cells need. So basically, we had this modular chamber that we can flush out all the air in and replace it with nitrogen. So basically, we creating ischemic lack of environment or lack of oxygen. And then we grow our cells without glucose. So we have these cell lines that are neuroblastoma cell lines that we stress with lack of oxygen and lack of glucose. And now we expose them to or mix them in the presence of mesenchymal stem cell. And what we found is that, yes, if you look at the figure here, trying to figure out anyway, okay. If I take you through quickly, these are our cells that have they have not been stressed, so they've grown very well. And then here in the bottom are the cells that have been stressed but not exposed to mesenchymal stem cell. And then this line is the cells that have been rescued by mesenchymal stem cell. You can see an improvement. And when we look at their cell viability, again, you can see the viability is very good in the control cells, and then really bad in the ones that are stressed with lack of oxygen and glucose. And then when we mix this with mesenchymal stem cell, you see improvement in cell viability. The same with apoptosis. The rate of apoptosis is improved with addition of mesenchymal stem cell. So that gives us some hint that while this maybe mesenchymal stem cells could play a role in rescuing or helping in treating stroke. While what happened in vivo, if we click on the rats that were treated with media, this is rats that have been subjected to stroke, and you can see, and this stroke is specific to a specific side of the brain. You can see that one arm is not being used, so it shows that this animal had a stroke. If you click on the rats that have stroke and are treated with 1 million mesenchymal stem cell, by the way, they don't like to move that much, so you have to be patient. But you can see the rat in the jar. They use in both arms. So this tells us that, and this is a way to quantify the effect of asymmetry, meaning if you have stroke, you will see high asymmetry. And then if the rats that were treated, you can see very minimal asymmetry. And this is another group of rats that were treated with per million mesenchymal stem cell. It's quite interesting to see that the 1 million stem cell actually tend to do better, suggesting that it's not dose related, meaning giving more cells doesn't mean that you get better response. And we're not the only one that observe this phenomenon. So this is a reminder to all of us that are in the field that giving more cells doesn't necessarily mean you get a better response. We have to identify the right amount of cells that would have the maximal effect. So after going through this, we are in the process of developing a clinical trials with as-grown mesenchymal stem cell to treat patients with recent hemorrhagic stroke. To do that, we have to grow the cells. And based on our studies, as you have seen, we need at least 1 to 2 million mesenchymal stem cell per kilogram. That translates to 100 to 200 million cells. Now, to grow that is quite challenging. If you look at the options that you have, clearly there aren't very good options. Mostly the way we grow them is in using tissue culture flask, using either single layer or multi-layers, and it takes time. To grow just enough cells for, let's say, 10 patients may take up to a month. So clearly we have to find a way to do that. Obviously there are new technologies using automated bioreactors that could expedite the expansion process, but it's not a good answer, because again, still it takes time. Now, why is it so difficult to grow stem cells? Because the way nature engineered them, naturally they keep their number so that you don't overgrow. And when there is need for them to replenish a damaged tissue, you just divide and still maintain their number in the exact same way. So that's what we call asymmetric cell division. Now, the challenge is how can we stimulate them to grow? So we thought of maybe gravity, being that it impacts on all our human physiology, maybe gravity can expand, increase their proliferation rate. And from the previous speakers, you have seen that clearly microgravity does impact on cell proliferation. So we proposed to CASIS to provide funding to ask simple questions. Can microgravity expand stem cell, specifically the mesenchymal stem cell that we have established in our lab that we already tested and we are about to treat patients with. And then if microgravity can expand, help to expand stem cell, what is the feasibility of growing clinical grade cells at the ISS and bring them back so that we can use them for clinical application? Basically, we're looking at the logistics, the sterility of the products, and all that. So more on a practical basis. And then the next question is, what exactly, are they safe? I mean, by just growing in microgravity environment, is their physiology change? Specifically, we're concerned about transformation. Do they become malignant? Obviously, we don't want to give cancer cells to patients. More specifically, we're looking at cell proliferation in the microgravity compared to Earth, similar to what Dr. Sharma has explained to you a little bit, studying how the cells grow and behave in space, and then we have a ground-based control to compare. We're going to look at their morphology, and look at their, how they interact and whether they form clumps or tumors or they grow in a single way. So we will study their morphological characteristic. We look at gene expression profiling, and then test them in our model, in our animal model, to see whether they can actually function and alleviate the symptoms of stroke. So because, again, the opportunity to go to ISS is so limited, we don't want to just go with one type of cells, mesenchymal stem cell that we've been studying. We want to take also hematopoietic stem cell, which is a non-adherent cells compared to mesenchymal stem cell that grow as adherent cells. We also want to take cancer cell lines and look for the cancer stem cell behavior as well, all to maximize the opportunity of going to the space station. Just like Dr. Lee's group, we're working with BioSab, and we're using similar biocells that they have used in their project. So we're working with a well-established implementation partner that have supported multiple projects. We are also working with Quest Orion to develop education module for high schools to entice them and get them involved into space research. We envision we're going to have either real live imaging of the cells as they grow at the station, or semi-real, where the images are captured and sent within a day or two. And there are several high schools around the world that would be studying them and looking at different, their number, morphology, aggregations, shape. We don't want to ask them or give them too much guidance. We want to ask them to look at the cells and come up with some observation just to stimulate their creativity. So these are some of the images that we've taken using the biocells in the preparation for the flight, which we hope would be either in January or February. So the ones that you have seen is hematopoietic cells. These are myeloid leukemia cells, and these are the mesenchymal stem cells that they grow. You can see they are all quite different. And we wonder how they will look when they are growing at the station. And these are the hematopoietic stem cell. I don't know if I have time. 2 minutes, all right. So while we are waiting, we've done a couple of experiments. One of the questions we ask is, can we go back to the, because I have 2 minutes. I think, well, go back again. One more slide. One more. Sorry. I told them if I don't have time, they should go to the last slide, and I think they quite go back one more. Oh, maybe I can do it here. All right, that's good. So we collaborated with a company called Terminal Velocity. Obviously, going to the International Space Station is quite challenging. It takes, it's only one bus that goes there, and there are a lot of passengers. So you have to be in the queue. So obviously we need to have a way of getting there. So this company developed a capsule, and they envisioned that the transportation of materials from the station or from another planet can be run by this unmanned or capsules that could be sent to the Earth or from us to the other side. So basically we evaluate whether a stem cell will survive re-entry into the atmosphere in that capsule. So this is the capsule, and it was launched by using a balloon. So we did this experiment in Oregon, and you can see it's a short flight. The capsule went up to like 100,000 feet and then comes back. Supposedly it should be done within like an hour or two. Well, that was a mishap, that the capsule could not be found. It was lost in a desert in Oregon, but it was found later. These are some of the images of the capsule with one of that containers contain our cells, and this is how they load it, and then it was launched. And then after like 7 days, we were able to get the cells back, and the cells are growing very well compared to the cells that we kept in the lab, and they are able to differentiate into either bone or fat, just as one of the properties of mesenchymal stem cell. So clearly that this, especially mesenchymal stem cells, are quite resilient, and they can be transported using this type of capsule. We also did some parabolic flight to evaluate the effect of microgravity on stem cell. Some of you may not know the parabolic flight, but basically it's a period between microgravity and hypergravity. So we had to improvise, use several simple materials, and then we transport the cells, and then we bring them back to the lab, and we show that the cells are also fine. More interestingly, for CD34 positive cells, which is a marker for hematopoietic stem cell, we see about almost doubling of the cells, meaning the microgravity may actually stimulate expansion of the cells. However, we see a suppression of differentiation, meaning these are markers for T and B cells, myeloid cells, and you can see their numbers went down as well. So clearly, other options to simulate microgravity is out there, but we need a long-term microgravity condition, real microgravity like that of ISS to conduct our study. So I would stop here. These are the people in my lab, the people that we have collaborated. Thank you.
We're running up against the next session. We have time for maybe one question from the audience. Okay, so I hope with today's panel you now understand the possibilities that are open to you for innovation, utilizing the International Space Station and other devices and other opportunities that enable you to utilize microgravity as a new environment to enhance your research going forward. If you have any questions for any of the panelists up here or for CASIS to find out ways for you to take your research into space, please find us afterwards. And I'd like to take this opportunity to thank our panel one more time. Thank you.
out this morning. We've got a reasonable crowd here. So my name is Franklin West, and I'm the moderator for this session. I'm from the University of Georgia Regenerative Bioscience Center. And so today's session is about stem cells and their potential use to help people with neurological diseases, disorders, and injuries. Unfortunately, there's too many neurological diseases and disorders to cover all of them. And so what we have done is we've actually compiled a group of preeminent scholars in the field to talk about some of their work. And so this morning we're actually going to hear from 4 individuals to talk about their experience and also some of their research. And so we only have an hour, so I don't want to spend too much time talking. So I'm going to go ahead and invite Dr. Jean Loring to the podium, and she's from Scripps Research Institute. Thank you for coming, Jean.
Thanks, Frank. Okay, I'm going to talk to you today, hopefully in not very much time, about a therapy for Parkinson's disease that we've been working on for around 5 years. We're hoping to have our first patients treated in 2 years. The faces that you see on this screen are of the people who are actually going to be treated by this therapy. The idea is to use cells, make induced pluripotent stem cells from individuals who have Parkinson's disease, to turn those into dopamine neurons, which are the cells that die in Parkinson's disease, and then to transplant them back into their brains. So I'm going to try to go quickly through this, and unfortunately this is what I'm going to be doing. I'm going to talk about the idea, the progress we've made, the plans we have, and mostly I want to talk about the group that has supported this research from the very beginning. We couldn't have started this project without these people who have raised money for the first few years before we could get grant support. Okay, so I have to say this because this came up again last night. There are 2 kinds of stem cells, so you can't just talk about stem cell therapies. You have to specify whether what you're talking about is taking fat out of somebody and putting it back in their bloodstream or putting it somewhere else in their bodies. That is the kind of stem cell or stem cell therapy. They may not even be stem cells, actually, we'll find that out later. This is the kind of therapy that clinics that are unregulated by the FDA, but should be regulated by the FDA, are doing all over the country. There's more than 500 of them. And there are several lawsuits right now. There's one against a company in San Diego. It's a class action suit against one company for false advertising, for saying that the therapy that they're offering was 100% effective for all of the diseases that they listed. And the patients are saying it was 0% effective for any of the diseases that they treated. And they charge $15,000 for each therapy. Okay, so these adult stem cells that come from fat, they're called mesenchymal stem cells, or they're called adipose cells. Some of these preparations are capable of making cartilage. They certainly can make cartilage in culture. It's not clear that they can make cartilage if they're transplanted before they differentiate. They can't make nerve cells, they can't make heart, and they can't make anything else. Okay? The cells that can make everything are called pluripotent stem cells. And they were first derived as embryonic stem cells from discarded embryos. This is from humans. The first ones derived from mice were in 1981. So this field is actually older than you think. The cells were derived from 5-day-old discarded embryos from IVF clinics, and these are called embryonic stem cells. The first publication on the human variety was in 1998. But in 2007, another source of cells, the same pluripotent stem cells, was developed by Shinya Yamanaka in Japan. He won the Nobel Prize for this work, I think deservedly, in 2012. And it involves taking skin biopsies from volunteers or other cell types from volunteers, and then converting those cells using some transient expression of a few genes. These cells are called induced pluripotent stem cells, and they have the same capabilities as embryonic stem cells. So some people prefer to work with embryonic stem cells, some people prefer to work with induced pluripotent stem cells. It depends on the application, but you can use either one. They have the same capabilities. And this shows how they are made from culture of fibroblasts at the top. They're reprogrammed, and they change. It's very easy to tell when a cell has been reprogrammed because they change shape dramatically. So this is not a really difficult thing for people to discern. The cells have changed. Over on the far right bottom is the, and that is not my pointer, so just ignore it. On the far right is some markers that demonstrate that these cells are, in fact, what we say they are. They're pluripotent stem cells. This is the way that I like to show, to point out and help people remember that these cells can give rise to every cell type in the body, and I do mean every cell type in the body. The only thing holding us back is the development of methods in order to make those cells in a culture dish. So some of these are easier to make than others. The cells I'm going to talk about, today, are the dopamine neurons for Parkinson's disease. We're in the upper right-hand corner. But there are also methods in development for multiple sclerosis therapy, for liver disease, for muscular dystrophies, for heart disease, for pancreas, for type 1 diabetes, and for macular degeneration. And in each case, the cells that are transplanted are the cells that have been made into the cell type that dies. It's a cell replacement therapy. It's not a stem cell therapy. We don't put stem cells into people. We use the stem cells to make cell types that we put into people. This is the idea behind the Parkinson's disease therapy. Parkinson's disease is caused by the death of a specific cell type in the part of the brain called the substantia nigra. It's pretty small. It's indicated there in the figure on the left. These cells send signals to another part of the brain called the striatum, and they control all the control over movements. So when people are diagnosed with Parkinson's disease, they've already lost at least half of these cells. So what that says is that you can't regenerate these cells in the brain. If you're going to get people back to the way they were, you're going to have to replace them. Drugs that have been developed to rescue these cells or keep them from dying off are extremely useful. It would be great to be able to freeze the disease so it doesn't get any worse. But if you want to restore people to health, you have to put more cells in. So this is the idea. We have a group of patients. We have made induced pluripotent stem cells from them, and then we've developed methods for making the same dopamine neurons that are lost in their brains in a culture dish. We have a reason to believe this will work because there were some experiments done in the 1980s and 1990s using fetal tissue as a transplant to restore dopamine neurons in Parkinson's disease patients. These very courageous researchers actually did clinical trials that were supported by the NIH. And what they found with 74 Parkinson's disease patients, they took cells from fetuses that are about 6 to 8 weeks old, which are really, really small, and they dissected out an even smaller part, and they had to put several tissues from several embryos into each side of the brain. So you can imagine this is challenging. It's much more challenging in the US now because of the abortion laws. It is, however, being reproduced right now in the United Kingdom under funding from a European Union grant. So there are people right now trying to reproduce the same methods that were used in the 80s and 90s. The problem with this therapy was lack of control over the outcome. In some people, there was no change. In a remarkable population, people experienced a reduction in their Parkinson's disease symptoms, which lasted for the rest of their lives. So they were essentially restored to normal with no disease pathology. However, in about 15% of the patients, there was a phenomenon called a dyskinesia. You never want to make patients sicker than they started out. The dyskinesias caused uncontrolled movements, and so that had to be treated separately, and it was clearly because of the transplant. So the reason for that is almost certainly the fact that there were other cell types besides those dopamine neurons transplanted into the brains of these patients. And when you transplant cells that aren't supposed to be there, then you can have some unexpected effects. So interestingly, this is an example of a patient who died 24 years after he had the therapy, after he had the transplants. Over here on the left is a series of pictures that show parts of the brain containing cells making dopamine. These are fetal cells that arrived in that part of the brain 24 years ago. So as you can see, they're perfectly healthy. The cells are sending out axons and connecting with the rest of the brain, which is obviously the reason why those cells have reversed the symptoms. And over here on the right, you can see a diagram of what happens after transplantation. In the green line, you can see the reduction in the Parkinson's disease score, which is a measure of the effects of the disease. And in the circles, the black line shows the increase in dopamine in the brain that occurs over time. So these transplants don't work immediately. There is an effect immediately because of the surgery, but that wears off, and then the effect becomes greater and greater with time. Okay, but we learned from these. These studies were discontinued around 16 or 17 years ago because they couldn't control the dyskinesias. So the quality control issues we learned about was the fact that you needed to use multiple fetuses. That meant each one of them had to be dissected to this tiny little bit of brain. You had to get the right part, and you had to put multiple fetuses in each person's brain. You had to get it right. And there aren't very many people in this world who know how to do this. So the fetal tissue is variable. Some of it is more viable than others. It depends on what its history is and how long you have to keep it before you transplant it. And there was no control over what types of brain cells you were transplanting and how many you were transplanting. You couldn't predict in advance. And then there was the unpredicted adverse outcome, which is dyskinesia. And of course, now, in 2016, there are complex legal and ethical issues associated with the use of aborted fetuses. So this is the idea that we have. This is to essentially do the same thing, but instead of using fetal cells, we're using dopamine neurons that are derived from the patients from their induced pluripotent stem cells. So the advantage of this is really quality control. Everything is about quality control. We can know before we put the cells into the brains what kinds of cells they are. We know whether they're the right kind of dopamine neuron. We can count how many we put into somebody's brain, so we can do it consistently from patient to patient. And we're not going to put in the neurons that are believed to cause dyskinesias. So we can avoid some of the serious issues and also have much more control over the whole concept, the whole procedure. And there are no ethical issues with using a patient's own induced pluripotent stem cells. So we don't have to worry about abortion clinics anymore. But there's another bonus, and that is that the cells match the patient, so we don't need to immunosuppress people. One thing I think we all have to keep in mind is if we put cells from one person into another person, that's going to cause rejection no matter where you put them. And so those people have to be immunosuppressed. And that also means that we can give these cells again because they won't build up an antibody reaction or an immune reaction to the original transplant. So if we need to give somebody more cells later, we should be able to do that without any problems. Okay, this is just to illustrate the kinds of quality control we've built in this system. This is not what FDA recommends. This is far more than what FDA recommends. We have both classical markers that we analyze the cells at 3 different stages, actually 4 if you count the fibroblasts. We look at them when they're IPS cells. There's an intermediate phase, which is a developmental phase that's en route to the cells becoming dopamine neurons. We look at the cells once they've become immature dopamine neurons. That takes 25 days in culture. That's not such a long time. And we also don't need that many cells. The number of cells that we need for each patient can be cultured in one well of a culture dish. So we don't have to expand the cells, which is important because once you start expanding cells, you start accumulating genomic mutations and epigenetic changes that are probably not desirable. We have, in addition to our classical assays, we're also developing a set of genomic diagnostics, which include looking at the gene expression signature, the epigenome, the whole epigenome of the cells, and we look at the sequence by whole genome sequencing. So at each one of these stages, we're going to make absolutely sure that those cells have not accumulated any mutations, that they're the right cell type. And then after the cells have been, we can keep them in culture longer and completely mature them in a culture dish. This would happen in the patient. And then we can do electrophysiology on them and show that they are, in fact, dopamine neurons from the right part of the brain. So we have a lot of control over this process. And this is an example of what these cells look like in culture. This is an antibody staining the cells for tyrosine hydroxylase. As you can see, there are lots of cells that are positive for this marker. Also, you can see that they haven't sent out really a lot of neurites. We want that because if we try to associate cells that are well connected with each other, they die. So we had to pick exactly the right time. Okay, so we've shown that these cells will recover, they will cause recovery in a rat model of Parkinson's disease. On the top, you can see that the top line is the control, and you can see it's flat. The reduction of those other three the other three lines shows that 3 different patients had the same amount, the same effect in this rat model. And I've also shown you here pictures from the rats, which have the 3 different patient neurons. Okay, so the progress we've made is to generate the induced pluripotent stem cells from, we started with 8, we now have 10 to 12. We've turned them in reliably. We've developed methods for getting the same dopamine neurons every single time. That has been done by Andreas Bratt-Leal in my lab. He's the director of this project. We had our first meeting with the FDA earlier this year. It's called a pre-IND. We plan to initiate the trial in 2018, assuming we have continuous funding. In 2016, we were awarded a grant from the National Stem Cell Foundation, which allowed us to start safety trials. And over the last 2 years, we've been awarded 3 CIRM grants, most recently one on Monday of last week, that will allow us to, from CIRM, California Institute for Regenerative Medicine, which will allow us to continue on the pathway towards FDA approval. Now I want to just mention Summit for Stem Cell, and then I'm going to close. The goal of Summit for Stem Cell was to use a patient's own cells to treat their disease. And it works because it involves the patients and the advocates in the science. The patients and advocates are in our lab learning about what we're doing. It started in July of 2010 when a physician, a neurologist, Melissa Hauser, and Sherry Gould, her nurse practitioner, came to visit me at the Scripps Research Institute and asked me whether there might be a stem cell therapy for Parkinson's disease. And we spent a short time talking about it. It was so obvious and so simple, but we didn't have funding. So in 2011, Sherry Gould took a group of people to the top of Mount Kilimanjaro, including Parkinson's patients. And this allowed us to launch our project because they raised $300,000 with that. This is why it's called Summit for Stem Cells, because there's a series of summits that these advocates and the patients achieve and have achieved over time. We've had lots more fundraisers since then. They've climbed to the base camp of Mount Everest, and they just got back from a trip to Machu Picchu, a trek to Machu Picchu. These are Parkinson's patients who are doing it. So there's a sense of community and we are also part of that community as scientists. They raised more than $2.5 million to support this project for the first 5 years, and we have thousands of donors worldwide. Let's keep going. So as I said, the main players here, Jennifer Robb is here, and she is the president of the foundation. She has Parkinson's disease. Sherry Gold and Melissa Hauser are the physician component. We're the research component. Andres Prat-Leal is working with both Summit and in my lab, in the stem cell lab, to develop these neurons. And now I'm going to just finish with this. This is a movie which is a little bit outdated now, but I think it gives you a really good feeling for what the people in this group are doing. Would you play this, please? Yo, guys back in the booth. Thank you. Oh, that was not what I asked for. Okay, go all the way to the end. And you should be able to just do that clicking thing on the little arrow. And if you can't, we're not going to see the movie. Yes, there you go. Just click on there. Thank you. And there should be sound, which there isn't. Can you make sure we have sound? Please. Okay, so read these people's lips. They're talking about their how they had. These are the patients. They're talking about how the therapy or how they discovered they had Parkinson's disease. No sound? You want to pause it and see if you can get sound? Thank you. You might want to turn up the sound on that computer because that might be muted, and that might be the problem. You want to try that? You want to check on my computer to see whether the sound is on? Just push that little button that has the... There you go. Still doesn't work. Okay.
A friend of mine who has been diagnosed with Parkinson's, I started checking the list.
Thanks. Sorry.
Well, it all started with a little stiffness in my left hand.
And they noticed my movement was decreasing.
Even before the tremor showed up, I was very depressed.
In the early 2000s, I started losing my sense of smell.
When I compared symptoms with a friend of mine who has been diagnosed with Parkinson's, I started checking the list and saying, I've got that too and that too.
We both cried.
Dr. Hauser is the person that told me that definitively that I had Parkinson's.
Parkinson's disease almost uniformly progresses.
What I understand about Parkinson's is there's no cure.
The neurons in my brain that produce dopamine are dying off.
When your symptoms manifest themselves, as mine did several years ago, 80% of those neurons are gone.
I control the symptoms by taking some medication several times a day. I'll need more medication as time goes down the road.
When I understood about some of the side effects, I was not very excited about that.
The best hope that I've researched and have heard about is the pluripotent stem cell procedure.
It seems like magic, but it is science. The great thing about pluripotent stem cells is that they can give rise to any cell type in the body.
Sherry Gould, my nurse practitioner.
That's it? We should probably quit that anyway. I think that captured the essence. Thank you.
Thank you, Jean. So you can see we have some truly passionate researchers working on this project. So I would like to go ahead and invite the next speaker up, Miss Emily Baker. She's actually from my laboratory, and she's going to talk about some exciting work we've been doing looking at stem cell therapies in the pig model.
Good morning. My name is Emily Baker, and I'm a research assistant in the Regenerative Bioscience Center at the University of Georgia. And the title of my talk today is Transplanted-Induced Neural Stem Cells Differentiate in the Post-Stroke Brain, Promoting Tissue Recovery in a Porcine Model. So everyone in this room probably knows someone that's been affected by stroke. Every 40 seconds, someone has a stroke, and every 4 minutes, someone dies of a stroke. Our lab chooses to focus on ischemic stroke, which accounts for 87% of all strokes, and results when a blood clot blocks the flow of blood to a certain region of the brain, resulting in the loss of that tissue. There's only one approved treatment for ischemic stroke, and that's tissue plasminogen activator, which goes in and breaks up that blood clot to restore blood flow. There's only one treatment for ischemic stroke, and on top of that, there is no regenerative therapies to replace tissue that's already been lost. Our lab chooses to focus on induced pluripotent stem cell-derived neural stem cells because we think it would be a great treatment option for ischemic stroke. Rodent studies have shown that these cells are able to modulate inflammation, promote angiogenesis, and differentiate to mature neural cell types, possibly leading to cell replacement. So the ultimate dream would be with this cell technology is to have a patient with ischemic stroke isolate adult cells from this patient and introduce reprogramming factors to induce pluripotency. Then we would differentiate those iPS cells to neural stem cells for transplantation back into the patient. This would theoretically be an autologous cell therapy and would avoid the risk of immune rejection. So the West Lab uses the pig as a model to develop these INSC treatments. Historically, studies of this nature have been developed in the rodent before moving to human clinical trials. However, over 700 drugs have gone to clinical trials for ischemic stroke, and there's only been one FDA-approved treatment. And this suggests that there may be a translational gap between the rodent and the human. And that's not entirely surprising due to the anatomic differences between the rodent and the human. The rodent brain is lissencephalic, meaning it has a smooth cortical surface and contains less than 10% white matter. On the other hand, the human brain is gyrencephalic, meaning that it has those gyri and sulci and those convolutions on the cortical surface and is greater than 60% white matter. And that's not to mention the size differences between the brain of the human and the brain of the rodent. The rodent brain is 650 times smaller than the human. We think that the pig would be a great model to bridge the gap between the rodent brain and the human brain because the pig brain is gyrencephalic and also greater than 60% white matter and only 7.5 times smaller than the human brain. So this leads us to our overarching hypothesis, which is that transplanted INSCs will produce regenerative trophic factors and undergo differentiation, leading to cellular and tissue functional recovery in a pig model of ischemic stroke. So to briefly go over our experimental design, stroke surgery was induced on day 0 through middle cerebral artery occlusion. And then 1 day post-stroke, we performed magnetic resonance imaging, or MRI. 5 days post-stroke, we came in and injected 2 injections of 6 million INSCs, or vehicle only, and there were 4 animals per treatment group. And then after injection, we did MRI 1 week post-injection, 4 weeks post-injection, and 12 weeks post-injection. And then after our last MRI time point, we collected tissues for histological analysis. So the MRI sequences that we ran were T2 FLAIR, diffusion-weighted imaging, diffusion tensor imaging, magnetic resonance spectroscopy, and perfusion-weighted imaging. We also collected cell transplantation sites to assess what the neural stem cells were doing differentiation-wise after transplant into the pig brain. We tracked our cells using the human nuclear antibody, or HNA, because we were transplanting human cells into the pig. We used the NUN antibody to assess neurons, GFAP for astrocytes, and OLIG2 for oligodendrocytes. So moving into our results, we first wanted to confirm that we had a true ischemic stroke. So here you're looking at MRI images taken 24 hours post-stroke. And so our T2 FLAIR shows us a hyper-intense region in the right cerebral hemisphere that's hyper-intense that leads to, that shows us that there is a brain lesion. DWI, the corresponding DWI images show a hypo-intense region, which is indicative of stroke. And then the corresponding ADC maps show a hyper-intense area, which indicates cytotoxic edema and a true ischemic event. So next we wanted to assess whether or not our neural stem cells had an effect on brain swelling or atrophy after stroke. And so what we did was we took the ipsilateral hemisphere, or the affected hemisphere, and compared it to the contralateral or unaffected hemisphere. So the graph that you see here is comparing the ipsilateral hemisphere to the contralateral hemisphere, where 0 is baseline. So 24 hours post-stroke, we see a significant increase in the size of the ipsilateral hemisphere, indicating brain swelling, and we see this in both our treated and our non-treated pigs. 1 week post-transplant, we begin to see brain atrophy, and then 4 weeks post-transplant, there is a significant reduction in that brain atrophy in our treated, in the black bars, compared to non-treated, which is the red bar. However, by 12 weeks post-transplant, that difference became less apparent, and there was no difference between treated and non-treated pigs. So this tells us that neural stem cells do not prevent brain atrophy, but it may slow the progression of that atrophy. So next, we wanted to determine if neural stem cells led to improvement in white matter integrity, and we did this through diffusion tensor imaging by measuring fractional anisotropy. So 0 is baseline, meaning comparing to the contralateral side, and we were assessing change in FA, or fractional anisotropy. So 24 hours, 1 week, and 4 weeks post-injection, we saw no difference between treated and non-treated animals in the change in FA. However, by 12 weeks, we saw that there was a significant reduction in the change in FA between 24 hours post-stroke and 12 weeks post-transplant, which tells us that the neural stem cells may be helping improve white matter integrity after stroke. So next, we used our spectroscopy data to quantify changes in neurometabolite abundance after stroke and treatment. So here we're looking at change in NAA, which is a healthy neuron marker, and we saw that 24 hours post-stroke and 1 week post-transplant, we saw no difference between treated and non-treated groups. However, by 4 weeks post-transplant and 12 weeks post-transplant, we saw a significant reduction in that change in NAA compared to the non-treated animals, and there was a significant reduction between 24 hours post-stroke and 4 and 12 weeks post-injection. We also looked at the cellularity marker choline and found a similar trend that by 4 weeks and 12 weeks post-transplant, there was a significant reduction in that change in choline in the ipsilateral hemisphere compared to the contralateral hemisphere. We also looked at the brain energy marker creatine and found that there was no difference between treated and non-treated animals 24 hours post-stroke and 1 and 4 weeks post-transplant, but by 12 weeks post-transplant, there was a significant reduction in the change in creatine compared to non-treated animals. So we also looked at our perfusion-weighted imaging data and assessed whether neural stem cells had an effect on cerebral blood perfusion after treatment. And so here the graph has changed a little bit, and we're looking at the 100% mark, where 100% would mean that the contralateral and the ipsilateral hemispheres are equal in cerebral blood perfusion. The two parameters that we looked at were mean transit time and time to peak, which are both related to the time it takes for the blood to move through the cerebrum. And so 24 hours post-stroke and 1 and 4 weeks post-transplant, we did not see a difference between treated and non-treated animals. However, by 12 weeks post-transplant, we saw a significant reduction in both mean transit time and time to peak, which indicates that neural stem cell treatment could be leading to the blood moving through the cerebrum faster by 12 weeks post-transplant. So I mentioned earlier that we also took our transplantation sites and assessed INSC differentiation in the pig brain. And so we did that through the use of the HNA antibody. So here is our HNA antibody, and the white arrows are labeling HNA positive nuclei, which points out our transplanted cells. We co-stained this with the neuron marker NUN and found that a large portion of our transplanted cells co-express NUN. So this is a higher magnification image showing our HNA positive nucleus co-localized with NUN, the neuron marker, and then the merge here. We also wanted to assess if our neural stem cells were differentiating to oligodendrocyte sites. And so we see that a portion of our HNA positive nuclei are co-localizing with the oligodendrocyte marker OLIG2, and then we see the merge here. So we wanted to quantify the percentage of HNA positive cells that were either co-localized with NUN or OLIG2. And we found that about 75% of our transplanted HNA positive cells were also co-localized with NUN, and about 25% of our HNA positive cells were co-localized with OLIG2. It's also worth mentioning that we stained for the GFAP astrocyte marker, and due to the nature of the GFAP stain, we were not able to confidently identify GFAP positive, HNA positive cells, so we chose not to include them in the quantification. So our conclusions from this is that neural stem cell treatment promotes brain tissue recovery through improved white matter integrity, improved neurometabolite abundance, and enhanced cerebral perfusion. Additionally, our neural stem cells are able to survive long-term and differentiate predominantly into neurons. So I'd like to take a second to thank my PI, Dr. Franklin West, and the rest of the West Lab, our funding sources, and our collaborators. Thanks.
Thank you, Emily. Great talk. So next, I would like to introduce Dr. Dilip Yagaval. He's a good friend from the University of Miami Miller School of Medicine, and he's going to talk a little bit about some of his research in humans and large animal models.
Thank you, Franklin. So I thank Bernie for inviting me to talk about the work we've been doing at the University of Miami on intra-arterial approaches to delivering cells in stroke. And what I'd like to cover in the next 10 to 12 minutes is how close are we to implementing this approach in patients. These are my disclosures. I don't have any patents on any of these cells or devices. We've been very fortunate to be funded with intramural grants and Florida state grants and federal grants. And the work is based on a fantastic team that I've been fortunate to have, including the small animal team. Dr. Bowan Lin is a senior researcher, and Dr. Ami Raval and my postdoc Pallab Bhattacharya, and the large animal team, some of whom are here, Dr. Watanabe. And the approach we've taken is to focus on the translation of delivering cells intra-arterially so that we can quickly use those findings to bring to early clinical trials. So one of the first things that is being made clear by the previous speaker is that the need for novel therapies for ischemic stroke is quite high. We actually now have a second approved therapy just as of 2015, which is endovascular treatment, which is what I do clinically, where we go up from the groin femoral artery all the way up into the occluded artery in the brain, and remove the occluding thrombus with a very strong efficacy with now about 60% of patients achieving independence if they get reperfusion with this therapy within 6 hours. However, despite that progress, what we see is even in the most recent trials of thrombectomy, almost 80% of patients are not able to return to their job. And the 40% who are not independent clearly need novel therapies. The development, however, is great because now we are able to open arteries to get cells to where they may need to be delivered using the intra-arterial approach. So the progress that's already there may actually accelerate cell therapy, especially with intra-arterial delivery. Our cells have, from the beginning, from 2009, been the mesenchymal stem cells. And what you can see is that these spindle-shaped cells are variable in size, and they can be as large as 50 microns. And I think that's where we focused our energy in the beginning, as to see how we could deliver these cells safely intra-arterially, because if the cells are big in size, they by themselves can occlude small vessels and perhaps cause ischemia. Just to take a step back, the idea that Dr. Loring presented is very relevant to our work. We see our work as a cell-based therapy where we are not trying to replace neurons with adult stem cells or glia, but we are really looking at giving a boost with trophic mechanisms to the endogenous repair that occurs after stroke, and also neuroprotection and anti-inflammatory action from these cells. As many studies have shown, these cells actually get phagocytosed in a few weeks and are thrown out. But the trophic action is what leads to benefit. The work in this field actually has been accumulating over 15 years, where several studies have shown that giving cells intraarterially, the adult stem cells, MSC specifically, can lead to benefit. And in fact, early clinical trials were done as early as 2000, not with MSCs, but other cells, and there has been some gap until about 2-3 years ago, where again the early clinical trials have started. What our contention is that as compared to IV delivery, which is obviously the most widely available, IA delivery, which is somewhat more invasive but much less invasive than direct transplantation, has several advantages. The biggest one is that it circumvents systemic circulation and trapping of cells in the lungs and the liver, and many more cells get into the brain, into the injured area, as compared to IV. And what you can see in data from Dr. Chop's lab in Michigan is that when you give IA cells, you have an immediate, almost immediate 4 hours distribution of cells in the ischemic hemisphere. That's the top panel. Whereas with the intracisternal and IV delivery, it takes several days for these cells to get there. These cells are ion tagged, so they're the dark spots. And with IV and IC delivery, the distribution is also very limited at about 3 or 4 days. So the ability for targeting a large number of cells to the area is terrific for intraarterial delivery. A similar result from Dr. Steinberg's lab in Stanford, where IA delivery led to cells immediately going to the brain, whereas IV delivery led to cells being trapped in the lungs and the liver in a stroke model. What we did in my lab is to look at what happens to these cells at 24 hours after IA delivery. And we took the slice of cortex and used a technique called 3D microscopy. Can you play the movie, please? And looked at where these cells go, and these are GFP labeled cells. And what you see is the cells are not only there in the dark areas, which is the stroke brain, but they are on the surface of the arterioles or small capillaries. And if you concentrate on some of the green dots, you can see them on the surface of the vessels and in the brain parenchyma area where the stroke has caused destruction. That is certainly very encouraging to see that at 24 hours, these cells delivered intraarterially do get into the area that we need them to get into. And the fact that they're on the surface of the blood-brain barrier and in the parenchyma suggests that the process is most likely diapedesis that leads these cells to get into the injured area. The timing of delivery still remains to be definitively worked out, but given the mechanisms of action of neuroprotection, it makes sense to give these cells early, and there are data to show that earlier delivery lead to benefit from other labs and from our lab, and I'll show you that in a second. When we started, the biggest challenge for intraarterial delivery was posed by a study from Hopkins that showed, from Johns Hopkins, that showed that intraarterial delivery of 1 million cells in a stroke model actually leads to ischemia in the middle cerebral artery territory in a large number of animals. And certainly that would be a no-go for translation of this to cells. So our study contended that this challenge to intraarterial delivery may be dose-dependent. So what we did was a dose de-escalation experiment where we decreased the dose from 1 million cells in the rat stroke model to 5 million to 500,000, 200,000. At 100,000, that problem of ischemia was completely mitigated. And so when you decrease the dose of cells given intraarterially, they do not lead to ischemia at the maximum tolerated dose. And that, when seen histologically, shows you that even at 100,000, these cells are big and sticky, and they do form these clumps inside vessels. But when you look at the cross-section in the middle, they are both inside and outside the vessel. And the last panel shows a cell half in and half out, consistent with diapedesis. And so we then took the 100,000 dose, which is the reduced dose, which is well tolerated, and did an efficacy study in the rat model. And we compared 2 time points, giving the cells at 1 hour and then giving the cells at 24 hours. And what we found was that only the 24-hour cell group had a significant benefit in their neurodeficit score at 4 weeks as compared to the other groups, including control, and surprisingly including the 1-hour group. So when you give the cells right away at 1 hour, they actually do not lead to benefit. So there is likely a need for a few hours to go by after which the cells can get into the injured area. This infarct size is reduced, and when you look at that, where the infarct size reduction occurs, it occurs mainly in the penumbral area, again consistent with a trophic neuroprotective action. So we did the similar experiment in a canine model, just like Dr. West's lab, to overcome the translational issue and to get surrogate markers like diffusion tensor imaging, similar to Dr. West's lab. And this is a endovascular stroke model where you don't have to do a craniotomy in the large animal. You take a catheter up all the way and from the groin and block the middle cerebral artery, and then have a MRI to confirm the infarct. And we give our cells at 48 hours. And what we've seen is that the treated animals have a significant reduction in their stroke size and less atrophy. And the numbers are just getting completed with our controls to have these results more statistically presentable. But with IV delivery, we have not seen similar results. What we've also been able to do, and this is a confirmatory, this is a study from a group in Korea that had similar results using the canine model, where umbilical cord cells led to a significant reduction in infarct size in treated animals as compared to controls. The controls are below. We also looked at diffusion tensor imaging. This is the neuroassessment scale that we use in the large animal. And again, what we see is, as compared to controls, we had a trend towards better improvement in the treated animals. What we're also able to do is diffusion tensor imaging, as I mentioned, where you look at the corticospinal tract at baseline after stroke and after cell therapy. And what we see is that in a control animal after stroke, the corticospinal tract size goes down, and this is tractography. It becomes very thin in the middle panel, and then it recovers some, but not completely. But in treated animals, we had 2 animals where we had a large stroke, and they recovered after cell therapy with a significant improvement, almost back to normal in the top panel in the corticospinal tract, and close to back to normal in the lower animal with the corticospinal tract. So this is pretty a powerful surrogate marker. We think that could be used in clinical trials. I'm going to finish up with showing you the first trial that I led with Sean Savitz of UT Houston, called the Recover Stroke Trial, where I forgot to mention all our lab work was with allogenic cells, but we did this study with Cytomedics, the company, and they had a IND for autologous cell therapy given intraarterially with ALDH bright cells, which are derived from the bone marrow. These were given intraarterially at 2 weeks, and the main goal was to demonstrate safety, and the secondary goal was to look for early efficacy. And what we had was a double-blind randomized trial, randomized 3 treated and 2 sham at 8 sites. The planned sample size was 100 patients, but we stopped at 48, not due to any safety concerns, but because the interim analysis suggested that if there was a difference, we would pick it up at that sample size. And safety endpoints were any neurologic worsening that may occur after giving these cells, and proportion of serious adverse events and death. And we also looked at the improvement in disability scales. And the patient was screened at 7 days, and then they were randomized, and then a bone marrow harvest or sham was done, and the cells were dosed within 48 hours of bone marrow harvest. They were not expanded. And they were given with a microcatheter using standard techniques in the intracranial ICA, internal carotid artery, just beyond the ophthalmic artery going to the eye. And we had a total of 29 patients in the treated arm, 19 in the sham arm, pretty good follow-up. And here are the results. There was no, the groups were well matched. There was no significant difference. There was no difference in the safety outcomes. None of the groups had any neurological worsening by giving these cells intraarterially. And there was one tumor that was seen in the control, in the sham arm, one brain tumor, none in the treated arm. And that was a metastasis from a lung cancer. There was one adverse event that was the DVT. DVTs were more in the treated group, 4 of them, and they were felt to be unrelated to the cell therapy. So I'm going to end there. Just in the interest of time, we did not see a difference in efficacy, and we also, thanks, Franklin. We also looked at one post hoc analysis. In 48 patients, we didn't really expect to see a big difference in efficacy, but when we looked at the 4-point change as a possible measure of worsening, we actually ended up having data on any 4-point improvements over time. And the sham arm had a 4-point improvement in the stroke scale in 30% of patients. But in the treated arm, there was about a 70% chance of a 4-point improvement. This is not a traditional stroke recovery endpoint, but still a post hoc analysis suggesting that there may be a signal of benefit with the intraarterial cell delivery of the ALDH bright cells. But to conclude about the results of the trial, the safety objective, which was the primary objective, was met, where intraarterial delivery of cells given at 2 weeks did not lead to any safety concerns, and there was a signal of a benefit. The primary efficacy endpoint was not seen, but with 48 patients, that would be hard to achieve. And the post hoc finding of NIHSS improvement by 4 points in the treated group suggests that there may be a signal of benefit. And so we have a good proof of long-term safety of this approach with the ALDH bright cells. And I'm going to skip these slides. We are proposing a larger trial to the NIH using an allogenic approach where the cells can be given earlier within 24 to 36 hours, again with the aim to have a neuroprotective and anti-inflammatory action in a larger number of patients. And we would use, along with the traditional endpoints, biomarkers such as DTI to look for efficacy. So with that, I'm going to conclude. Intraarterial delivery really is a widely available approach around the world, and it appears to be safe for delivering cells. And then several early trials have shown safety of this approach. Ours, the trial that I showed you, is the biggest trial, but there are smaller phase 1b studies that have shown that this approach is safe. And then the optimal cell type and cell timing of delivery still is being worked out, but this approach of using MSCs early on it seems to be at least one way where there is likelihood of benefit, a high likelihood of benefit, and larger studies are on the way to really prove this definitively in the future. Again, I want to thank my collaborators and mentors. Dr. Josh Hare has been a major force behind my going into this field, and several others who have helped with all the work. Thank you so much.
Thank you, Dilip. So today I've failed as a moderator at some levels. So I know Graham is shaking his head like, gosh, really, Frank? So we're over time, but I would like to say that right now is the break time, so snacks and etc. So if you're willing to forego the break and stay with us a little longer, we would appreciate that, of course. Our next speaker is Miss Brenda Castle, and she is actually from the Parkinson's Warriors group, so a patient advocacy group. And then we're going to open it up for some quick discussion because we know it's time is growing short. So gentlemen in the back, if you could turn on the microphones that are on the table, that would be excellent. Brenda?
Well, thank you, and I appreciate the opportunity to be here to speak. I am obviously, as you can tell, I have Parkinson's. But I'm also a warrior fighter of this disease on a daily basis. And symptoms started in 2009. By 2012, I got my diagnosis. At that point in time, my leg was dragging, my right arm was dragging, my face was frozen. On behalf of all the patients, stroke victims, anyone that has a neurological disease, this is the most fascinating research, and we have to keep fighting because 3 years ago, I was in a wheelchair 30 to 40% of the time. Exercise has got me to where I am today, but that's not the only solution. So stem cell, I truly believe in it, very passionate about it, fighting for everyone that fights on a daily basis. And I'm honored to be here with this panel, and I look forward to what is yet to come, experiments, trials, whatever it can be, because the patients, the brain is a very, very powerful, powerful tool. And exercise is a key. And if we can retrain our brains with the loss of dopamine that controls movement, there's so much more that we can do. So again, I appreciate the opportunity to be here and be on such a great panel.
If anyone has any burning questions, again, I know we're over on time, feel free to come to the microphone and we'll field any questions that you may have. Thank you.
It's for Jane. Why do you have to wait to 2018 if you have FDA approval?
I'm sorry, you're asking me?
Yes.
Well, we don't have FDA approval.
Oh, okay. I thought,
right. We're on our way to the pre-IND stage, and then we'll have to do the IND enabling studies. So even though it's working really well, we do have to prove to the FDA that the cells are safe, and that's the main objective of the work we're doing right now.
Safety is still the problem, okay. Yeah.
Thank you all the speakers for the great presentations, and thank you for your advocacy. I've got a question for my good friend Jean. As you know, each morning I shave with Occam's razor, so I want you to kindly explain to us why you infer that the dyskinesias are due to other cells and aren't due to inappropriate release of dopamine in the striatum.
So I failed to show the paper that demonstrates that. There has been a paper published. It's difficult to do because there's not really a rat model of dyskinesia. But there is strong evidence that serotonergic neurons that were included are responsible for the dyskinesias. It's correlative, really, and might be causative. I think the other alternative is that you just had too many neurons, but I think that doesn't really make sense given the numbers of neurons that have survived in these patients. They're not different from the ones with dyskinesia and the ones without dyskinesia. So I don't think it's just simply a matter of being overdosed with dopamine. Otherwise, I mean, it's one of those unknowns. It's an untestable at this point.
Good question. All right, well, if there's no other questions, let's thank the panel one more time, and thank you for coming.
everybody. Sorry, we're gonna get started in here. Thank you all for coming. My name is Marcel Dadi. I am from Texas Biomed. I want to welcome you to this session on the use of non-human primates in translating studies just before you get them into the clinical trial. So we have really some exciting 2 talks. It looks like they put my talk now first. We're going to talk first about about Dr. Dr. Shiva. He's going to present some studies on using non-human primate for treating myocardial infarction, which is basically heart attack. And I'm going to talk about the using non-human primate for a neurological disorder that we are developing these stem cell, and it's for Parkinson's disease. So if you were in the previous session, this is a perfect continuation of this technology for this disease. So I think I have here the slides. So Dr. Shai, I'm going to go ahead, since they have it up. I can just talk about Parkinson's disease, and then you will talk about the stroke. And then we have questions and that can be asked either between session or at the end of the session. We keep it interactive and good. So so really the main goal is to translate some basic discovery from the lab to patients that are suffering from Parkinson's disease, and I can talk a little bit about Parkinson's disease first. It's a clinically defined by a tetrad of motor symptoms, low frequency in resting tremor, rigidity, and skeletal muscle, face and hand, and also in reduced motor activity or bradykinesia in general, and also postural instability. Well, what's interesting actually now is in addition to these all these motor deficits, there is also a little bit of cognitive deficit that it's just we're starting to detect, and that's why I'm going to be talking a little bit about the non-human primates and the ability to not only monitor the motor symptoms but also cognitive symptoms. The Parkinson's disease, a little bit about the statistics. So it's about 1 million just in the United States, about 60,000 diagnosed each year. It's second after Alzheimer's disease, and it's expected to impose increasing social burden because of the aging. And and the cost is about $25 billion per year in the United States alone. So here I want to show you a little bit about how Parkinson's disease patient look like. This is a typical posture of a Parkinsonian patient, and it's drawn many years ago in 1886 by Dr. Sir William Gower in the UK. So as you can see in here, this is a stooped posture of a patient with a forward tilt. There is a rigidity and trembling in the extremities of the arms, and also a rigidity and trembling of the head. The hip and the knees are slightly flexed. There is what we call a mask-like face, which basically the Parkinsonian patient, they lose facial expression. It's really hard for them to, and then there is the arm is a little bit flexed, and also the elbow, it's the elbow, and then the arm is a little bit flexed, and there is a reduced arm swing as they walk. They don't have any more that arm swing for them to help them move forward, and the steps, they shuffle a little bit the steps. And this is really interesting. You wonder what's happening, why these patients, they show this. And it's basically because of these dopamine neurons in the brain that projects into the basal ganglia. They die slowly, and as they die, you start to see these motor deficits. So the motor deficits, it's very flagrant. You can see them, you can't miss them. The cognitive one is a little bit hard to detect, at least at the initial stage. So the standard therapy for Parkinson's disease, there is multiple drugs that actually activate the dopamine inside the brain. The main one, the gold standard, it's called L-Dopa or Cinnamide. And this drug is kind of interesting. When a patient takes it, it crosses the blood-brain barrier and it gets turned into dopamine. And when it's turned into dopamine, so all of a sudden, patient, they have dopamine inside their brain and they become active. So it works like magic. Unfortunately, as the disease progresses, the terminals of dopamine that process this L-Dopa goes away, and then they can't process the L-Dopa anymore. And what happens is you have not only that, dopamine start to get actually processed by other kind of cells, and then you start to have what we call L-Dopa-induced dyskinesias. Another approach or therapeutic approach that is used for Parkinsonian patient is the implantation of electrode into the brain. And again, this electrode, they're implanted into this area of brain called the subthalamic nucleus. And once they implant these electrodes into the brain, they stimulate the subthalamic nucleus because it's overactive. And once they stimulate this area of the brain, the patient actually, the symptoms goes away. So both these approaches, drug or stimulation, they treat only the symptoms, and the disease keeps going. So really, there is no cure for the disease. I just entered it's been going on for quite a while. The cell therapy, there is a really strong proof of concept that cell therapy works for Parkinson's disease because it's a one cell type that it's die in this disease. It's a dopamine neurons, and it is it can be transplanted into the striatum. And recently, there is a team especially from these three guys actually, Ole Lindvall, Andrew Björklund, and Patrick Brandon in Dr. Lee's lab in Sweden. They've been doing this for many years, and they have shown that a patient in here with a 24 years grafted fetal cells. So the key word here is fetal, because these cells, they're dissected out from a aborted tissue. So they go after these aborted tissues, they dissect the tissue from the midbrain, and they transplant it into patients. So as you can imagine, this is unsustainable. There is a lot of ethical issues, and there is also practical issues, because you can't really have a quality control of this tissue. So the technology that we are working on, and actually a lot of labs are trying to pursue this technology because it's so promising. There is multiple labs. Our lab, there is others in New York. If you hear the previous talk by Jane Lorraine in the Scripps, there is in UK and in Sweden. So a lot of labs are actually developing this technology, and it's just really a matter of time that we're going to put all this together and get it into the patient. So the technology is called induced pluripotent stem cell. It's discovered by Dr. Yamanaka in Japan, and also another paper came out from the Thompson lab in Wisconsin. The way it works is you take a biopsy, whether it's a skin or a blood cell. You take these guys and you put a 4 gene, and you can reprogram a skin cell. You can reprogram it into an induced pluripotent stem cell, which is a cell that have the ability to generate any cell type. So once we generate these iPS cells, we can expose them into mitogenic factors, and we can generate what we call a neural stem cell. And these neural stem cells have the ability to give rise to neuron, astrocytes, and oligodendrocytes. But also, we developed a technology over about 20 years to move these neural stem cells into the dopaminergic lineage. And these are cells that express dopamine in vitro. And I'm going to show you another slide of that. So this is a typical colony of iPS cells, induced pluripotent stem cells. This is exactly how they look. Initially, they kind of look like this, but as soon as you get the gene inside these cells, the 4 episomal factors inside these cells, they turn to become pluripotent, and they become like this one in here. And you get here a lot of cells. They look like a pancake in here, but they are amazing cells because they have unlimited ability to grow and to generate other cell type. So when you take these cells, the next thing, we differentiate them, as I mentioned, and we can stain them, and they generate these kinds of neurons stained with the red. Red is tyrosine hydroxylase, which is marker for dopamine neurons, and tubulin, which is marker for neurons. DAPI, it stains all the nuclei, which is basically all the cells in the cell culture. So this technology I've been working on for many years. It's a technology that uses actually a glial conditioned media that instruct precursor cells to become dopamine neurons. So it's a really very well-controlled technique to generate dopamine neurons. But in addition to generating the cells, some really of the bottleneck to get into the Parkinson's disease, actually, this is really can kill actually technology if you don't know how to deliver the cells. So first of all, you need to have like a relevant animal model. So that's why we use non-human primates. As I mentioned, it models both motor and cognitive aspects. And also, we are going to talk to you today about developing an MRI-guided delivery of these dopamine neurons inside the brain. So the non-human primate model of Parkinson's disease, you may ask, how do we generate this animal model? This is a well-established animal model, and the way it is established is by giving them this neurotoxin called MPTP, and when you give MPTP into the non-human primate, they develop symptoms of Parkinson's patient. And so MPTP induced the degeneration, specifically of the dopamine neurons in the substantia nigra in the midbrain, very similar, actually similar, exactly similar to what's happening into the human. And they show major symptoms for Parkinson's disease, including tremor, akinesia, bradykinesia, rigidity, and postural abnormalities. And the animal, they respond all the therapies that we use in human, including L-DOPA, and they also actually respond to deep brain stimulation as well. So they have been proven to be predictive of the drug of action in man, and they are currently really the gold standard of modeling Parkinson's disease. So, you know, if I want to develop a therapy and I want to put it in someone's brain, and if someone wants to put cells into my brain, I really would like, it's going to make me feel more comfortable to show that these cells actually work on an animal model that really mimics what's happening into the human. So addressing the safety and efficacy in a non-human primate, and if you can actually treat those kinds of symptoms in a non-human primate, you are pretty much sure that's going to work on humans as well. And so that's why we're going. I'm going to show you a little bit of an animal model that we're working with. This is a marmoset model of non-human primate, and we have this test that I'm going to describe to you. So basically, it's a box, and we put the food in here so the animal can pick either the right hand or the left hand. Can you go back one more? Can you go back one more slide, please? Can you please stick on this one? Okay, perfect. So you can see this animal marmoset. They love marshmallows, so they they go for marshmallow, and they're going to use their left arm in here to pick this one. So you can click on this one. They're very quick at eating marshmallows. So we use marshmallows. So this is a typical test for just motor use of their arm. And then if you go to the next one, so we treated them with MPTP, and you can see there is a lot of symptoms of the Parkinson's disease, and that is, you can see the rigidity in their use of the arm, and before they can pick up the food. And this one in here, you can see a typical tremor that you see in patient with Parkinson's disease. And so really our goal now, if you can actually take away these symptoms before you can get into the human, and that's actually what the FDA require. So before you get the FDA, the Food and Drug Administration require you to use those kinds of studies before you get into the human, at least in the dosing response, because there is the differences between a rat as an animal model and also a primate as an animal model. So the dosing is critical before you go to patient. There is another species that we work with. These are baboons. So these are baboons, and if you click on this, they are much more, their brain is quite similar to the human, because in this one they're going to use their left, their right arm in here to pick this piece of grapes in here, so actually, these guys, they are so smart, and one time they learn it very quickly. And one time, my postdoc, he was a little bit slow at pushing this thing up, and the monkey just pushed it up. He said, "I can help you with that." So they pushed it up, and then he picked the food. And so what's interesting about this is because the brain is so large and it has all the anatomical structure very similar to the human. You can see exactly the putamen versus part of the basal ganglia, which is the target for Parkinson's disease. You can actually actually apply exactly what you're going to use in human, like the cell dose, the exact areas of the brain, and you can also test the safety in this kind of animal model. And if you show efficacy in such an animal model, that's what the FDA want to see, and that's actually what patients want to see, because a patient is not going to take a stem cell that's not going to work. Ideally, this is a different technology from the usual, some of the stem cells that are ongoing. So cognitive and motor testing, I can tell you a little bit about that specific test that I showed you. It's called object retrieval task with barrier detour. So the food well, as I mentioned, is placed with a clear plastic box, and we have the food, and then the animal can pick either from each side, and we can change the orientation of the box 20 times. So we measure these outcome measures in here. The barrier reach, which is a reaching from the wrong side. So this is a little bit of cognitive, because if the animal can see where the food is, but he still go to the wrong side, it means its cognition is a little bit defective. The motor problem is when they reach into the open, but they can't really retrieve the food. The reaching number, we calculate how many number the animal keep going into the same place, the success and the correct performance of the movements. The hand preferences, we monitor that, they are left or right, and this is before the MPTP, if they have a hand biases. And then the awkward reach, when they start to use either, instead of using their right, they start to use their left. And there is another one which is also test a little bit the cognitive aspects of the movements, which is a perceptive response to the reaching task. And so the animal, they know that they wouldn't know, and they keep going into the wrong side. And the initiation time, which really calculate the bradykinesia, how long it takes them to initiate a movement, which is typical in Parkinson's disease. This is another test that we use. It's called activity monitoring. It's an ActiWatch, and it's an accelerometer basically, and it's record all the activities of the animal, and it's basically as very similar to Fitbit that we are using right now in human. And it gives you also, in addition, it gives you the sleep analysis, the naps that takes during the day, and the sleep during at nighttime. So this is a typical diagram of activity, a summary of the activity of the animal during the day. You can see from here from about 7 a.m. and here to about 7 p.m. the animal is very active. You can see these activities in here. You may take some few naps in here, but during nighttime you can see this is completely, the animal is sleeping. It's from 7 p.m. to about 7 a.m. in here, and then back to the activity. It's really nice to track in here all the activity during the day as well as at nighttime. And it's a software that enables us to really analyze various parts of the day, during the day or during the nighttime. You can actually measure the nap time, how many naps they take during the day, and also we can do a lot of sleep analysis or assumed sleep analysis, how long, what time they get up, what time they want to sleep, and when it starts, when it ends, and if there is any activity during nighttime, so we can monitor their sleeping habit. So from the behavioral, I am moving now into the delivery. So there is, you can have the best cell, but if you don't deliver them correctly into the right place, I see actually a lot of clinical trial failing just because of the delivery process. And this is actually a preclinical study that we did before going to patient. This is a gene therapy approach that we used using the rhesus macaques. So you see the difference between this animal in here and this animal in here. So in this animal, the therapeutic product, it's right here in the putamen, which is the codates and the putamen in here. And this animal in here, he received a same therapeutic compound, but it's infused in a different speed, in a different technique. So you can see a homogeneous distribution of the viral vector around the whole striatum, while in here we have these what we call hotspots. So when you have these hotspots, there is overproduction of dopamine in one single area. And when you have this overproduction of dopamine inside there, this animal actually started to show side effects. And one of them was dyskinesia, because it's an overspill of dopamine in a very specific areas of the brain that overstimulate the host neurons and use dyskinesia. While these guys in here, they didn't show any side effects. So the delivery can really kill your clinical study or can make it successful. So I'm going to talk to you to be careful. We are really moving very, very carefully to move this therapy to the clinic. And one of the aspects of it is how we can deliver it precisely into the areas of the brain that it's needed. And we're using this what's called a frameless device. It's an MRI guided device. It's from MRI Interventions right here actually in California. And this is the device. So it's enable you to actually, this is the trajectory where you can put your needle through to the brain in here, and you can actually change the angular of this trajectory forward or backward, or you can what we call pitch, which is left and right, and you can do also X and Y, and you can really adjust exactly the trajectory, and I'm going to tell you how you're going to adjust that. So before to be able to track these neural stem cells inside the brain, we use, this is a very well-established technique. It's been around for a long time. We use the super paramagnetic iron oxide SPO. And this SPO, which is nanoparticle, it's an MR contrast agents, get into the cells. So we incubate them with the neurons or neural stem cells, and the SPO get inside the neurons. And then these are actually an EM picture of these nanoparticles. And here they are inside the neural stem cell, these blue dots inside the neural stem cell. And then once they are inside the stem cell, this enables us to track them in vivo. So we can actually monitor them as you're injecting them into the brain. And which is a really cool technique. So this it enables you to study the dynamic of the cells inside the brain. And initially, we just wanted to see how the cells behave this is actually a phantom gel that mimic the physical and chemical, the physical properties of the brain. And here we can see, if we inject the cells at 1 microliter per minute, you see a nice distribution of the cells in here coming out into this phantom gel. And in here, if we increase actually the speed of injection, just at 5 microliter per minute, the cells, what happen is they go up actually the track, and they're coming up right here beside the track. And again, this is so simple. But if you are not careful, your cells actually is going to come up right up the track, and basically you can kiss your clinical trial goodbye, because the cells, instead you are expecting them to be in here. to target specific area and increase both the safety and efficacy. I'm saying the efficacy because some of the studies that are designed so far in clinical trial, we are learning so much of them because the neurosurgeon has to combine imaging with the delivery. If you take a Parkinsonian patient and you take a PET scan and you're going to see a dopamine, you're going to see areas of dopamine where it is actually lacking because Parkinsonian patients sometimes they don't have 100% wiped out of either the dopamine. There are some spots area that are lacking dopamine, and you want to put your transplant exactly into that area. So combining PET and MRI, you're going to really have this targeted precision for this graft into the area that it's needed. And I think I have to thank people who did some work. This is my lab in Texas Biomed in San Antonio. These are members of the lab that did the work, Roy, Kristen, Elias, and Nick, and April. It's a young lab, I guess. And this is the people, I'm sorry, it doesn't come out very well. This is the contributors. This is the MRI. It's UTASCA, UT Health Science Center. Without them, we wouldn't be able to do it. This is the actually imaging facility. So the imaging facility is in UTASCA. So we take it's been worked out very well between the National Primate Center and UTASCA. So we take the animal back and forth. So about 10 minutes away from us, Peter Fox and Jeff Clark, as well as John Lee, they do MRI. And Peter Hornsby, my collaborator in the IPS cells. These are the engineers from MRI Intervention, Shruti and Chris Conrad and Tim Gobel. They're all involved in the delivery process, and we wouldn't be able to work without this amazing foundation that helped us through and grants and as well as the NIH support for the National Primate Center. And I stop at there. Thank you guys so much. So we could take one or two questions now, or we can wait until Dr. Shiva, talk. Okay, we'll move on. And okay, next is going to be Dr. Shiva. Is going to talk about the use of iPS cells for the myocardial infarction. Thank you.
Thank you, Dr. Marty. Today, I'd like to talk about my recent work involving allogeneic transplantation of iPS cell-derived cardiomyocytes in non-human primates. Pluripotent stem cells are attractive tools for regenerative medicine because of its unlimited self-renewal. We can prepare a number of cardiomyocytes from pluripotent stem cells. Here, we add activin A and BMP4 to monolayer cultured undifferentiated ES cells. After 2 weeks of differentiation, the cells spontaneously start beating like this movie. When we transplanted these cardiomyocytes into injured guinea pig hearts, the cells survived and improved cardiac contractile function. But most of the preclinical studies are using pluripotent stem cell-derived cardiomyocytes were performed in xenotransplantation model. But there are two critical limitations because all of the studies were just performed in xenotransplantation model. First, few immunological studies with pluripotent stem cell-derived cardiomyocytes were performed. Second, the little information is available on electrophysiological consequence following transplantation of stem cell-derived cardiomyocytes. Usually in xenotransplantation model, there are substantial difference in beating rate between host and graft heart rate. For example, human heart rate is somewhere between 50 and 100 beats per minute, whereas mouse heart rate is over 600 BPM, and rat heart rate is for over 400 BPM, and guinea pig heart rate is over 200 BPM. So to better understand the effectiveness of this treatment, allogeneic transplantation study is required. For allogeneic transplantation, we selected a cynomolgus monkey as a new model. No one would dispute that monkey is a good model for preclinical transplantation study, but there are some drawbacks. First and most importantly, it is expensive. Second, care should be managed almost like humans, which means a number of people need to be involved in this study. Finally, it is hard to find an appropriate facility to perform primate work since there are only a few primate centers in Japan. To minimize immune rejection following allogeneic transplantation, we established a major histocompatibility complex mass transplantation model. Here, we established iPS cells from MHC homozygous monkey and differentiate them toward cardiomyocytes. The recipient animals are MHC heterozygous, but either of the haplotypes is identical to donor MHC. First, we established iPS cells from MHC homozygous monkey. The iPS cells express pluripotent markers by RT-PCR and immunohistochemistry. When we transplanted these iPS cells into immunodeficient mice, they form typical teratoma, including intestine, cartilage, and skin tissues, meaning they are real iPS cells. Finally, the iPS cells showed normal karyotype. Next, we generated cardiomyocytes from monkey iPS cells. We successfully generated monkey-derived iPS cell-derived cardiomyocytes by the same protocol as in human ES-derived cardiomyocytes. They are essentially positive for cardiac troponin T. This slide shows in vivo transplantation study protocol. Myocardial infarction model was produced by ischemia reperfusion injury. I'll show you this in the next movie. And animals were treated with methylprednisolone and tacrolimus, and either iPS cell-derived cardiomyocytes or just vehicle were transplanted after 2 weeks of myocardial infarction. Cardiac contractile function was evaluated by micro-CT as well as echocardiography before cell transplantation and at 4 weeks and 12 weeks after cell transplantation. Cardiac arrhythmias were monitored by Holter ECG at every 2 weeks, and 12 weeks after transplantation, the animals were sacrificed and graft cardiomyocytes were electrically analyzed by GCamp imaging. I'll show you this later, and histological analysis was performed. So I'll show you animal surgeries in this movie. A myocardial infarction model was produced by ischemia reperfusion injury. First, left coronary artery, shown here, is sutured at the mid-level. And then polyethylene tubing, shown here, was threaded with a suture. And put another tubing here, was put on the original one, and tied off the suture. And after 3 hours of ischemia, the heart was reperfused by just removing the tubings. At 2 weeks post-injury, 400 million iPS cell-derived cardiomyocytes were injected directly into infarcted myocardium and border zone as well. So after 12 weeks of transplantation, we analyzed graft cardiomyocytes by histology. As you can see, a substantial amount of graft cells labeled with GFP survived in the anterior part of left ventricle. Most of the cells are cardiac troponin T positive cardiomyocytes. The next pair of pictures shows clear sarcomere structure identified by another cardiac marker, alpha-actinin. And graft area was very vascularized identified by CD31 positive endothelial cells. But all of the CD31 positive cells are negative for GFP, indicating endothelial cells are derived from host tissue. The expression of gap junction protein connexin 43 in the graft area was relatively weak, but it's consistent with our previous work. Cardiac contractile function was evaluated by micro-CT as well as echocardiography. This movie shows base level of contraction. You can see no difference between the two groups. But when we see apex level of contraction, recipients of iPS-cell-derived cardiomyocytes show better contraction compared to PSC vehicle. So when we see longitudinal view, you can see the same difference. And the apex area, the contraction of apex area was improved by the transplantation. This slide shows quantitative analysis of micro-CT and echocardiography. Ejection fraction before cell transplantation is not different between recipients of iPS-cell-derived cardiomyocytes or just vehicle. But recipients of iPS-cell-derived cardiomyocytes show better ejection fraction at 4 weeks and 12 weeks post-transplantation. Echocardiography revealed the same trend in fractional shortening as ejection fraction. Next, we try to confirm if transplanted cardiomyocytes electrically couple with host cardiomyocytes. To visualize contraction of cardiomyocytes, we generated iPS cells expressing fluorescent calcium indicator GCAMP. This movie shows cardiomyocytes flashing in sync with their contraction. Now I'll show you how we can see host graft electrical integration in vivo. This is a monkey heart that was transplanted iPS-cell-derived cardiomyocyte 12 weeks ago. The heart was perfused with cold cardiopressure solution through ascending aorta. The right atrium was cut, and then the heart gradually stopped beating. And after stop beating, the heart was excised and transported from the vivarium to the lab. And in the lab, the heart was reperfused with thyroid solution. Then the heart started beating again. And beating heart was put on a Langendorf apparatus. This is Langendorf apparatus, and this is pumping part. And data imaging part. And data acquisition part. And GPM transients was recorded as well as ECG. And this is GCAMP imaging. You can see multiple graft area flashing in the heart. The flashing cycle is in sync with the ECG, indicating host graft electrical coupling. The graft cardiomyocytes keep coupling even after the heart rate was increased by the electrical stimulation of 3 hearts and now 4 hearts. And even 5 hearts, which is 300 beats per minute. And this slide summarizes the movie. There were 3 graft areas identified by GCAMP imaging. The 3 color traces in the lower panels represent GCAMP transients. GCAMP transients are always in sync with the ECG. These data support the idea that graft cardiomyocytes are electrically integrated with host cardiomyocytes. Now I'd like to show you the analysis of Holter ECG. There was ventricular tachycardia shown here, observed. The right graph shows fraction of animals showing ventricular tachycardia. As you can see, the incidence of VT was significantly increased in the recipients of iPSC-derived cardiomyocytes. The right bottom graph shows fraction of animals showing sustained VT, which was sustained for more than 30 seconds. As you can see, sustained VT was observed only in the recipient of iPS-derived cardiomyocytes. The left bottom figure shows duration of VT in each recipient. The duration peaked on days 7 through 14 and was gradually decreased thereafter. Now, I'd like to summarize my presentation. iPS-derived cardiomyocytes survived in infected hearts for at least 12 weeks following the MHC-matched allogeneic transplantation. Graft cardiomyocytes electrically coupled with host cardiomyocytes and improved cardiac contractile function. Although transient and probably non-lethal, transplantation of iPS-derived cardiomyocytes significantly increased the incidence of ventricular arrhythmias. In conclusion, iPS-derived cardiomyocytes regenerate the infected heart in non-human primate, but post-transplant arrhythmias need to be overcome before clinical use. Finally, I'd like to acknowledge the people listed here, and this work was supported by NIH equivalent grant in Japan called AMAID and JSPS. Thank you very much for your attention.
Please.
For Shiba, why don't you use cardiac stem cell instead of cardiomyocyte?
Why not am I using cardiac stem cells?
No. Why will it not be more efficient if you use cardiac stem cells? Because they have been identified quite some time ago, and they might perhaps integrate in a better way than the cardiomyocyte you are using. So if you use cardiac stem cells, they might integrate in a better way in the heart. I don't know how long it would take for them to differentiate, but they should go very quickly.
So actually, I have never tried cardiac stem cells. The only experience I had is differentiated cardiomyocytes from iPS cells or just EA cells.
No, but I mean with the iPS differentiation. But instead of stopping, instead of going to the last stage of the cardiomyocyte differentiation, why not using the cardiac stem cell step? Because perhaps they might integrate in a better way.
Yeah, I understand. Some people are using cardiac stem cells, but I'm not sure.
No, it's just a question. I think what you have shown is very good, but I think perhaps you might have a better integration, and for the rhythm, it might eventually much simpler because. A stem cell is usually, if it finds its niche, then it will integrate smoothly, I suppose.
Maybe you are right, but I'm not sure.
But your presentation was very good. Yeah, for Marcel, the question is, when you do the needle through the brain, is there a risk for the patient to have some, how do you say, best side effect, or is it very secure?
The injection process?
Yes.
Yeah, it's really very secure. I did mention this process, it's actually currently in clinical trial. The same device has been used by multiple institutions at the clinical trial. So they use it for taking biopsies from the brain, and they also use it to insert these deep brain stimulation. So it's really safe, very straightforward. The safety is guaranteed because of that imaging process that enable you really to see what you are doing, what you are going through as you go through.
I've been impressed by the system. I think it's very good.
Thank you. Thank you so much. So, any questions, please?
So, very nice presentations. I have a question for both of you. If you use iPS-derived cells, do you think to what extent can these cells actually mature into full-grown tissue if you apply them like this? So will they remember the reprogramming and never achieve that differentiation state?
Yeah, it's Dr. Shiba, do you want to address that question? Sure. And I'll give you my take on it as well.
The maturity of iPS-derived cardiomyocytes is a very important issue, actually. And as I've shown you, the ventricular tachycardia was observed after the transplantation. And one of the possible reasons is that the cardiomyocytes transplanted were just immature and still have fast beating automaticity. So, but actually, the cells we are transplanting is, I have to admit that it's still immature. And we are now working on how to get them matured.
Or could they mature when they are injected and see that environment, or will they never mature because they were reprogrammed?
Some papers show that after transplantation, the cells get matured in vivo condition. That's my understanding.
Yeah, so it's really the same thing for the heart and the brain, but they are slightly different iPS cells from human embryonic stem cell. Both of them are pluripotent. We see that embryonic stem cells, they're a little bit faster in maturation, reaching between the initial precursor stage to the fully mature stage. There are a few differences in vitro, a few weeks, difference couple of weeks, to become functionally, and we test the function in the tissue by making synapses. Once you put a neuron, we differentiate them to put a synapse. And once it makes a synapse, it starts to fire action potential. And that's how we know that it's functionally integrated and fully mature. But they do take them a little bit of time because they come in completely different tissue. And the technique, as the question was, whether to put something completely differentiated or maybe a little bit in the precursor stage and let the environment shape it. So we really think that these cells, a little bit precursor stage, when they go into the environment in vivo, they respond to the milieu there and they integrate into the whether it's heart is actually a very similar system or the brain. Thank you. Any other questions? Well, I want to thank everybody so much for being here today. Thank you.
still have time. Keep the music. Keep the music. Everyone, thank you so much for joining the 5th annual Japan Symposia. Industry, academia, open innovation in Japan for application of human embryonic stem cell and induced pluripotent stem cell lines. This is our 5th year of doing this symposia. We're very honored to have a tremendous team of experts from Japan. This is what we call informally the Japan Day of the World Stem Cell Summit. We understand that Japan, from its regulatory, government, industry, academia, is leading the field towards cell therapies. We have much we can do together and collaborate together, and we're honored to be able to present this to the audience that is listening to this live streamed and recorded. We hope that you will reach out to some of the speakers that you hear today. If you are a researcher interested in collaboration, commercialization, if you are working in a regulatory field from around the world, there's much that we can learn and do together. I want to introduce our moderator for this panel, Dr. Takashi Asada from Kyoto University, who is a dear friend. He personally escorted myself and my wife to some of the sites and stem cell sites throughout Japan in our visit in August that opened my eyes and helped me understand the importance of us working together in a collaborative fashion. Thank you.
Thank you, Barney, for the kind introduction. So today is the 5th time for Japan Symposium. We have 2 academia, also 5 industries attending this symposium. My name is Takashi Asada from Kyoto University. First speaker is Professor Norio Nakatsuji from Industry, Cell and Material Sciences, Kyoto University. He talked about introduction to this symposium. Professor Nakatsuji, please.
Thank you very much for taking time to attend this session. My role is overviewing the whole session and talk a little about our own research. So keyword here is academia, industry, or industry-academia collaboration, open innovation, and human pluripotent stem cells. Okay, so please allow me to introduce the whole view of our institute called ISEMS of Kyoto University. It's 10 years ago, Japanese government took a very ambitious project to make 6 or 10 international new-style institute focusing on cross-disciplinary field and internationalization. So I was a leader of the one application, and fortunately or unfortunately, it's successful. So I spent my more than 5 years to building new institute, combining cell biology and chemistry and physics. So that's led to current my research, my own research, combining iPS-derived, ES-derived cardiomyocyte cells and nanofiber materials to make a three-dimensional matured tissue-like structure. And for the stem cell concept, this cell material integration looks like this one. So it expands the capacity of the cell biology and cell engineering to the more combination with the chemical compound and material for propagation, differentiation, and three-dimensional construction and transplantation. So we are heavily working on the chemical libraries or functional materials. And although I started as an academic basic researcher, my recent interest is how to make a good application of this very attractive pluripotent stem cells to the human health and drug discovery. So then, rather than biological mechanism, our focus is something like large-scale production, manufacturing, and quality control, and cost cutting reduction. And still challenging with the functional maturation of the pluripotent stem cell-derived cells, and quality control, and cost tend to be very high. So first example of our own research is how to make large-scale production. So traditionally, pluripotent stem cells and other cells are cultured on the flat surface, like culture dish or flask. But it has obvious limitation for the expansion. So we succeeded to make aggregate culture. We call sphere culture of around 100 micron or 200 micron diameter, and just simple subculturing makes 10 times more cell production compared to the surface culture, so like this one. So this method was combined with the polymer, functional polymer, that keep the such small particle suspended without agitation or stirring. So currently, we are working heavily with the Fujifilm group to apply this project for the bioreactor of 10 liter or even higher without stirring. So that's a new concept, because stirring agitation damages this very sensitive, fragile stem cells. So that's a proof of concept of using 200 milliliter culture bag to culture like 10 to 8 or more cells. So that necessity to such a large production is obvious. So scientists, laboratory scientists think like using culture dish to cure 1 or 2 mice. But human, you need at least 1,000 times more cells for the actual application. Okay, so another aspect is, so large number of pluripotent stem cells has no use. So it should be transformed to the useful cell types. So we worked on the cardiomyocyte, and not traditional cytokines or protein application, we try to replace those by small chemical compound, by screening the chemical library. And that tends to be very successful one. It's already published. So currently, our group can differentiate cardiomyocytes to the relatively matured one, not totally matured, with 95% or more efficiency without any sorting by only using small chemical compound combination, no cytokine, no protein. So it's reduced the cost to maybe 100 times less. So that's this kind of progress should be more advanced, expanded to the affordable, realized affordable cell therapy. So that's our method is very robust. So every pluripotent cell line we tested responded very well. And another last example is yes, it's one thing to make single cells or cell sheet, but how to make three-dimensional structure, it represents body part or organs. So rather than putting the single cell layer one to one, but we made aligned nanofiber and put the cell suspension on top of the nanofiber bundle, make autonomous, spontaneous, three-dimensional aligned cardiomyocyte. And that resembles very well with our own heart tissue. So it looks like this one. So it's very easy to handle by forceps. You can handle and transplant. We are collaborating with Professor Shiba, of next speaker, and so this is patented and now licensed to the startup company. We founded a company called Stem Cell and Device. So device is a keyword combination with the stem cells. And later, another member will give a presentation about our company, but the concept is developed at Kyoto University, and almost all the scientists joined this startup company. So this is one example slide of the SCAD. Okay, so how many minutes do we have? It's okay. Okay, so maybe I'd like to add one relatively recently published our study is making Alzheimer's disease model neurons. A very different subject, but very useful application of pluripotent stem cells is making, you can make any kind of genetically modified cells. With the advancement of the CRISPR and other technology, it becomes easier and quicker. This is, our work is done with a very traditional method using human ES cells. So we introduced the patient disease causing mutation. So take one ES cell line we derived many years ago and put, so either wild type gene or mutated gene could be integrated. So that makes very nice pair of the cell line of only one gene different, others the same. And we differentiate into the neurons, and that shows actually reduced activity in synapse current. So it's shown the DPSC. The top is the wild type, and the lower is the mutated one. So that shows the neuron model of the something deficient, compromised neurons. of Alzheimer, probably. And we screened one example of the application. We screened the FDA-approved chemical compound to whether some can improve the such deficiency. And we found one or two. So that's a proof of concept of application of such cell line for the drug screening. So, this is the later part of my talk, just introducing the following talk about our government project supported by Ministry of Economy, Trade, and Industry. So it's a industry-oriented project funded by the METI ministry. So from 2010, I led one group of including a few excellent scientists, academia scientists with excellent technology-oriented Japanese companies. And I'm very proud that some of them developed a new product on the market based on our project, like Nipro, Liprocell, Takara Bio. And the current project is still going on. It's a rather ambitious project of manufacturing, starting from the cell stock, either ES or iPS cells, and expand to the 10 to 8 or 9s using the already developed Nipro company's automatic bag culture system. And then next step is 10 liter or 100 liter of the suspended sphere culture carried out by Fujifilm, currently in progress. And also differentiation induction, changing the system or culture media and make 10 to 10th or 10 to 11th cells of cardiomyocyte or neurons or other cell types. And the quality control system we are working on, even packaging and transportation system, like using the temperature controlling devices. So that's my brief introduction to the whole session. Thank you very much for your kind attention.
Thank you, Professor Nakatsuji. Question and answer, we have some less of the time to produce. So next speaker is Dr. Yuji Shiba from Shinshu University. He talked about from the preclinical transplantation study of iPS-derived cardiomyocytes for cardiac repair. So Dr. Shiba, please.
Thank you, Dr. Asada, for the introduction. So before starting my presentation, I'd like to take a moment to introduce Matsumoto City, where our university is located. So Matsumoto is surrounded by Japan Alps and famous for its beautiful view of mountains. And this is Matsumoto Castle, which was built over 400 years ago, and now one of the oldest castles that ever exist in Japan. And the right picture shows my favorite place, Kamikochi National Park. And I just went there, and as you can see me here. So let's move on to today's topic. The primary purpose of the study is to cure heart failure caused by so-called heart attack. The right figure shows, the right top figure shows survival rate of patients diagnosed with heart failure. As you can see, most of the patients died within 10 years. For these patients, heart transplantation is usually last resort, but usually only effective treatment. The right bottom figure shows number of heart transplantation worldwide. As you can see, the number of heart transplantation is somewhat on the decline for the past 20 years. So new treatment for heart failure is required. One of the promising treatment for heart failure is cardiac regeneration. So far, many cell types were tried, including bone marrow cells, skeletal myoblasts, mesenchymal stem cells, or resident cardiac stem cells. Surprisingly, most cell types have been shown to improve cardiac contractile function, at least to some extent, in both preclinical and clinical studies. But now we know that transplanted cells are not transdifferentiated into cardiomyocytes in vivo. They just work via paracrine effect. But our goal is brick and mortar regeneration using cardiomyocyte and make them pump in sync with the host heart. So I started out this project in 2008 when I was in Seattle. This picture was taken when I was there. There were 3 Japanese here in this picture. Ichiro was at bat, and Matsuzaka in Boston Red Sox took the mound, and I was here again. So pluripotent stem cells are attractive cell sources for cardiomyocytes because of its unlimited self-renewal. We add activin A followed by BMP4 to monolayer cultured undifferentiated ES cells. After 2 weeks of differentiation, the cell starts beating spontaneously like this movie. When we transplanted these cardiomyocytes in injured guinea pig hearts, the cell survived and improved cardiac contractile function. Most of the studies using pluripotent stem cell-derived cardiomyocytes were performed in xenotransplantation model. But we really don't know the immune response when the cells were transplanted into the same species. So allogenic transplantation is required to better understand the effectiveness of this treatment. For allogenic transplantation, we selected cynomolgus monkey as a new model. And no one would dispute that monkey is a good model for transplantation study, but there are some drawbacks. First, it is expensive. And second, care should be managed almost like humans, which means a number of people need to be involved in this experiment. And finally, it is hard to find an appropriate facility to perform primate work, since there are only a few primate centers, at least in Japan. To minimize immune rejection following allogenic transplantation, we established MHC matched transplantation model. Here, we established iPS cells from MHC homozygous monkey and transdifferentiate them toward cardiomyocytes. The recipient animals are MHC heterozygous, but either of the MHC haplotypes is identical to donor MHC. First, we tested if this MHC matched transplantation system inhibits immune rejection in vitro. The upper panel shows in vitro mixed reaction. When 2 lymphocytes from 2 different animals were co-cultured, they identify different MHC and proliferate, like the second bar in the bottom graph. But when we co-cultured lymphocytes from MHC homozygous and heterozygous monkey, the proliferation was inhibited. Next, we generated iPS cells from MHC homozygous monkey. The iPS cells express pluripotent markers by RT-PCR and immunohistology. When we transplanted these iPS cells into immunodeficient mice, they form typical teratomas, including intestine, cartilage, and skin tissue, meaning they are real iPS cells. Finally, the iPS cells showed normal karyotype. Next, we generated monkey cardiomyocytes. We successfully generated monkey cardiomyocytes from iPS cells by the same protocol as in human ES-derived cardiomyocytes. They are essentially positive for cardiac troponin T by immunohistochemistry and flow cytometry. Sorry for the busy slide, but it shows in vivo transplantation study protocol. We did MHC mismatched and matched transplantation study. For MHC mismatched study, myocardial infarction was produced by ischemia reperfusion model. And animals were treated with methylprednisolone and tacrolimus. And after 2 weeks of myocardial infarction, 400 million iPS cell-derived cardiomyocytes were transplanted And animals were sacrificed after 4 weeks of transplantation, and histological analysis was performed. For MHC mat transplantation study, a myocardial infarction model was produced, and animals were treated with the same protocol. And either 400 million iPS cell-derived cardiomyocytes or just vehicle were transplanted, and cardiac contractile function was evaluated by micro-CT as well as echocardiography before cell transplantation, and 4 weeks and 12 weeks post-transplantation. Cardiac arrhythmias were monitored by Holter ECG at every 2 weeks, and animals were sacrificed after 12 weeks of transplantation. And graft cardiomyocytes were electrically analyzed by GCamp imaging. I'll show you later this, and histological analysis was performed. And histological analysis revealed that only a tiny amount of graft cells labeled with GFP survived with significant infiltration of inflammatory cells in MHC mismatched model. Many of the inflammatory cells are CD3 positive T lymphocytes, suggesting grafts were acutely rejected. On the other hand, in MHC matched transplantation model, a substantial amount of grafted cells survived without significant infiltration of inflammatory cells, even after 12 weeks of transplantation. So we further analyzed the graft cells by histology. As you can see, substantial amount of graft cells labeled with GFP survived in the anterior part of left ventricle. The most of the graft cells are cardiac troponin T positive cardiomyocytes. And this pair of pictures shows clear sarcomere structure identified by another cardiac marker alpha-actinin in the graft area. And the graft area was very vascularized identified by CD31 positive endothelial cells. But all of the CD31 positive cells are negative for GFP, suggesting endothelial cells are derived from host tissue. And the expression of gap junction protein connexin 43 in the graft was relatively rare, but it was consistent with our previous work. So now, the cardiac contractile function was evaluated by micro-CT. So this movie shows the base level of contraction. There was no big difference. But sorry. Can you start the movie? Oh, thanks. So in the base level, there was no big difference in contraction between recipients of iPS cell-derived cardiomyocytes or just vehicle. But when we see apex level of contraction, the recipients of iPS cell-derived cardiomyocytes show better contraction. When we see longitudinal view, we can see the same difference. The contraction of apex level is better in iPS cell-derived cardiomyocytes. This slide summarizes the movie. So ejection fraction before cell transplantation was not different between recipients of vehicle or iPS cell-derived cardiomyocytes, but recipients of iPS cell-derived cardiomyocytes showed better contraction at 4 weeks and 12 weeks post-transplantation. Fraction shortening by echocardiography revealed the same trend as in ejection fraction. Next, we try to confirm if transplanted cardiomyocytes electrically coupled with host cardiomyocytes. To visualize contraction of cardiomyocytes, we generated iPS cells expressing fluorescent calcium indicator GCamp. This movie shows cardiomyocytes flashing in sync with their contraction. So when we transplanted these cardiomyocytes into in vivo, and you can see graft cardiomyocytes flashing in sync with the ECG, indicating host graft electrical integration. The graft cardiomyocytes keep coupling even after the heart rate was increased by electrical stimulation. Now on 4 Hertz, which is 240 beats per minute. and even 5 Hertz. This slide summarizes the movie. There were 3 graft areas identified by the GCAM imaging. The 3 traces in the lower panel shows GCAM transients, and GCAM transients are always in sync with the ECG. Finally, I'd like to show you the analysis of Holter ECG. There was a ventricular tachycardia shown here observed. The right figure shows fraction of animals showing ventricular tachycardia. The incidence of VT was significantly increased in the recipients of iPS cell-derived cardiomyocytes after cell transplantation. The right bottom figure shows sustained ventricular tachycardia, which was sustained for more than 30 seconds. The VT was observed only in the recipients of iPS cell-derived cardiomyocytes. The left bottom figure shows duration of VT in each recipient. The duration peaked on day 7 through 14 and was gradually decreased thereafter. Now I'd like to summarize my presentation. iPS cell-derived cardiomyocytes survived in infarcted hearts for at least 12 weeks following MHC-MAST allogeneic transplantation. And graft cardiomyocytes electrically coupled with host cardiomyocytes and improved cardiac contraction. Also, transient and transplantation of iPS cell-derived cardiomyocytes significantly increased the incidence of non-lethal VT. In conclusion, iPS cell-derived cardiomyocytes regenerated the infarcted heart in non-human primate, but post-transplant arrhythmias need to be overcome before clinical use. So thank you very much for your attention.
Thank you, Dr. Shiba. From the industrial talk, first is Mr. Takuya Yokokawa from Fujifilm Corporation. He'll talk about Fujifilm's related medicine. Please, Yokokawa.
Thank you very much. Hello, everyone. I'm Takuya Yokokawa from Healthcare Business Development Office of Fujifilm Corporation, Japan. The Fujifilm is known as a photographic film manufacturing company, and actually still we continue to make it, but the business volume of that is very small nowadays. I used to be the photographic R&D person, and for 20 years I had developed various types of photographic materials. Then I moved to the pharmaceutical R&D, and then now the regenerative medicine business office. So today I'd like to share with you about Fujifilm's regenerative medicines. This slide shows the pharma and regenerative medicine business structure of Fujifilm Corporation. Central part is Fujifilm's technology platform such as compound design, analysis, nanotechnology, and image diagnosis, and quality control system of manufacturing, and collagen technologies, all from photographic development and manufacturing and its relating technologies. To establish medical treatment business, we Fujifilm had added 3 groups of operational companies. One is a small molecule farmers, Toyama Chemicals, Fujifilm Fine Chemistries, Fujifilm Farmers, and the right side of biopharmaceuticals, such as Fujifilm Diosense Biotechnologies, Fujifilm RI Pharma, and Perocell's Proteomics, and also the Fujifilm Kyowa Kirin Biologics. And third one is regenerative medicine-related companies, the Japan Tissue Engineering and Cellular Dynamics International. I'll comment some details a little bit later. And also, the Fujifilm Pharmaceutical and Healthcare Research Laboratory and Regenerative Medicine Research Laboratory. That is the Fujifilm's original basic research organizations. And this slide shows the Fujifilm's regenerative medicine initiatives, our first step. We understand cell science-based technologies 2 layers. Firstly, cell science-based related basic technologies, such as devices, scaffold material technologies, and services, and also the iPS cells for drug discovery. And secondly, the cell therapy applications. Our RCP technologies will be applied for the JTX tissue engineering products and also three-dimensional tissues or organs with iPS-derived cells. And this slide shows the comparison of photofilm and cells. Left side is the electron microscopic picture of the cross-section of photofilm. Thickness of the photosensitive layers is about 20 micrometers. Within this 20 micrometers, there are about 20 layers, and each layer contains a nanocrystalline, nanodispersed crystalline silver halide, and also the nanodispersed functional particles together with collagens. Look at the right side. This is a cross-section of liver cell, same magnification of electron microscopic pictures. We can see the well-designed, fine structure of that. I think you can understand that Fuji's nano-sized manufacturing technologies also adaptable for the biotechnology areas. And collagen is one of the major raw material of photographic materials. So we have many specialists of collagen, and they had made infinite characteristics of collagen, uniformity and similarity to human type 1 collagen using gene technology and yeast cells. We call it the recombinant peptides, RCP, and already we succeeded in a large scale-up manufacturing 4 years ago. Of course, we are good at manufacturing many kinds of materials, such as the micropieces, sponges, hydrogels, microspheres, films also. When we combine the particular shape of RCP particles with cell, we call it cell-like. We saw the very interesting features. So we introduced a pancreatic islet with MSC cell-like to the diabetic model mice. And please look at the light graph of, in this slides. In this slide, the blue line is a normal mice. And the top pink line is a diabetic mice, and the red line, upper line, down line is the diabetic mouse with islet with MSC cell-like, including our RCP materials. So again, we are very much interested in the scaffold technologies, and we can provide our RCP as a research use and manufacturing use also. And this slide shows our group company, CDI, founded in 2004 by world-renowned researcher, Dr. James Thompson of the University of Wisconsin-Madison, the first in the world to generate ES cells. Fujifilm acquired CDI in May 2015 and spreading its research and business to worldwide. We are very much interested in drug discovery system using its iPS-related cells and its regenerative medicine products also. And this is our group company, JTIC's feature. JTIC is founded in 1999, already gets 2 products in the market, JACE and JAC. JACE autologous cultured epidermics approved in 2007, and JAC autologous cultured cartridge products approved in 2012. Now expanding its sales in Japan, also they started CDMO business using their know-how to develop regenerative medicine products in Japan. And in December 2014, JTIC became a considered subsidiary company of Fujifilm. And lastly, I'd like to share with you about firm activities, because Fujifilm has contributed to do the leadership of firm since its starting. Now, the chairman of firm is Mr. Toda, who is Fujifilm's CTO, and I am the head of steering committee. Firm has now 204 companies from various kinds of business areas, And here shows some of the well-known companies. And lastly, one of our activities in international collaboration. Recently, we have focused in the Asian Pacific collaboration and exchanged MOU with Australia, Korea, and China. And we'll be exchange MOU with India and Taiwan soon. That's the end of my presentation. Thank you very much.
Thank you, Mr. Yokokawa. Next speaker is Mitsuru Inamura from Replocell. He's talking about practical application of iPS cells from drug development and release medicine. So, Dr. Inamura, please.
Thank you for introduction. It is a pleasure to here to talk about my company activities. I came from Replocell, Japan. Replocell company founded by Dr. Nakase-sensei, and recently acquired 2 companies in US and 2 companies in Europe, StemGen, BioServe, Regenerative, and BioOpta, to cover iPS technologies. Our main, currently, our sales activity focused in research area and drug discovery area, and provide a reagent, media, and iPS cells. And now we are moving toward regenerative medicine area. This is a roadmap for regenerative medicine area, divided into 3 steps. First is the upgrading our research reagent area, reagent into the clinical media reagent. And step 2 is a somatic regenerative medicine. And the final 3rd step is a regenerative medicine by using iPS cells. It is essential for delivering clinical iPS cells for regenerative medicine area. I am now building the GMP grade RNA reprime kit, coating solution, and media, and cryotemso-free freezing media. Last year, we launched the cryotemso-free freezing media. This media can be stored iPS cells with high viability and high growth after swing. And we are now starting to manufacturing GMP grade, and register will be finished to the beginning of the early next year. And Replocell already a global company, so we are located in Japan and US and Europe, so we can consult with local regulatory authorities such as FDA, PMDA, EMA, so we can design manufacturing tailor-made media. And let me shift to the RNA reprime systems. My main topics in this presentation. RNA reprime systems are the latest technology to establish of iPS cells. There are many advantages in repriming systems: virus-free, DNA-free, and non-genetic integration, and provided a GMP competitive grade. First commercial available repriming kit is launched to the 2010 years. This kit enabled to generate iPS cells from fibroblast. And last year, we launched to the new RNA kit. This kit enabled to establish from blood cells. And this year, we can again launch to the new repriming kit, non-modified RNA repriming kit. This kit is able to establish the iPS cells from skin, blood, and urine, 3 kinds of the sources. It is easy to isolate the epithelial cells from sources and efficiently transfected in the RNA. The protocol is very simple. Plate the fibroblast or donor cells and transfect RNA with repeatedly. And daily media change and colony identification. So you can obtain the iPS cells within 2 weeks. And RNA is no integration, so you don't have to screen. And efficiency is very high. For example, from fibroblast, you can pick up to the 1,000 colonies per well, and the differential capabilities is no difference confirmed. And this commercially available kit is provided 3 components and provided as a GMP compatible grade and manufactured by RNA Therapeutics company exclusively for ReproCell. And we can provide the RNA and iPS cells on your request as custom order. And there is no evidence existing the exogenic reprogramming factor in iPS cells within 2 or 3 passages. And this is the most prominent advantage of reprogramming systems. RNA reprogramming reduces the number of copy number validation. Please see the please see the lower table. In iPS cells, by using retrovirus vectors, there are a lot of copy number variation changed. The reduced copy number or lost single genes or double genes. On the other hand, by using RNA reprogramming systems, reduced the risk or frequency of changing the copy number variation. So by using RNA reprogramming systems, you can get the intact, clean iPS cells. So already a lot of institutes use RNA reprogramming systems. Recently, New York Stem Cell Foundation is published the paper Automated High Throughput Deliberation of iPS cells by using RNA reprogramming systems. So last of my slide is show the comparison with RNA and centralized vectors. RNA reprogramming systems delivered a lot of iPS cells from several disease or donor cells. Okay. For example, the Parkinson or breast cancer stem cells RNA efficiently the iPS cells than Sendai virus vectors. Sendai virus vectors is the main, a lot of measure of the reprogramming system in Japan, but our reprogramming systems is superior. So in summary, ReproCell coverage iPS cell technologies produce cells reprogramming by RNA and differentiation. So it is happy to help or assist for genetic medicine area or drug screening. Thank you, attendance. Thank you.
Thank you, Dr. Inamura. Next speaker is Mr. Hiroshi Ozawa from Ajinomoto Company. He's talking about development novel xeno-free medium stem fit for human pluripotent stem cells. So, Mr. Ozawa, please.
Thank you very much, Professor Sada. Hello, everyone. My name is Hiroki Ozawa from Ajinomoto. So first of all, I'd like to thank the organizers for allowing me to speak at this fantastic conference. Today, I'd like to introduce our product, StemFit, a medium for pluripotent stem cell. Before beginning my talk about the cell culture medium, I'd like to give you a brief introduction of my company, Ajinomoto. Ajinomoto is the world's leading manufacturer of high-quality amino acid. So we were established in 1909 for manufacturing the monosodium glutamate as a umami seasoning for contribution to the nutrition of Japanese people. Since then, over 100 years, we have contributed to human health globally by creating unique values in foods, amino acid application, and pharmaceuticals. Here are our products. We have developed a variety of food products and biopharmaceutical chemicals and pharmaceuticals and so on. So we have developed a variety of businesses and products based on our technology platform, such as amino acid nutrition research, powder technology, which is mainly applied in the nutrient development, analytical technique, and biotechnologies. So taken together with our platform technology and business experiences, we have just succeeded in the development of the pluripotent stem cell medium, StemFit. Actually, we've been working in the cell culture media nearly 30 years. So we launched an ASF medium, Ajinomoto serum feeding medium, in 1987. To develop for the optimal media formulation, so it is needed to combine several technologies. We especially expertise in the amino acid, which is an essential ingredient for cell culture medium, and analytical technologies. Analytical technology is contribute to significantly to development of the media, because media usually consists of dozens of the components, and their concentrations vary very widely from picomolar to millimolar. So it is needed to analyze every component and comprehensively for media formulation. So now we focused on the emerging market, regenerative medicine. iPS or ES cell hold a great promise as a cell source of the regenerative medicine, because these cells can propagate unlimitedly and they can give rise every type of the cell in the body. So first, we focused on the stage of the establishment and the expansion of the iPS cell. For development of the media, we have collaborated with the Kyoto University. So with high expertise in iPS cell research of the Kyoto University, we have succeeded in the development of the prototype medium, StemFit AK01. So based on this medium, we have developed a clinical grade media, AK03M. So all protein of this medium are switched to the recombinant ones. And also, we have developed the research grade media, AK02M, with high cost performance components without changing the composition. Now, we just launched StemFit Basic 02 in USA last September. So here are the features of StemFit. StemFit is a xenophilic medium for feeder-free culture with various matrices. The cell can dissociate in a single cell and can be inoculated with the exact number. It is very important to establish the reproducible and stable protocol. And the cell can expand more than 100 times in the single passage with less frequency of the medium feeding. So let me introduce following three features in detail. The first feature of the StemFit is a superior and stable growth performance. So in this experiment, on feeder cultures, iPS cells were transitioned to feeder-free culture with laminin 501E8 or vitronectin. As you can see, red line culture in the StemFit display a very linear growth curve with a high expansion capacity while expressing the pluripotent markers. And we also tested other commercially available matrices like laminin 521 or Maltesian. So we found StemFit enable growth with all of the commercial matrices, even at low seeding densities. The second feature is cost effectiveness. So usually, everyday media changing is needed for the pluripotent stem cell culture. StemFit can maintain the cell without weekend feeding and with less media consumption. From both sides of the labor load and the media consumption, StemFit enable cost-effective culture. The third feature is a high colony forming efficiency. StemFit enable high colony forming efficiency, which are applying the downstream of the applications such as genome editing. So the gray bar in this experiment, so 9 cells per well are seeded in the 96th well, and after 1 week, we found many colonies can get within StemFit. So in this graph, the gray bar represents the number of the wells, and the upper two graph is a result of the stem fit, and the down of the competitor medium. So now stem fit is used by many customers, not only in Japan, but also over the world. So now we got 27 publications so far. That's all. Thank you for your attention. And if you are interested in our product, please visit our booth 516. Thank you.
Thank you, Mr. Ozawa. Next speaker is Dr. Seigo Izumo from Takeda Pharmaceutical Company. He talked about T-Cyra, industry-academia collaboration in attempt to overcome Death Valley. Dr. Izumo, please.
Thank you very much, Professor Asada, for kind invitation for this exciting session. As Professor Nakatsuji mentioned in his beginning talk, the theme of this session is industry-academia collaboration as it relates to stem cell the technology. Takeda Pharmaceutical Company traditionally has been the pharmaceutical, particularly in a small molecule company, but we have embarked on regenerative medicine area a little over a year ago. And the vehicle we have used is a program we call T-Cyra, which I describe in great detail. It has a number of untraditional features with an attempt to overcome the so-called Death Valley, which I will also elaborate later. So, brief introduction about Takeda Pharmaceutical Company, founded 235 years ago. Under new leadership of Christophe Weber, he's actually a Frenchman, first foreign CEO to be a CEO of Takeda. He has focused on the therapeutic area into four, GI, oncology, CNS, and a vaccine. And these are the main area. Of course, we have other businesses. And we have about 1,800 billion yen, or US $16.1 billion revenue, with a number of employees in 31,000 in more than 70 countries, or 90 countries rather. And this is our mission to strive toward a better health for people worldwide through leading innovation in medicine. So innovation is a key word for our company. So back in 2015, Takeda and IPS cell research application, CYRA, at the Kyoto University, agreed to start a 10-year-long research collaboration, T-CYRA joint program. The unique feature of T-CYRA is first a long-term, 10 years at least, committed by both CYRA and Takeda, aiming to develop cell therapies and drug discovery using iPS cells. The program is solely directed by Professor Shinya Yamanaka. This is a little bit unusual because typically industry-academia collaboration, we have joint steering committee. Equal number, represent, each party. What happens is that means that no one really has a power to make a decision, and often lead to protracted discussion. Usually, company reserves the right to, for the things that they cannot agree. Company may have a last word, but that also leaves sort of a hard sense, sort of hard feeling, so to speak, on academia side. So we decided, and also together with the fact that the Professor Yamanaka really doesn't have a time for committees, he said. So we said there's no committee. Dr. Yamanaka decides. So this is the unique feature. And open innovation housed in the center of a drug company's research headquarters. In fact, traditional academia industry collaboration, usually industry provide the money and the research take place in academia. But in this case, it is research take place in the Pharmaceutical Research Institute. And the Takeda provide the total research budget of approximately 20 billion yen over 10 years, plus in-kind support of at least 50 Takeda FTEs, researchers, drug discovery technologies such as screening and other R&D know-hows. And over 100 researchers are at one site already, and the program recruits scientists not only from SILA, but from other academia as well. So this is a framework of a T-SILA collaboration. Let's see what it's, oh, here, oh, it's working. So SILA provide iPS cell science and technology, drug target in some cases, prototype assays, and PIs and the postdoctoral researchers. Takeda, on the other hand, provide R&D know-hows, facility, equipment, researchers, additional researchers from company employees, basically, and drug discovery platforms and the budget. So this is a framework. So we work on the cell therapy, drug discovery, drug safety and efficacy, and the tool development. The only thing is really, you have to use iPS cell at some point during the research of each project. And the collaborative effort, if it's a cell therapy, together with a pharmaceutical science division, my unit, the regenerative medicine unit, will take it forward for cell therapy and gene therapy and clinical development. If it's a drug discovery program, Takeda's drug discovery unit will, where the large number of chemists also work, they will take the hit to lead to a lead optimization and appropriate preclinical studies to file the IND and the new drug clinical development through the therapeutic area units. So although this portion is 100 plus people, but this is supported by the Takeda Shonan Research Center with more than 1,000 people, with has a various expertise in the compound library screening, the pharmaceutical expertise and DMPK toxicology and so on. And it is entirely possible that not every clinical candidate come out from a T-SILA program Takeda is able to develop for future clinical application. That could be, maybe simply that too many candidates, which is actually not a bad thing. But maybe that, by then, when these things mature, that companies' therapeutic area strategy may not necessarily fit with what can come out through this. So in that case, in order not to sort of let the project languish on the shelf, we actually return the IPs to SILA so that the SILA is free to collaborate with other companies in order to capitalize on their investment. So this is actually where T-SILA is located. It is literally in the middle of the Takeda's Shonan Research Center, and the various laboratory equipment, office area, extensive cell culture system, and also they have access to screening and basically all the machines in Takeda that has within this building is accessible to T-SILA program, including vivarium that can actually not only do small animals, but large animals as well. So this is, total space is about 5,000 square meter, and total budget is 32 billion yen over 10 years, 200 billion yen for direct research and about $12 billion yen worth of support, in-kind support. By the way, this T-SILA, the logo up here, this actually, yes, some of you might have noticed that it takes 4 colors of SILA, represent the 4 Yamanaka factors. And center is a paper crane, which in Japanese means a prayer for the sick and unfortunate. So we put as a theme of T-Syra, reprogramming the future, with the hope that this research will lead to novel therapies for patients suffering from intractable disease. And these are the principal investigators in T-Syra program. I don't read each one of them, but it suffice to say they are professors, associate professors, assistant professors at Syra initially, but also we have associate professor from Yokohama City University. We have just adopted another person, not from Syra, not from Yokohama. So, and then interviewing another person from outside of Syra. So we are still building, but principal investigators are not really restricted to Kyoto University faculty. And these are the kind of project we are working on the beta cell, the development of the device for type 1 diabetes, and as well as drug screening. And in the CNS area, we are working on a neurodegenerative disease, in this case, at this stage, primarily on the drug screening using a patient-derived iPS cells. Cardiomyocyte project consists of a cell therapy program as well as a drug discovery, again, using a patient-derived iPS cells. Skeletal muscle disease, we are doing a small molecule screen for Duchenne myopathy, and as well as a gene editing for the Duchenne muscular dystrophy by means of exon skipping. And in terms of liver, we are working on using a mini organ developed by Dr. Takebe at the Yokohama University. We are using it for prediction of liver toxicity as well as the drug screening using the mini organ itself rather than a single cell type. And oncology, we currently focus on the iPS-derived the T cell with engineered T cell receptor, antigen-specific T cell receptor. So this is the retreat we had in August. Dr. Yamanaka here. So now there are the more than 100 members actively engaged, and we are all very excited about literary. Academic faculty, as well as the postdoc and the researchers. Kyoto University recognized T-Syra as a branch of Kyoto University because Dr. Yamanaka has a total say. I mean, he is directing the program. So we have an opportunity to actually train young people on the drug discovery or cell therapy in actually at the forefront of research. Let me actually touch upon the so-called death valley that you're all familiar. Japan, has a great academia and a number of new discoveries are made, but unfortunately, there are the sort of vehicle to bring academic seed to a full product. The infrastructure, sort of a community is missing. And some, the discovery made in the Japanese academia eventually become product, but it's actually in the hand of a foreign the ventures or foreign pharmaceutical company. So what can we do about it? And what are the issues on the death valley in Japan? So these are the reasons that are often said. The very few venture companies or angel investors in Japan for a variety of reasons. And typically, these venture companies, there's one research project, one company. So when the project dies, the company dies, and then this will sort of cause a loss of momentum as well as sort of a negative feeling, and sort of discourage people to, discourage the people to recruit the talented people. Okay, and also the shortage of fund. Typically fund is like 3 years, 4 years at most, and the difficult and the restriction of research facility. These companies often have to work in the area that are very suboptimal, so the application research. and also conflict on intellectual properties. When you have academic industry collaboration, which side did more work? 90% is actually done by pharmaceutical science, and what is the sort of equitable way to divide, IP, etc. So at T-Syra, we try to sort of address it in the following way. So the Takeda is playing like a role of a venture in this case, as well as the incubator, because we are providing this space and money and also FTEs. And we have a research portfolio, multiple projects. So one project die, that a program still get maintained. It will be replaced with a new one. So this actually sort of we believe that lead to the better decision, because when your entire company depends on the one product, you try not to kill the project when it actually should be killed. But we have portfolio, so we should just let the science drive. And we, as I mentioned, sufficient fund and a sufficient duration. So long duration, so the academic faculties are actually interested in tackling a problem that require a long time but highly innovative. And direct participation of researchers on the same site, this will create actually a very productive environment. And also at the T-Syra, basically every IP come from T-Syra, jointly owned by Kyoto University and Takeda. Even it's that particular IP is 1% Takeda contribution, 99% the Syra contribution, or vice versa. We said all 50-50. This is like a sort of like a marriage. After you get married, who earned the money actually doesn't matter. Everything after your marriage, all things acquired is 50-50. So this will actually avoid the sort of a daily squirm over who did more what. So the last slide, in summary, while there have been a number of success in the past, traditional industry-academia collaboration have largely faced the difficulties and to deliver clinical useful results. T-Syra program is a new form of industry-academia collaboration in research, a facility of a pharmaceutical company in order to deliver cell therapies and new drugs using iPS technologies. And the T-Syra program is an attempt to overcome this barrier toward the innovative therapies. I have to say disclaimer, because T-Syra just started a little over a year ago. So whether this hypothesis is correct or not, you will know 10 years from now. Thank you very much.
Thank you, Dr. Izumo. Last speaker is Dr. Margaret Ann Craig from Stem Cell and Device Laboratories. So, Dr. Ann Craig, please.
Good afternoon, everyone. So I'm delighted to be part of the Japanese delegation this afternoon, although you may realize that I am not actually Japanese. But I actually am based in Glasgow, and I travel to Japan every month, and fortunately to work with Stem Cell and Device Labs in Kyoto. So I'll give a little introduction to the company and our technologies, where they fit in the current market, and the use of these technologies for the drug discovery screening market. So Stem Cell and Devices is a company which is based in Kyoto. It's founded by Professor Nakatsuji, who is a real trailblazer in the field, and he has successfully formed a stem cell company and now has a second company. So not content with retirement, he's decided to keep working and to commercialize more of the products and the patents from Kyoto University. So the company was founded in May 2014, and it has several patents licensed from Kyoto University. What its first product is to develop cardiac stem cells for use for drug screening. So the stem cells that currently exist in the market are generally 2D models, and they have issues with maturity and also with the responses to compounds. And so now the future is to look at 3D models of cardiac stem cells for drug screening. So Professor Nakatsuji and his team have been working to develop 3D models. They differentiate cardiac cells using no cytokines, and also they have these proprietary cells, and then they add these onto nanofiber technology. And they've been developing and patenting these nanofibers so that we have a stable and reproducible 3D structure, which imitates what the natural heart would behave and look like. So these 3D structures then are developed in Kyoto, and they can be shipped internationally to any lab globally. So they have been developed, they have a high purity, the cardiac cells have a high purity, around 90–70% of cardiac cells, and they can then be added to the nanofiber structure. And these structures can have a stable frame. The structures are about 15 millimeters by 10 millimeters, and they can be handled easily with forceps, and so they are transported as live beating structures to any lab globally. So why are these tissues so important and so relevant just now? So for many pharma companies, the ability to look for cardiac toxicity of any potential compound they're developing is a serious business for them. And over the last few years, there have been many notable failures of cardiac drugs in the market. So the FDA does have regulatory testing methods to look at the potential toxicity of compounds on the heart, and their regulatory assay, the ICHS 7B guideline, was put together in 2005. But what they recognize is that this guideline is no longer fit for purpose, as that too many compounds are going through the pipeline and are failing in very late stage clinical trial, or else are going into market and then failing. And if we look at the compounds that have failed over the last few years, these are an example of some compounds from pharma companies, and the costs and the liability costs of these are averaged to be around about $150 billion. These are not the development costs, these are just purely the liability costs. And so cardiac toxicity is a serious issue for pharmaceutical companies. So normally what a pharma company will do is they will test a compound and engineered cells, such as a CHO hex cell, that has one ion channel present within that cell. And then we'll also look at high doses of this compound in volunteers in a thorough QT test and a phase 2-3 of clinical trial testing. So these are the normal methods that I've used, but what is happening is these compounds are going too far through clinical trial testing into market, and the toxicity is often detected when in market. Okay. So now what we need to do is have new methods to be able to detect this toxicity at an earlier stage, because this is time and money that should be spent elsewhere. With the advent of cardiac stem cells, we now have new tools that will allow us to assess cardiac toxicity. This audience will be aware of the normal ECG measurement of the heart. It's a flow of salts in the solution surrounding the heart. Normally then what we need to do for pharmaceutical companies is to test to make sure we do not cause any issues with cardiac normal salt solution diffusing through the membrane of the cell. So this is what we see in an ECG measurement. Anything that prolongs this waveform can result in arrhythmias, torsade de pointes, and it can then cause serious issues for individuals. And so now with stem cells, we have fully integrated cells that we can use in order to assess better the effects of compounds in the heart. So in 2013, the FDA formed a committee that decided that what they will do in future is to use cardiac stem cells for toxicity measurements to the heart. So they have formed a committee called SEPA, and what they're doing is looking at the whole integrated heart cell together. They're not focusing on what used to be the original potassium ion channel, but they're looking at multiple ion channels because these ion channels can work together and they can negate each other and compensate for each other in a fully functioning cardiac cell. So now the FDA has been validating stem cells that exist on the market, and they've gone through a 2-phase validation procedure, and their intention is December 2017 that they will implement cardiac stem cells as a new regulatory test. So this is a great example of stem cells being used in the marketplace to speed up drug discovery processes and to be more predictive and to produce safer medicines. So at Stem Cell and Devices in Kyoto, we produce our own cardiac stem cells through technology license from Kyoto University. And what we're doing now is building initially 3D models with these stem cells to be more predictive than the current 2D models which exist on the market. It also allows us to be able to take more measurements from the 2D models. So the FDA has chosen 2 technologies in order to be able to screen these compounds as regulatory tests. The first method and platform they will implement is the microelectrode array. The second are voltage-sensitive dye work. So what we have been doing as a company is taking our 3D models and validating them on these platforms using the compounds that the FDA recommends. So this is an example of the waveform, and showing that they're very reproducible results from these tissues. This is showing that we can pick up from the microelectrode array all of these action potential and electrophysiology waveforms, extremely reproducible across all of the platform. Also, that was microelectrode array. This is voltage-sensitive dye. These tissues are reproducible and stable, and this is then being shipped 3 days, still beating at 37 degrees on a plane from Kyoto to Glasgow. And the field potential duration from them is very reproducible from tissue to tissue. What we see here, this is an example of a compound that affects the heart. It will increase the waveform of the action potential, and we very easily pick up the effects and any arrhythmic effects from these from this drug. So this is an example of one of the compounds. And these are examples of other compounds that we have tested on the heart, looking at both potassium, sodium, calcium channels, sodium channel effects, and we can easily pick up these effects of the compounds within these microtissues. That is voltage-sensitive dye from the microelectrode array, again showing that we can pick up effects of these compounds that would have gone into market and would have not have been detected at in vivo, in vitro, or clinical trial stage. So what we have then are 3D products which are very easy to use, can be shipped live globally to any company or any academic lab globally. They arrive in perfect condition, they're beating and they're ready to use. It takes away the tissue culture time, the technician time required, and they're very predictive of adult responses, and they can be used for long-term acute chronic studies of compounds, so you can look at structural and functional effects of compounds. This is the initial market for this company, but the plan is to think about these for attaching to repair the heart for future. And this is some of the members of the company, and thank you, and please get in touch if you would like to hear anything else or get some more information from us. Thank you.
Thank you. Thank you for attending the symposium. Time is exceeded, so question and answer, please drop by our Kyoto University site. Thank you very much.
Ormond Street. Wait, I gotta look at my notes. The Greater Ormond Street Hospital, the University College of London. And he's gonna talk about the use of amniotic fluid stem cells in treatment of necrotizing enterocolitis. Thank you.
Thank you, Joanne. Thank you for the opportunity to discuss here something that is very close to my heart, in the sense I'm a pediatric surgeon at Great Ormond Street Hospital, and we face almost on a daily basis with patients that look like this one in the picture. So those are premature babies generally, and they get a terrible disease of the intestine primarily, and they die for a multisystemic organ failure that affect primarily the intestine, but then gets on and destroy their liver, their heart, their brain. Necrotizing enterocolitis doesn't still, unfortunately, have a cure. About 40% of these babies, when they got NEC, they die. As surgeons, we do a very old-fashioned therapy in the sense that we go in, do a laparotomy, and we take out the intestine that is diseased, or make them very sick. So they are left with a short bowel, and if they survive, they are condemned to have TPN and transplantation. So we are really lucky that there's a very reliable model of NEC that is made in pap rats. These are delivered just a bit prematurely, and then they go on to have the same stimuli that those premature babies in human have, in the sense that have formula feeding instead of breastfeeding. So we gavage feed 4 times a day these 4-gram pap rats, and credit should be given to the postdocs doing that. Hypoxia is another stimuli which is very important to create a model, and LPS mimic the bacterial translocation. So in these pap rats, we use stem cells derived from amniotic fluid. We described these cells first with Dr. Tony Atella in Boston at that time, about 8 years ago in the original paper, but essentially we described the possibility of deriving c-KIT positive lineage negative in the amniotic fluid. These cells may have a potential for therapy. Certainly they grow very well outside the body, and they can show different type of engraftment. With Marina Cavazzana-Calvo in Paris, we demonstrate that we can engraft these cells in all hematopoietic lineages in rag minus mice after irradiation, and we have shown that their potential could be just beyond what is recognized of mesenchymal cells, and in fact, we could reprogram these cells to iPS without any genetic manipulation. So by culturing the cells in valproic acid, we can generate truly pluripotent stem cells that are able, when implanting immunodeficient mice, to generate a teratoma. So in these pap rats, we inject cells in the peritoneal cavity. And as you can see here, the numbers of PAPs that we did to validate our results were quite high, about more than 100 PAPs for each arms, and we could see that we could lengthen the survival of these rats. So these rats are not made as a model for long-term survival, because you cannot introduce the same maneuver that you would introduce in these babies, such as intubating or giving TPN. It's just not feasible in the 4-gram rats, but we could lengthen survival in a significant way. And in fact, we proved that only the amniotic cells, but no, for example, bone marrow or a lineage of inert myoblast could significantly increase the survival. So when we look at what was happening on those rats, we demonstrate that there was a restore of the motility of the intestine, while the absorption there was a tendency, but that did not reach significant difference. We wanted to have, of course, a parameter to look at that, independent parameter. And so the radiologists look at the MRI of these pap rats, and they could clearly blindly assign the normal pap rats, in which you define a different structure, the one affected by NEC, in which you see fluid inside the lumen of the intestine and outside in the peritoneal cavity, as you commonly see in these babies, also in the clinical model. But most importantly was in the one that were treated with the amniotic fluid stem cells, these features were not recognized. When we look at surgically and sacrifice those animals, clearly you can see in the breastfeed animals there was a normal looking intestine, which was necrotic in the one that had NEC and was very similar to the normal one in the rats that received the amniotic fluid stem cells. There was some area of inflammation and some probably strictures. The pathology look blindly at those tissues, and you can see disloughing of the mucosa and necrosis of the villi in the animal that got NEC, and the animal that received the cells show a pretty normal restoration of villi anatomy. But when we look at what the cells did, we recognize that only a very few number of cells engraft into the intestine. As you can see here, we could target the cells because they were GFP positive. We could see that they were moving from the mesentery to the wall of the intestine. We now know this probably through a lymphatic way. And these cells are however localized in specific places in the intestine, which are very important for stem cell regulation. So while they move into what it seems the epithelium, but actually is below the epithelium in the axis and in the crypt of the villi, which is very important for the cells and the epithelial restoration. So when we look at how the cells act, because we could not see much direct regeneration, as you observe very few cells engraft, we look, of course, at the microarray first, and we saw, not surprising, that they were involved in proliferation and downregulation of apoptosis and inflammation. This was described before by Stappenberg, who described a very neat model in colon cancer, which is regulated by COX-2 positive cells. So COX-2 positive cells are present in the axis of the villi and move down to the crypt when epithelium needs to grow. Of course, in colonic cancer, there's a dysregulation of this mechanism, and so you have growth and anti-apoptosis lead by the COX-2 positive cells. When we look at the COX-2 positive cells, we recognize quite clearly that in the NEC animals, the COX-2 positive cells were almost disappearing while they were restoring when we were injecting amniotic fluid stem cells. Interesting enough, when we compare breastfeed animals and amniotic fluid stem cells animals with NEC, we observed that there were more cells under the crypt. So when we look at numbers, we could see that there was no significant difference between normal breastfeed animals and animals with NEC that were treated with amniotic fluid stem cells, which is reassuring because of the risk of having high number of COX-2 positive cells can lead to tumor. However, when we look at the position of the cells, we recognize that in the animal that had NEC and had the amniotic fluid stem cells, the cells were mainly present in the crypt, which makes sense because it's where the cells help the epithelium to proliferate. And this is so relevant that when we look at the correlation between the number of COX-2 positive cells and the damage of the epithelium, there was an inverse correlation between the number of the cells in total and the epithelium. And this correlation was even more significant when we look only at the cells present under the crypt. So, of course, you have to randomize your patient, in this case the pup rats, to observe if this is really true. So what we have done was to use breastfeed animals, NEC, and NEC with the amniotic fluid stem cells. And then we randomized to 4 subgroups. We randomized to have an inhibitor of COX-2 positive cells, an inhibitor of non-so selective, so COX-1 and COX-2, a COX-1 inhibitor, and just the vehicle that was the DMSO that was given with gavage. So in the survival, and again in the different experiments, you see the curve is exactly the same of what I showed you before. What happened is that if we use COX-1 inhibitor, we did not observe any difference. So the animals were still having the effect of the amniotic fluid stem cells. However, and in support of our hypothesis, when we inhibit the COX-2 pathway, then the effect of the amniotic fluid stem cells was completely abolished. And this doesn't come as a surprise. I'm sure that when we use COX-1, 2 non-selective inhibitor, there was no differences. So our hypothesis is that the cells that we inject through a paracrine mechanism that we are studying at the moment activate the COX-2 positive cells that migrate down into the crypt and activate the epithelium to proliferate and to restore the intestine. We know, for example, that some of those factors are involved. So we observe that when in vitro we stimulate the cells with LPS to mimic what happened in vivo, and then we look at how the cells react to that. We observe a much higher expression of key factors like FGF2 or PDGF-beta were compared, for example, to bone marrow stem cells. So we think that beside thinking that amniotic fluid stem cells can be a source for therapy, we also think that in the necrotizing enterocolitis model, these cells may be therapeutic. Thank you for your attention.
Great, thank you, Paolo. We're going to go through all the talks and then have a panel at the end, so we'll save questions for the end. And I'm going to go next. I'm Joanne Kurtzberg from Duke, and I'm going to talk about using cord blood in regenerative medicine applications in children. If I can have this slide set. Great, thank you. So I need to tell you a little bit about the beginnings of the cord blood story, which was that cord blood was used in 1988 to rescue hematopoiesis in a 5-year-old boy with Fanconi anemia after discovery that his baby sister's cord blood was a match and that his baby sister was not affected with Fanconi anemia. And the transplant was done in France by Alain Gluckman, shown in the white coat in the picture. And fortunately, the transplant was successful. It restored hematopoiesis in the same rate that we would have expected with bone marrow. And the patient is alive and well 28 years later. He's shown in a picture last year with me. And most importantly, his bone marrow and immune system are still durably engrafted with his baby sister's stem cells. We took that work into the clinic, and I don't have time to go into the history, but unrelated cord blood transplant was discovered and validated, and today there are public banks, private banks, and a large source of unrelated cells for transplantation held in various registries. And 40,000 or so unrelated cord blood transplants have been used around the world to treat patients with the same diagnoses that you would treat bone marrow transplant for, like malignancies, hemoglobinopathies, immune deficiency, marrow failure, and certain inherited metabolic diseases. And on our work at Duke, we used cord blood as a donor for hematopoietic stem cell transplantation in patients with inherited metabolic diseases in the categories of leukodystrophies, which are listed on the left hand of the slide. And we observed in that work that the cord blood neurologic outcomes were better than bone marrow transplantation, and over the years have switched to prefer this source for all children, regardless of whether they have a related donor. We also have shown that over time, this treatment affords an 80% event-free survival in these otherwise lethal diseases. But it requires myeloablative chemotherapy, a long stay in the hospital, a risk of graft versus host disease, so it's not benign. Just some examples of treatment effects we've observed. This is a child with Hurler syndrome. Untreated, these children die at about 5 years of age. They die of cardiomyopathy, but they also have severe mental retardation. And the curves on the right show the neurologic cognitive development of the first consecutive 20 Hurler patients we transplanted with unrelated donor cord blood, showing in the black dotted line compared to normal children in the red line that their cognitive function is falling off in the first few years of life, but with treatment, they start to correct and can come back up in the normal range. And I'm old enough now to have followed these patients for a long time. So the two girls in the pictures here are both Hurler patients transplanted when they were about a year of age. They wouldn't be surviving without transplant, but they're both going to their proms. That's their prom picture. But they both have normal intelligence. They're both going to college now. So we know that this cognitive correction is maintained and not just a temporary effect of the treatment. Now, we know in cord blood that there are multiple types of cells, and we know that the hematopoietic stem cells, which is a very small population of the total cord blood cells, are the ones that are responsible for restoring hematopoiesis after myeloablative chemotherapy. But we also in our work have found, and I'm going to show you data about this, that there are other interesting populations of cells in the cord blood, and one that we are focused on is the monocyte population, which contains CD14 cells, which represent roughly 5 to 7% of the cord blood cells in total. And those CD14 cells, when they're isolated as fresh cells, have certain properties and are able to rescue tissue in hypoxic injury models. And we also can manufacture a cell from this CD14 cord blood cells called DUOC, which is effective in inducing remyelination. So with that background, we went into the area of using cord blood without chemotherapy as I think Jan Nolte said yesterday, a living medicine to treat children with various types of brain injury. We started with autologous, or the child's own cord blood, and that required that the family had chosen to bank their baby's cord blood in a private bank when their baby was born. We tested safety and have published results in Transfusion in 2010 and found that an intravenous infusion of autologous cord blood in older children was safe. And I'll show you scattered results of studies that we're doing in babies with birth asphyxia or hypoxic ischemic encephalopathy and in cerebral palsy and autism. In addition to that work, we're also working with allogeneic cord blood cells. We're manufacturing a cell we call Duke O cell version 1, or DUOC, and using it to treat children with leukodystrophies, supplementing a standard myeloablative transplant. And then we're looking at unmatched, unrelated cord blood in adults with stroke and sibling cord blood in children with cerebral palsy, trying to address the issue of donor availability to, in the end of the day, have a therapy if this is effective for every patient in need. So first I'll talk about the cell we call DUOC that we manufacture from cord blood. And on the left, you can see a picture of a section of the brain of a child who died from Krabbe disease, but had received a cord blood transplant with myeloablative chemotherapy 10 months earlier. And in this child, who was a girl, we were able to show with XY FISH that there was engraftment of donor male cells in her brain, and those are the green dot cells with Y chromosomes. And when we looked at what those cells were, they were microglial and oligodendrocytes, and we embarked upon a project to manufacture those cells from both fresh and cryopreserved cord blood, and the cell product you can see swimming around on the slide there is grown in 3 weeks of cell culture with growth factor supplementation. It's an adherent cell, and we can harvest it, and we've created a protocol to treat children intrathecally to supplement a standard transplant. When we studied the function of this cell in various models, we found that it was able to induce remyelination. So this is the Cooperzone model, where you treat mice with Cooperzone, and they demyelinate their corpus callosum. So the top panel shows a controlled mouse that got Cooperzone. And you can see on the left panel, there's very little myelin, which would be blue. In the middle, you can see astrocytosis in the pink, or scarring. And on the right, you can see that by electron microscopy, the rings of myelin are destroyed. Below, we have the same mice treated with Cooperzone, but then rescued with an intracranial injection of the DOOC cells and assessed a week later. And you can see on the left, remyelination by Luxel Fast Blue. In the middle, there's no astrocytosis, and on the right, that the rings of myelin are back to the normal morphology by EM. And that and many other studies we've done have allowed us to see that this cell has the potential to act through a variety of mechanisms, including enzyme replacement, cytokine secretion, which modulates inflammation, drives oligodendrocyte proliferation, and promotes myelination. And we were able then to go into a clinical trial for children with leukodystrophies who also have evidence of corticospinal damage at the time of transplant. And essentially, we take the big portion of the cord blood unit and use that for the systemic transplant. And then a week out, we start the culturing from the small portion of the cord blood unit of the DOOC cells. And 4 weeks post-transplant, we harvest them, and if the child's engrafted systemically without GVH, we give an intrathecal dose of the DOOC cells to augment transplant. So the baby in the picture is a baby with Krabbe disease whose sister died at 10 months of age, but was moribund at 5 months of age, and he is shown in the picture on the bottom left at 5 months of age, doing well, and here he is a year later, year post-transplant, doing well. So we know the transplant has a benefit here, but we also are hoping that we're arresting disease progression earlier with the addition of the DOOC cells. We've treated 9 patients. We have a few more in the pipeline. We have not had safety issues, but we know we can't really assess efficacy in this setting, so we're planning to do manufacturing from the whole cord blood unit and study adult demyelinating diseases without myeloablative chemotherapy. That's on our docket for the next couple of years. Going back to the fresh CD14s, we've looked at them in various models of hypoxic injury. So the histologic slides on the top show you a model where we can isolate brain tissue ex vivo and propagate it, and then expose it to oxygen glucose deprivation. And if we do that and look at the cells 72 hours later in the middle, you can see that in pink there's a huge astrocytosis and that the green neurons are gone. But if we do the same experiment and add CD14 cells post-shock, you can see on the left that the architecture is preserved, the neurons don't die, and the astrocytosis does not occur. And with that and many other studies, we've gone into studies in the clinic under IND in babies with hypoxic ischemic encephalopathy, giving them back infusions of their own fresh cord blood over the first 2 days of life, and also in adults with ischemic stroke using non-matched allogeneic cord blood. And that one box there is from a paper we published in the Journal of Pediatrics a couple of years ago, showing with a non-randomized concomitant control group that if babies with HIE got cooled and also received cells, they had better one-year outcomes, meaning better survival with normal function than babies who got cooled alone. And based on all of this data, we've been able to go into the clinic now and randomize phase 2 studies in HIE, which is a multi-center study, and also adult stroke. So maybe next year I can tell you more about that. The other thing we focused on is looking at the effect of autologous cord blood in treating children with cerebral palsy, particularly spastic cerebral palsy due to stroke or HIE or PVL or prematurity at birth. And over the past several years, we treated 63 patients on a randomized, placebo-controlled, blinded trial of autologous cord blood, where children received a single IV infusion in the outpatient clinic after pre-medication and were followed with multiple testing. The placebo that we used was tissue culture media with DMSO, so everybody got pink stuff and everybody smelled. And I'll show you the scheme in a minute. And our primary endpoint was change from expected GMFM score, which is a motor function score, at 1 and 2 years. So this was our scheme. Children were qualified, their cord blood was qualified, they were enrolled and randomized, and they either received cord blood or placebo, had a bunch of tests done, came back a year later, which was our primary evaluation, and crossed over to get placebo or cord blood, whichever they didn't get the first time, all blinded, and then they were evaluated at year 2. And the primary endpoint was based on this gross motor function measure, where 66 different points are evaluated by an observer of the child at each time point. And children can be graded by their severity and then also by age. So in this score, different children will have different responses depending on when they come onto the study by age and level. And to assess a change, we had to look at where the child was, where they were predicted to be, and then where they really were and how different that was from where they were predicted to be. So you can't just use an absolute number, because a young kid with a mild form of CP will be expected by natural history to have a lot of change. And that wouldn't be a response to the therapy. So we did all those analyses, and long story short, we were able to show that children, and these are 1-year assessments, that the children who received either a higher dose of cord blood cells, which was 25 million cells per kilo or greater, in green, had a statistically significant improvement in motor function at a year compared to either placebo in blue or low-dose cells in red. And we're confirming that with 2-year follow-up now, but the data is looking good. We also did MRI surrogate or biomarker studies, and the top panel shows you the differential increase in connectivity in the low-dose group on the left or the high-dose group on the right. And again, there was a statistically significant increase in connectivity in the high-dose group. And you can see it in the picture. So the top picture is baseline in 1 year of a child who had a high change score and a high clinical improvement and a high dose. And if you look at the red, you can see that at 1 year it's thicker. And on the bottom, you can see that an example of a child who did not improve and did not get cells and had actually more tissue in the red dye at 1 year, at baseline, sorry, than 1 year. So this is a good correlate, we think, with clinical outcomes. Our conclusion from all of that was that cord blood was feasible to give as an autologous infusion in kids with CP, and that kids getting an appropriate dose had improvement. And I didn't show you the data, but this was not impacted by age or severity of CP within the group, which was 1 to 6 years of age when they came on study. But we know that since a lot of children don't have their own cord blood but could have cerebral palsy, we felt we had to look and see if an allogeneic product would have similar results. And so our current work is focusing on that. And we've done a safety study in 15 children getting sibling cord blood. And we're planning to open this winter or spring a phase 2 randomized trial of allogeneic cord blood in children with CP who don't have their own cells. Finally, I just want to show you some results of a trial we're doing in children with autism. Again, we started with autologous cells, and in this trial, they had to have a qualified cord blood, and our hypothesis was that the cord blood cells would secrete factors that would modulate inflammation and suppress microglial activation in these kids. We performed an open-label study in 25 children ages 2 to 6, and we used the study to evaluate both safety and also define endpoints for a phase 2 study. We developed an in vitro assay to look at microglial activation. So normal microglia are on the left. When they're exposed to LPC, which will induce inflammation, they get microglial activation on the upper right. And when we put cord blood cells in on the bottom, you can see that that microglial activation is suppressed. And we plan to use that as a potency assay going forward. So in the phase 1 study, we did not notice any serious adverse events and felt it was tolerable. And we defined endpoints that we think are going to be useful in a phase 2 study. And we did describe clinical outcomes at 6 months and a year. And we did see improvement in more than half of the children on the study in a number of the subscales of different tests that are used to assess behavior in children with autistic features. And these are now the endpoints we chose for the next study. This is the Vineland, and I just want to show you that when we looked at the Vineland overall in the patients on the lower right, we did see at 6 months statistically significant increase in function in the overall population or improvement in behavior in the overall population, which was sustained but did not further improve at 12 months. But when we looked on the bottom right at which children were responding, the children with the nonverbal IQ of 70 or higher in red had a better response than children with a lower nonverbal IQ. And again, we're using this modeling to go into a phase 2 randomized trial, which is now open, will enroll close to 200 children, has enrolled about 40 so far. But this is what we call a best donors trial. So children are evaluated, their donor is evaluated, if they're eligible, they're randomized, and it's a 2-to-1 randomization between cells and placebo. And if children have autologous cells that qualify, they get them. If not, they get unrelated donor cells. We're not using sibling cells in this trial in case there's some genetic component to autism. And the children are screened to not qualify any children that have known mutations associated with autism, ASD syndrome diseases. So in summary, cord blood therapies show promising results for treatment of diseases with no currently available therapy. Results using autologous cells in babies and children with HIE, CP, and autism suggest efficacy. Safety of allogeneic cells has been demonstrated, and we're moving forward with efficacy studies of allogeneic and best donor cells. Don't have time to talk about it today, but regulatory pathways for both of these types of therapies need to be further defined to really bring this to the clinic. And I'll just end by thanking my whole team. It takes hundreds of people in all sorts of disciplines to really get these studies done, and it really takes a village. And we have support from multiple foundations, which has really enabled the clinical trials to be done, specifically the Julian Robertson Foundation, the Marcus Foundation, the Cure CP Foundation. And the 3 kids in the picture are kids we took to the FDA to help get the indication for allogeneic cord blood, unrelated cord blood, to be expanded to allow for treatment of kids with metabolic diseases. The little girl has, on the bottom of the blue shirt, has Krabbe disease, transplanted at 19 days of age with unrelated cord blood. The girl in the black sweater is one of the kids at the prom with Hurler syndrome. And the little boy in the blue check shirt is a kid with adrenoleukodystrophy. And they all, I think, were instrumental in convincing the FDA to use the right language in the indication to enable the therapy. So thank you very much. So it's my pleasure now to introduce Julie Alexson, who's director of the Wake Forest Regenerative Medicine Center at the Wake Forest Institute of Regenerative Medicine. I may not have said that exactly right. And Julie is going to talk about amniotic fluid cells, banking, and their manufacturing for use in the clinic. Thank you.
Good afternoon. Thank you. I'd like to thank the organizers and Dr. Kurtzberg and the Cord Blood Association who sponsored this session today. So I'm really excited. There's a lot of efforts moving forward in perinatal tissue. So I wanted to be able to touch on that and talk a little bit about some of the banking that we're doing at Wake Forest. So we have about 450 faculty and staff that make up the Institute of Regenerative Medicine. That's led by Dr. Tony Atala. And I really have to show the mission before I start because that's what drives everybody. It's this incredible energy that you feel when you get into the Institute. So we've had a lot of firsts in the field, but one of the things that we've worked on is stem cells from placenta and amniotic fluid, which Dr. Jacoby spoke about the amniotic fluid. So this is about 15 years of an effort so far, working with the cells in preclinical studies and looking at process development and moving them into a clinical master cell bank. So we know these cells can be collected at any time during development and then postnatal. So there's a lot of opportunities to collect these cells at 10 to 12 weeks gestation in a source of amniotic fluid or CVS, and then at term in placenta or amniotic fluid or the amniotic membrane. So these are just a few of the different sources that are used and that are actually being looked at in preclinical studies as well as being moved to clinic. So this is the paper that was led by Dr. Jacoby, a pivotal paper in 2007 that described the amniotic fluid and the placental stem cells and their plasticity and all the work that had been done, looking at the fact that these cells are non-tumorigenic. So they're close to an embryonic stem cell, but they're in between, I guess, what I would say bone marrow and embryonic stem cell, but closer to the embryonic stem cell. So this is the profile, which is basically the same as a mesenchymal stem cell, except you have a strong SSEA4 and OCT4 that's presented. And when we look at these cells, if we look at the cells from the 10 to 12 weeks gestation, we see that we can take them out over 250 population doublings, and we see that they maintain their telomere length from 20 to 250 population doublings. You see that in line 3 and 4 of the telomere length. It retains a normal DNA content as well as the karyotype. So these cells are pretty hardy, they're very plastic, they can differentiate into the 3 different germ cell layers, and they actually have immune modulation capacity. So as we see, these cells can inhibit lymphocyte activation and producing factors that can regulate the immune response. And as we compare those to bone marrow mesenchymal cells, we see a few additional factors that they produce, which may produce a little bit of a different effect than the mesenchymal stem cells. This is just an example of some of the tissue engineering that we looked at with bone differentiation. So taking the amniotic fluid stem cells, differentiating them to osteogenic cells, and then seeding them into the scaffold and implanting those into a mouse model. On the left, you see the scaffold alone that we took out by a Vancosa stain, and then next to that is the amniotic fluid where you're seeing the dark staining, so the mineralization from the calcium. Next to that is a micro CT scan of 18 weeks after implant, and you see on the left the scaffold, or the right, I'm sorry, was just the scaffold without the cells, and then on the left is the construct that was implanted, and you can see that out 18 weeks. So the cells have been used functionally and looked at functionally in several different animal models, and this one is just demonstrating the capacity to produce neonatal tissue, and they've been able to differentiate into podocytes, so they are compatible with the kidney tissue. And this is a few of the recent publications, but I'll tell you there's probably 100+ publications looking at the cell characterization as well as the functionality of the cell. And I guess I'd just call your attention to 2 of the studies. One is in osteogenic imperfecta, where the cells were able to differentiate and amend the brittle bone disease as we looked at those in an animal model, as well as Allaport syndrome. So again, it was able to differentiate into podocytes and demonstrate efficacy, decrease the blood urea in the animal, and inflammation as well. So I went back to look at the clinical trials, so how many cells have gone into clinical trials, and did a search on looking at placental cell therapy. I found about 82, and I'm sure that, you know, maybe they weren't all placental, but there were a lot of them. I noted some of the applications that we see a lot for diabetic neuropathies and critical limb ischemia. Amniotic cell therapy, about half that you see, which includes amniotic membrane, a whole membrane, a decellularized membrane, and the amniotic fluid stem cells. and umbilical cord tissue, which came along a little bit later, maybe about 5 years ago, where people really started looking at privately banking that and then started looking at the efficacy of those cells. So at Wake Forest, we bank placental cells. We set up a collection kit that goes over to the hospital. They collect the placenta either at a C-section or vaginal birth. That comes back to our facility, and then we decontaminate that organ. We biopsy the section of the tissue that's appropriate. We digest that tissue, and then we expand those cells. The selection of the tissue is very important because you have not only the fetal but the maternal cells that are present in the tissue. So it is a challenge. You need to be able to get to the right space in the tissue to be able to collect those cells. Otherwise, you'd have maternal cell contamination. If you get a certain amount of contamination, maybe even close to 30%, those can outrun your fetal cells and you could end up with maternal cells. So it is really important to be able to characterize and determine what your cell population is at the end. So just to go over, these are the characteristics. Mesenchymal stem cell with the SSCA4 and OCT4 expression. They double about every 36 hours. They can maintain their telomere length. These clonal cell lines that were produced go out over 250 population doublings. We see no evidence of tumorigenicity and really the capability of being able to differentiate into cells from the 3 germ cell layers. So we produce these master cell banks in our GMP facility. It's about 4,000 square feet. We work on tissue engineering organs, cell therapy, biomaterials, and small molecules in this facility. And we're really set up for innovation and really organized for translation. We have these core facilities that specialize in the area. They do training. They might do the testing for us, and they're staffed. So that really helps to speed along the translation as the product moves into preclinical development through process development and manufacturing. We're right now setting up a whole GLP facility that simulates a clean room so that we can make our preclinical test model articles in there as well as our process development. So we're not mixing anything in the manufacturing facility. It's all separated. This is our So I won't go through all this, but these are the steps, the process map as we're producing the master cell bank and all the testing that has to go on, which is really according to FDA criteria. And just want to say that there's a lot of opportunities as you're taking these cells and products to clinic. So right now with this product, as we're making master cell banks, until we really find a clinical application, those cells won't be required to be registered or an IND. Once we determine the clinical application, then we'll put in an IND. So right now the FDA doesn't really want to look at the cells as they're going into a master cell bank. Certainly you could tap on them for questions, but as you're moving to a cell therapy, you have a lot of opportunity to speak to them. So I guess I would really encourage you to go in with a strong plan and then get their feedback, because you are the experts at your technology, but you definitely want their feedback. You don't want to have to go back and repeat testing after you've gone through all the effort and costs. So I guess in summary, I just want to say that birthing tissues are definitely maturing. We're seeing a lot more collected, stored as private banks, and we think there are a lot of opportunities to be able to use these cells as an off-the-shelf product. They have potential as they're plastic. They can be controlled, so better than an embryonic stem cell, and the fact that there's no teratomas. There's people using the cells, the tissues, the exosomes. There's a lot of different products from these cells and tissues. And again, I would recommend that you consult your regulatory authority to be able to speed along to your translation to commercialization. Thank you.
And our last speaker is Todd McAllister, and he is director of the Amnion Foundation, which is a public stem cell bank. And he's going to tell us a little bit about it.
Thanks, Joanne. We wanted to finish the session with just a few minutes to remind everybody of the role that public stem cell banks play in this development. And I think as we've seen All of these technologies are starting to march towards the clinic, but we're still in relatively small numbers. So the question, of course, is how do we translate all of this from small, limited clinical studies to widespread clinical use? And just as a very brief overview for the session in general and some of the things that we've seen on the other days of this meeting, I think it's quite clear that there's not only expanding indications for cord blood derived cells, but also an expanded source of cells. So we've talked about mesenchymal stem cells from umbilical cord tissue. Paolo and Julie also talked about the amniotic derived stem cells. And again, while it's been very promising work, these are still relatively limited and generally autologous studies. And the challenge, of course, and I think Joanne would probably be the first to tell you, it takes years to enroll these patients in relatively small numbers if we're limited exclusively to the autologous model. So all of our goals, I think, is to be able to deliver this to widespread clinical use, and in general that means an allogeneic model. So the public banking model, I think, is very well established for cord blood, but not so well established at this point for these newer tissues and these newer sources that are coming along. So whether that's umbilical tissue or, again, amniotic-derived cells, that model is not yet into clinical practice. But I think the timing is appropriate. Again, Paolo and Julie both spoke to the fact that the amniotic-derived stem cells have now got more than a decade of developmental work in the clinic, and we're marching towards at least limited clinical use. So in parallel with those academic efforts, we've started to build a public stem cell bank located also in Winston-Salem, in fact, just across the street from Wake Forest. And our objective, as I'll show you in the next slide, is to be able to build a large public bank of these immunologically matched cells available to treat patients for these broader therapeutic applications. I won't go into detail on the science of this. I think you've heard that now a couple of times. But again, I think the key point here is we've got a very strong academic foundation for these cells as well as other cell types, and we're interested in being able to exploit that on a wider spread basis. So what is Amnion intending to do? Well, we're a nonprofit stem cell bank, as I said, located in Winston-Salem, and we really have three primary objectives. The first and most obvious is simply to put together the stem cell bank and build a bank of tissues to serve a large population. The second, and maybe a little bit less obvious, but we're also trying to support academic research using cord blood or placenta-derived stem cells. So we are philanthropically supported, but part of our mission is to partner with the best and the brightest from the academic community and support those studies. And finally, and this is something that's not often talked about, but again, I'm sure Joanne can talk to this point, most of the patients really have no understanding or no clue of the power of these cells, the risks, the benefits, et cetera. And while there are now a variety of different patient advocacy groups out there, one of our principal missions is to be able to provide educational tools and outreach tools for these patients to really truly understand what the risks and benefits are for stem cell therapies. And so I'll finish my little blurb here on Amnion by simply saying we are actively now pursuing partnerships and collaborative opportunities. That could be partnering to accelerate our tissue collection. It could also be grants and partnering opportunities that we're sponsoring and supporting. And of course, as I just mentioned in the last slide, trying to partner around patient education and patient advocacy. So with that, I'll finish my little 3 or 4 minute discussion on the impact and the importance of public stem cell banking, and encourage you to look at our website to learn a little bit more about what we'll be building over the coming years. Thank you.
So since we got started late, I want to give you, the audience, the opportunity to ask any of the speakers a question before we chat ourselves. Can we have the microphones on, please? There you go.
Actually, I don't want to destroy the party here, but I have major fundamental questions here. I heard the word stem cells about 1,000 times in this conversation here, and I wonder, are these really stem cells? they were defined as something between embryonic to adult stem cells, while it's questionable whether adult stem cells are stem cells. And I really tried to get point by point. As to the placental cells, the stromal placental cells have been shown by various researchers are not stem cells, are very potent cells. They have indirect effect on regeneration. They induce stem cells proliferation, but they by themselves have no stemness. So the stemness is questionable all along this discussion that we had here. And I'll go point by point. When we look, for instance, at the gut, and we see few cells that are integrating within one crypt, I look at it and physiologically understand they don't act as stem cells there, because every crypt is formed by single stem cells. So if there were stem cells, I would expect to see a whole crypt which is formed by these cells and not integrated in. I don't say that they don't help regenerate the tissue, but they secrete, there are secretors that help the body regenerate itself. And it has been discussed in various forums here, especially by Kaplan, who actually formed the terminology mesenchymal stem cells, and now admits that he actually was wrong. These are pharmaceutical factories rather than being stem cells that are involved in building tissues as building blocks, as one would expect. So it seems very sexy and nice, but actually it hasn't been proved. Thank you.
So we're going to have a two-pronged answer here. I will challenge you and say that in cord blood there are blood stem cells that engraft and repopulate and reconstitute hematopoiesis for decades, and we have evidence of that. But there are other cells that are effector cells, you're right, and are not stem cells. Why don't you guys address the amniotic cells?
Yeah, I mean, on the amniotic cells as well, we demonstrated a similar way in the sense that, for example, for the matter of time, I just show, because they are published data, we've shown that in the RAG minus mice, after irradiation, we can repopulate at first, secondary, and tertiary transplantation. So they behave as stem cells. In other example, we have seen, like in the intestine, as you say, that they have a paracrine action. I think we have to agree on the nomenclature in the sense that we consider stem cells that are like embryonic stem cells. Clearly these are not stem cells, but however stem cells have different potential, and I would define them as broadly multipotent, as we demonstrated in other experiments.
Yeah, I would echo what Paulo said, and I am cautious to say multipotent, so I think that there's different actions depending on therapeutic application.
Okay, we'll take one more question because we have to stay on time. Yeah.
Hi, I'm Victoria Nicholas from Prolacta Bioscience and from UCLA. This question is for Dr. DeCoppi. So as you know, breast milk feeding is the one intervention in preterm infants that actually reduces and nearly eliminates necrotizing enterocolitis. So I wonder if in your model system where the amniotic fluid stem cells actually repopulate the COX-2 positive cells, so is the effect in the breastfed model, is that due to a stem cell from breast milk, which is known to exist, or is that a factor in breast milk that supports that population of cells, the COX-2 positive cells in particular?
Yeah. This is a very, very interesting question. The breastfeed, unfortunately, reduces the incidence of NEC, but doesn't eliminate, in the sense that you have breastfeed term babies that get NEC, unfortunately. That type of experiment, unfortunately, has not been done yet, and I think it should be done. Clearly, there are some progenitors, let's use this term, in the breastfeed that may regulate the intestine and actually may be very relevant to the NEC. Other factors, though, may contribute as well in the breastfeed. You have antibodies in the breastfeed, you have immunological cells that can help as well. So the breastfeed is certainly a very interesting source, is undervaluated at the moment, and is very important, certainly in a newborn. Yeah.
Okay. Well, I want to thank our speakers and thank everyone for attending. Sorry we got started a little late, but we're going to stay on time now. The speakers are here, and if you have other questions for them, you can come up and we can talk individually. Thank you very much.
and retinopathies to the neural retina, we're talking about some of the leading causes of vision loss in the world, so macular degeneration, glaucoma, and diabetic retinopathy. Unfortunately, vision loss and blindness account for a huge economic burden, which was estimated to be $145 billion in 2014, and it's only expected to rise over the next 20 to 30 years. Vision loss and blindness remains to be one of the top 2 health concerns among the general population, with cancer and death actually coming in second and third. So people are very afraid of going blind because of the quality of life that entails. Today we're going to be talking about retinal neurons, like I said. The retina is a 7-layered structure, or at least the neural retina is a 7-layered structure in the back of our eyes. It's responsible for receiving the light, transducing that into an electrical signal, and then through the retinal ganglion cells, which form our optic nerve, project those electrical signals back into the visual cortex of our brain. Now, my focus is on retinal ganglion cells, how to generate new retinal ganglion cells from stem cells, and how to implant these cells back into the retina, hopefully to induce some regeneration. Retinal ganglion cells are the cells that are mainly affected by diseases such as glaucoma and diabetic retinopathy, as well as trauma to the head and traumatic optic neuropathies. Now, one of the central dogmas in central nervous system development and regeneration is that the central nervous system is something that's fixed, ended, immutable, and this was developed, or this was proposed, advanced by Ramon y Cajal over 100 years ago. And in his foresighted work, he decreed upon us, the science of the future, to, if possible, change this harsh decree. Now, I don't know if we're at that stage yet, but I know that stem cells are playing a big role in our advancements of regenerating neural tissues, and hopefully we will get there someday. So there's been a lot of talk throughout the conference about the duality in nature of stem cells, or if you want to call them medicinally stimulating cells, as Dr. Kaplan just alluded to. There's numerous slides of evidence showing that transplanted cells in the eye that do provide this trophic or paracrine support for deteriorating cells that increase visual outcomes over a short period of time. However, the main focus remains in cellular replacement, in can we get a new retinal ganglion cell layer to form the connections in the retina and then eventually transmit these signals back into the brain. Now, my lab focuses primarily on induced pluripotent stem cells, and I'll talk a little bit about that. However, I'd be remiss if I didn't mention some of the challenges to translation in cellular replacement, and this is true for any tissue in the body, but especially for central nervous system tissues, where functional repair is going to be found in somewhere in the intersection between ideal regenerative cell sourcing. We need cells that are capable of forming the different types of retinal ganglion cells or different retinal neurons. Cellular delivery or engraftment, like I said, the retina is part of our central nervous system, so getting cells to actually engraft and get past the blood-retinal barrier or the vitreal retinal barrier is a challenge. And finally, the million-dollar question is, how do we get these cells to integrate into the existing neural circuitry? How do we get them to project to the correct visual cortex targets? As we know, our retinas are mapped, pinpoint mapped, all the way back to our visual cortex, and that's what allows us to create this coordinate system that we guide ourselves by. And Roger Sperry in the late 60s actually showed that in amphibians that are capable of regenerating the retina, if you cut out the eye and transplanted, rotated 180 degrees, the cells actually find their original targets, and the frogs in this case were actually aiming in a diametrically opposite way to where they were thinking that they were going to. So if the fly was up here, it would shoot its tongue down here, because the projections of the retina to the brain remained guided by the same signaling pathways. So one of the challenges in neural integration, especially in mammals, is figuring out what visual function or what vision patients that receive cellular transplantations are going to end up with. Is it going to be a scrambled image? How plastic is the brain to reinterpret some of these signals and regain our Cartesian coordinate system? So like I said, we focus mainly on induced pluripotent stem cells and more broadly on pioneering transcription factors. We know that this is Nobel Prize winning work by Dr. Yamanaka, where he introduced transcription factors that were involved in the mid-blastula transition. We know now that these act as true pioneer transcription factors, where they can bind their partial DNA binding motifs in repressed chromatin sites. recruit other transcription factors, and help open our chromatins to make more of the genome available for transcription, and that's how they regain their pluripotency. They establish these topologically associated domains, which we know are now the hallmark of pluripotency. And in that sense, we look at pioneer transcription factors in their involvement in regeneration, endogenous repair in different tissues. Now, in the retina, we know that lower vertebrates, reptiles, like the xenopus and the newt, are capable of regenerating the retinas. And some people have shown that it's through pioneer transcription factors that some of the cells within the retina are capable of de-differentiating and then regenerating some of the retinal neurons. So as part of a major retinogenesis program that we have ongoing in Bascom-Palmer, we have used rainbow animals to try and map some of the expression of pioneer transcription factors in the retina. And these are two examples of true pioneer transcription factors, ASCL1 being of the basic helix loop helix transcription factor family, and SOX2, which is one of the original Yamanaka factors. What we've been able to find is that these cells, even postnatally in the mammalian retina, remained in expression in Mueller glia, which are some of the cells that people have shown are capable of de-differentiating and giving rise to new neurons. Now, how these cells de-differentiate and then regenerate in the xenopus is something that's ongoing in our lab, and we're trying to mimic that in the mammalian retina. But we do know that following injury, and most of this is attained by activating a gliotic state in the Mueller glia, these cells are capable of re-expressing these pioneer transcription factors and undergo some level of de-differentiation. However, in the mammals, we've lost that transition from reactivation all the way back to regeneration. So we've been looking, and like I said here, over development, pioneer transcription factors, as you can see here, over the first 2 weeks in the mice, you lose expression of these transcription factors. And if you look at publicly available DNA-seq data, you can see that over the first 2 weeks, the DNA's hypersensitivity regions around the promoters of these two genes actually get silenced. They get clumped up into repressed chromatin. So one of the things that we're interested in is looking at promoter occupancy studies as well as epigenetic drug screening to try and reverse some of these repressive marks that get built onto pioneer transcription factors in the retina to see if we can elicit some sort of endogenous repair. Now, unfortunately, this is our long-term strategy, which we're still starting to develop. So until we can figure out a way of triggering endogenous repair, we're looking at exogenous stem cells and how we can generate new retinal neurons as a replacement therapy. So in recent years, induced pluripotent stem cells have been shown to be capable of forming self-organizing retinal cups given the right conditions. This was shown originally by the Sasai group in Riken, and then followed up by the Johns Hopkins group, where we now know that if you culture these cells in aggregate formation under the right conditions, you can actually get them to laminate into a newly formed retina. Both of these studies were looking at the generation of new photoreceptor cells for macular degeneration. However, we've found several other potential uses for this technology. One is, of course, a source of regenerative cells, but we can also study specific retinal lineage differentiation. So we've generated some of these cell lines from our rainbow mice, for example, and we look at how different transcription factors promote the differentiation of the different retinal layers. And then we're optimizing this technology to try and get a source of new retinal ganglion cells or retinal ganglion cell progenitors. Now, what we've found is there is this window during where you get the lamination stage of the different retinal layers where you have the appearance of a retinal nerve fiber layer. However, after day 60, and this has been shown by several other groups, that retinal nerve fiber layer undergoes degeneration. The main hypothesis being that since these cells never reach their targets in the visual cortex, they lose the trophic support and undergo the normal pruning that we see in excess retinal ganglion cells as we are born. So what we did is look within this window of lamination, and the first stage that we looked at was day 51 retinal cups, We went on to characterize whether we could find a nice retinal nerve fiber layer in these cells. And then we did a couple of things. One is we switched the retinal cups to an RGC differentiation and maintenance media. And as you can see here, even at day 83, we still find a very nice layer of Thy1 positive cells that express both the MAF5 or ATOH7, because these are human cells, and BRN3B. But then we also did dissociation of these retinal cups to look at the viability and the morphology of some of these retinal neurons that we have growing in the aggregates. So the natural next step was, this is a mixture of all retinal neurons that are formed in the retinal cup, but could we get a homogeneous population of retinal ganglion cells to try and transplant? So what we did is we again went back to our day 51 retinal cups aggregates. We digested as you would a normal retina. We did immunopanning for Thy1 as is done normally to get pure cultures of RGCs out of animals. And then we cultured the cells into laminin and PDL coated cells. Now, most of the immunopan cells are neuronal, and they express the RGC markers that you'd like to see. These are human RGCs. The cell viability that we were able to achieve is greater than 80%. However, the drawback here is that cells that were capable of extending any discernible neurites were less than half. There were actually roughly 38 to 40% of the cells actually extended any neurites, which leads us to the question is, are we selecting these at the optimal time where we have a synaptic plasticity enough for the cells to extend neurites, but at the same time they are not fully mature neurons, which we know are not amenable for manipulation, for extraction and then transplantation. However, what we've been moving forward, and as we determine, so we've gone back into earlier stages in this lamination window to see if we can find an RGC committed progenitor that's not necessarily a fully mature RGC cell. While we do that, we've been looking at ways of transplanting these cells into the retina. Now, most people deliver cells through the vitreous, which is the most adequate and probably the straightforward way of delivering cells. However, from several studies, and this has been shown for many, many years, transplanted cells into the vitreous very rarely make it into the neural retina itself. It's very variable as to how many cells are actually going to engraft in the right retinal layer and whether they're going to form connections in there. Nevertheless, as we've talked about the duality of stem cells and their application in regenerative medicine, some people have shown that even in the vitreous, some of these cells exert a neurotrophic support on the underlying retina and promote its survival for a moderate period of time. So how do we improve transplanted cell engraftment, regardless of whether they're iPS-derived RGCs or embryonic RGCs, as some people are trying to do? And we've been focusing on modulating the inner limiting membrane. Now, the retina being part of the central nervous system is very well guarded on the back end, the front end, as well as the vasculature that runs through it. The inner limiting membrane forms the barrier between your vitreous body and your retina. So the front of the eye would be over here. And this is where most cells get injected into when you're trying to recapitulate this top layer. The inner limiting membrane is formed by the glial components, primarily by the end feet of Mueller glia, some displaced astrocytes, and it has a basement membrane component to it. The idea is, can we temporarily disrupt these connections between Mueller glia to try and enhance the engraftment of cells in the retinal nerve fiber layer or the retinal ganglion cell layer? So what we did is we took some samples for inner limiting membrane and we submitted them for unbiased proteomic screening through mass spectrometry. And we started looking or filtering out what was considered to be cell adhesions, tight junction markers, as well as other cell-cell adhesion markers that we could attribute to end feet and tight junctions being formed by the end feet of the Mueller glia. And then we confirmed some of these by immunohistochemistry, and here you can see NCAM1 was found primarily in the inner plexiform layer of the retina, so it's probably not the best target to try and knock down, even temporarily. However, one of our first targets that we started looking at is cadherin-2 or N-cadherin, which we could localize very nicely with Mueller glia to the inner limiting membrane. So the first step here was to culture some Mueller glia out of mice and start looking at RNA interference targeting sequences to see if we can knock down N-cadherin and the cells would remain viable. We screened roughly 20 different sequences for an shRNA for N-cadherin. We could see a very nice knockdown both of the gene expression and the protein expression. And as you can see in culture, the cells lose their tight junctions and adopt a different morphology. And then the question became, can we deliver this in vivo? Mueller glia are notoriously for being difficult to transfect in vivo. There's some people looking at AAV serotype 9 capsids that have some tropism for Mueller glia, but we started looking at different RNA interference methods, and what we ran across was self-deliverable RNAs. So this is a technology which is a double-stranded RNA for RNA interference that gets chemically modified. It has these moieties built onto it or cross-linked onto it that allow it to be membrane permeable, meaning you don't need lipofectamine or other transfection reagents for them to cross the membrane, and they make them more stable. Now, the stability, as we've seen for some of our sDRNAs, is 3 to 4 weeks in vivo, after which the sDRNA is actually lost. We looked at a sample sDRNA. This is a scrambled control in the retina, and as you can see here by the Cy5 marker, it does a very good job of transfecting throughout the retina and including the Mueller glia feet. So again, we used the company Adverna to manufacture some of our sequences, and we saw some that had very nice effect in knocking down N-cadherin. This is our CD24 candidate. And both at the gene expression as well as the protein level, we got a very nice knockdown. So we went ahead and started using this in vivo to see if we can increase cell engraftment into the retina. We used an optic nerve crush model and then delivered intravitreal sDRNA. We did 3 micrograms of the sDRNA 2 days prior to introducing GFP-labeled dental pulp stem cells, and then we created flat mounts to look at where these cells were actually engrafting or where they were getting to. Now, if you can see here, the scrambled sDRNA produced similar results as you get with introducing no knockdown. You get some cells that actually make it to the nerve fiber layer, but very few of them. After we knocked down N-cadherin for a couple of weeks, we could see a vast number of cells had actually made it into the retina. When we went ahead and looked at 3D, you can see here in the control animals, the GFP-labeled cells remain on top of the retina, presumably sitting on top of the inner limiting membrane. This is the vitreous and this is your retina. Here, the nerve fiber layer is labeled in magenta, whereas after we treat with the N-cadherin opening sDRNA, we could get the cells to engraft better in there. And I have, again, these are videos, if you could play them, please. Yeah, so this is a 3D representation of what I just showed you in the confocal, showing that the cells sit on top of the retina in the control animals, and then the other video will show a very nice integration of the cells within the nerve fiber layer of these animals. So in summary, there is a potential source for endogenous regenerative cells within the mammalian retina, and we believe these to be the Mueller glia. We're still working on the epigenetic modulation of pioneer transcription factors that can be exploited to cause some endogenous repair coming from these cells. Hopefully I've shown you that iPS cells are a viable source for new retinal ganglion cells. We still have the question as to what the epigenetic memory of the parent cell line, how it affects our obtained phenotypes. We've had some issues creating some of the retinal cups from blood reprogram cells. However, when we do some of the naive pluripotency cultures, when we use HDAC inhibitors and some of these epigenetic modulators to try and regain a ground state pluripotency, we're able to recapitulate their capacity to form neural tissues nicely. I showed you that some of these retinoid aggregates can be used as a viable source of new RGCs, some of which extend neurites. We're looking at how improve the recovery of neurite extending cells and how early we can obtain a truly RGC-committed progenitor. We've known for several years that cell engraftment needs to be improved in order for cellular therapies to work in retinal ganglion cell replacement. Hopefully I've shown you that temporarily disrupting the inner limiting membrane can serve for delivery of cells. What we're moving into right now is combining both the iPSC-derived RGCs with temporary opening of the inner limiting membrane to induce engraftment. And then I have some studies, some preliminary studies into how we're working on getting these cells to integrate into the neural circuitry. Now, given the time constraints that I had today, I didn't include those slides, but if anybody wants to talk to me offline, I can discuss those studies with you. And then the million-dollar question is what functional recovery, what kind of vision are we going to get once we achieve this neural integration of the cells? With that, I'd like to thank my laboratory members, my collaborators in the Miami project, as well as in the oncogenetics group that have helped me with some of the genetic studies for the pioneer transcription factors, the group at Adverna for the production of the sDRNAs, and my funding sources. Thank you very much. All right, now I'd like to introduce Dr. Jason Mayer. He's from Indiana University, and he also works on iPS-derived RGCs.
Okay, so acknowledgements. I'm done, I guess. I'm going to try to go back. Usually acknowledgements is at the end. There's a way back there to switch to the beginning, so I'll just go through everything. That would be great. There we go. Thank you. Okay, so it's my pleasure to talk to you today. I'm excited to talk to you about some of the work we're doing. I think that hopefully this will be quite complementary to the work you just heard about. My lab also works on retinal ganglion cells that we derive from induced pluripotent stem cells. And some of the applications that we can use these retinal ganglion cells for, with the focus so far largely on in vitro assays that can then be translated, hopefully eventually, to more in vivo applications. So, of course, to start off with, I will just refresh your memories from just a few minutes ago. Retinal ganglion cells serve a very important role in establishing the connection between the eye and the brain. And if this connection is ever severed through any kind of disease or injury, it leads to vision loss, to blindness. So as you can see on this diagram here, with our eyes, there we go. So eyes up front here, the axons from these retinal ganglion cells extending back into the brain, either staying ipsilateral, going contralateral at the optic chiasm, finally forming their synapses back towards the visual thalamus, like the lateral geniculate nucleus. I feel like there are a lot of stem cell applications that we could take advantage of for diseases that affect these retinal ganglion cells, collectively known as the optic neuropathies. So these optic neuropathies include diseases like glaucoma, Leber's hereditary optic neuropathy, dominant optic atrophy, et cetera, But of course, of the list of the optic neuropathies, the most common is going to be glaucoma. Next closest isn't even remotely close, because glaucoma currently has an incidence of greater than 60 million people worldwide, with an expectation of exceeding 70 million by the year 2020. So we think the four diseases that affect the retinal ganglion cells, stem cells can serve in multiple functions. Initially, and perhaps more in the short term, we think these stem cells can be used for the development of an in vitro model so that we can study these diseases and get a better understanding of what's going wrong. And connected to that, also with applications for drug screening using these cells. Once we figure out what's going wrong, can we establish high-throughput screening mechanisms to identify compounds that might have a neuroprotective effect in rescuing these retinal ganglion cells. Perhaps a little further down the line, we think stem cells certainly have the potential for the replacement of RGCs following their loss due to some kind of disease or potentially injury. But to effectively derive retinal neurons from these induced pluripotent stem cells, or the HPSCs, meaning human pluripotent stem cells, in order to effectively do this, we need to make sure we have a thorough understanding of retinal development. How do these cells ordinarily develop in the human? And can we recapitulate this process in an in vitro situation to derive retinal ganglion cells? So the retina itself starts to be obvious quite early in development and starts growing out as these evaginations from the diencephalic region of the neural tube, indicated here in yellow. Shortly after this outgrowth in the neural tube, these structures here, known as optic vesicles, will start folding in on themselves to form this optic cup-like structure, with the inner layer here in blue giving rise to the neural retina, all the neurons of the eye, and the back becoming more the support cells, the retinal pigment epithelium. And through a series of rounds of proliferations and differentiation, we will form our neural retina here in blue with its support, the RPE, behind it. So our goal in the lab is to use pluripotent stem cells to derive retinal ganglion cells and then apply these cells for a number of potentially therapeutic options. So pluripotent stem cells themselves are found in two major classes. Traditionally, it's thought that pluripotency means embryonic stem cells over here on the left, which are going to be derived from the inner cell mass of the early blastocyst. Once these cells are derived, they can be expanded indefinitely in vitro and have been for about 20 years now and still going, and can also be directed to differentiate into any cell type of the body, as indicated in the bottom here with a few examples: muscle cells, blood cells, and nerve cells. More recently, as we heard before, Shinya Yamanaka's group, almost 10 years ago now, identified an additional path to pluripotency by taking adult somatic cells, such as skin fibroblasts that we see here, introducing key transcription factors that will genetically reprogram these cells to pluripotency. So now we have a pluripotent source with a non-embryonic pool there. And these cells can be patient-derived, so they could be potentially useful for transplant therapies. And like the embryonic stem cell counterparts, these cells can be expanded indefinitely and directed to differentiate into any cell type of the body. So when we differentiate these cells, after a few weeks of differentiation, these cells are growing as free-floating aggregates in our culture plates. And these aggregates, when we're looking at them after about 3 weeks of differentiation, we see two distinct morphological features. We see some of these aggregates, like these kind of darker, more shaded-looking ones here that really don't have many distinguishing features, and then these others that are a little bit lighter, kind of brighter ring around the outside. We use these morphological features to separate these cells into two distinct populations. Those with this kind of brighter ring around the outside, my apology that this laser pointer is kind of cutting in and out on me, but these cells with the kind of brighter ring around the outside, and what I'll point out about these is that in their three-dimensional state, while they look round in a two-dimensional picture, these are not spherical. These are somewhat cup-shaped, and we think these are very closely analogous to the developing optic cup of the eventual eye. This lower population down over here is indicated by this kind of darker, more shaded appearance. So when we separate these two populations, we can analyze their gene expression patterns. And we find that both of these populations are very highly neural in nature based on their expression of some key transcription factors, such as these three that we see here, that are expressed pretty ubiquitously through all the cells in both of these populations. So both of these populations are very highly neural. But when we dig a little deeper, what we'll find is that only this top population, with this kind of brighter ring around the outside, will express key retinal developmental genes, like CHEX10, RAX, and 6-6, as we see up top here, and these are absent down the bottom. These bottom ones will express genes like SOX1, DLX1, and EMX1, which are characteristic of the developing forebrain. These transcription factors are absent in this top population. So what we seem to get are two distinct populations after about 3 weeks of differentiation. One that seems to be kind of analogous to the optic cup, expressing a variety of retinal developmental genes, and another that seems to be more analogous to developing forebrain. And this really shouldn't be too surprising because it's coming from the same source, and the retina and the forebrain are very closely related. We also look in greater detail. Remember, these retinal populations seem to be more cup shapes. When we cut sections through these, as if you're cutting through top layers of a cup, you will have a hole in the middle. And that's what we find in these sections. These are kind of have a hole in the middle, but when you look at the cells that are there, these cells are very highly enriched for retinal progenitor cells, as indicated by the expression of these two transcription factors here. So at this stage, after about 3 weeks of differentiation, we have a very highly enriched population of cells that seem to be retinal progenitor in nature. They're also somewhat organized in nature. This is not just a random clump of cells, they've organized into this cup-like structure that we feel is analogous to the optic cup itself that we find during retinal development. If we allow these cells to differentiate a little further, we're interested in seeing, can these cells start laminating into different retinal layers, as we kind of heard about a few minutes ago. And with a little further differentiation, what we do find is that indeed we do see some layers forming where the inner layers of these retinal organoid structures are highly enriched for these BRIN3 expressing cells, which is indicative of the retinal ganglion cells. These retinal ganglion cells are found in the inner layers of these retinal organoids, just as they would be found in the inner layers of the retina itself. We also see expression of this protein recoverin, mostly on the outside, on the outer layers of these retinal organoids. Recoverin indicates the retinal photoreceptors. They're on the outer layers of these organoids, just as they will be found in the outer layers of the retina. So using this procedure, we find that we can generate these retinal organoid-like structures that bear a lot of resemblance to the developing retina itself. But we want to apply this for studies of the optic neuropathies. And we think that there are two main basically time points, or two areas that we really want to emphasize. If we're talking about the optic neuropathies, there are early stages of these diseases where we would have a moderate degree of cell loss and some loss of vision, and then the later stages of these diseases where there is more profound cell loss and a very severe loss of vision. And I think these are two very distinct and important differences here, because I think the treatments that will be developed for each of these time points will probably differ quite a bit in nature. Whereas for these early stages, it's probably best, since most of the retinal ganglion cells are probably still there at the early stages, our best approach is probably to develop an in vitro model with these iPS cells for this retinal degenerative disease, with applications then for screening compounds for some kind of therapeutic efficacy. So while the cells are still remaining, they might be damaged, but while they're still there and the neural circuitry is still set up, can we rescue these cells and restore vision that way? But of course, at the later stages of the disease, when we have more profound cell loss and severe loss of vision, there really isn't anything there to fix. So at this point, at these later stages of the disease, we're probably more likely to have to go towards methods that will explore the potential for the replacement of retinal ganglion cells following their loss. So when we differentiate these cells, what we find in this population, we have this nice little neural rosette going on in the middle here. And after about 40 days of differentiation, we start to see the onset of retinal ganglion cell-associated genes, such as this BRIN3 transcription factor. These cells will shortly start putting out wispy little neurites, as indicated by MAP2 standing here, starting to sense their environment. Within just a few days to a week, what we find is that we have a number of retinal ganglion cells, as indicated in this picture on the right, with their neurites extending in many different directions, trying to make some kind of contacts and sense their environment. This is also a pretty efficient process. What we find is that about a third of all the cells we differentiate become these BRIN3 expressing retinal ganglion cells. We wanted to find out also about the functionality. Not only do they look like retinal ganglion cells, but do they function as such? So in this DIC microscopy image up over here, we see some of these cell bodies, and we're looking for some of the morphological features we would expect, including some of the longer neurites extending from these cell bodies. We initially identified these cells based on these morphologies that seem to be retinal ganglion cell-like in nature. And then we did a patch clamp analysis to look for electrophysiological features. And upon doing this patch clamp analysis, we also filled these cells with the dye lucifer yellow, which is indicated here in green. So after our analysis, we confirmed these were indeed retinal ganglion cells based on the expression of BRIN3 in their nuclei. And of these cells that were confirmed to be retinal ganglion cells, we found these cells were capable of firing action potentials, as we would expect retinal ganglion cells to do. And in the retina, most of the action potentials being fired, or nearly all of them, one might argue, will be in the retinal ganglion cells. These cells also maintain a hyperpolarized resting membrane potential and exhibit inward sodium currents that can be blocked by the addition of TTX in a very voltage-dependent manner, as well as outwards potassium currents that can be blocked by TEA, also in a voltage-dependent manner. So what we find is that these cells not only appear to look like retinal ganglion cells, but appear to be functional in nature as well. So we want to then apply this to see, can we model features of the diseases affecting retinal ganglion cells, most notably glaucoma, because as I mentioned early on, this is a very prevalent disease with an incidence approaching 70 million people by the end of this decade. But especially in the Western Hemisphere, we typically associate glaucoma with elevated pressure within the eye. This is something that's going to be pretty difficult to model in an in vitro setting. So what we have turned to are some of the genetically inherited determinants of glaucoma, because we believe that these could be an effective tool for studying some of the basic cell biological functions of what's going wrong in glaucoma in the absence of elevated intraocular pressure. So we turn to mutations in a particular gene known as optineurin. And optineurin satisfies these criteria where this is a genetically inherited determinant and appears to have a lot of features that we would associate typically with the glaucoma-like process. So we've isolated some skin fibroblasts from a patient with a particular mutation in this optineurin gene, reprogrammed them to pluripotency, and to see here some colonies of these iPS cells we generated, stained for these pluripotency-associated genes. And in this mutation, this E50K mutation in the optineurin gene, it's a glutamic acid that's been mutated to a lysine. And this gene mutation has been conserved even after the reprogramming process. This is an autosomal dominant disease, so our N call right there indicates this autosomal dominant nature, and the karyotype of these cells remains normal. So even though this reprogramming process can sometimes be quite disruptive to the genome of these cells, in these lines that we have isolated, they are karyotypically normal. We then differentiate these glaucoma patient iPS cell lines to a retinal ganglion cell lineage, as I indicated before. Initially through these optic cup analogous populations of cells that are highly enriched for retinal progenitor markers like CHEX10, and eventually we can isolate populations of retinal ganglion cells, as indicated here based on the expression of BRIN3, as well as things like MAP2 and the metabotropic glutamate receptors. But of course, if these are cells that are derived from a glaucoma patient, we need to have some kind of phenotype before we can then start looking into drug screening purposes. So as we're differentiating these cells, we run them in parallel with an unaffected cell type. So we have our controls up over here on the left, and our glaucoma patient cells here on the right. In the undifferentiated state, these are undifferentiated based on the expression of NANOG, we start screening these cells for indications of neurodegeneration or cell death. Basically the activation of caspase-3. And in this undifferentiated state, you see very little red signal here, and there's no significant difference between the two. We start differentiating the cells towards the retinal ganglion cell lineage. They can become neural, and then retinal, and eventually retinal ganglion cells. And we don't see any difference between the two until we get to the point where they are retinal ganglion cells. And at this point, the retinal ganglion cells down below here are indicated in green based on the expression of BRIN3 in the nucleus. And when we look for the activation of caspase-3, we still see very little in our control cells, but there's this huge explosion of activated caspase-3 in our patient-derived cells. So clearly there is a lot of neurodegeneration going on that seems to be specific for our patient cells and our control cells. And this is a very significant difference between these two lines. So there is a basal phenotype that we can identify in these glaucoma patient cells. So now that we have a phenotype, our next application of these cells would be to explore the potential for these cells for drug screening purposes. So if we look at this stage here where we have all this cell death going on, all this red activated caspase-3, we looked into some candidate neuroprotective compounds, and can we establish a proof of principle that these cells can be used as a platform for drug screening purposes. And in parallel cultures, when we add some of these candidate compounds, like BDNF or PEDF here, we see a pretty drastic decrease in the activation of caspase-3, a decrease in the expression of this red signal here. This was found to be highly significant difference compared to our untreated glaucoma patient cells. So I think this data helps to establish a proof of principle that we should be moving forward now into more high throughput screening options to identify the most suitable candidates for a drug development purposes, particularly for the early stages of glaucoma, while there are still cells there to rescue. And let's see if we can develop drugs to rescue these cells. But it then brings us not to the early stage of the disease anymore, we'll focus on the late stage of the disease, when there's a profound cell loss and a severe loss of vision. With this profound cell loss, it means that we don't really have much there to fix. We're past the point of trying to fix it, and we're trying to get to the point now where we are replacing these cells. So we'll explore the potential for replacement of RGCs following loss. And we saw some of this very nicely in the previous talk. For any kind of in vivo experiment, it's important to be able to effectively track these cells. So we have collaborated with Don Zak at Johns Hopkins University and are using a fluorescent reporter to help to identify our retinal ganglion cells, a red fluorescent reporter. And in these retinal organoids, remember before the retinal cells were in the inner layers, so you see our red signal in the inner layers here, it seems to be pretty specific. When we histologically analyze these retinal organoids, we still see our red signal in the inner layers, and they seem very tightly associated with ganglion cell markers like BRIN3. And they do not seem very associated with photoreceptor-associated markers, which are more around the periphery, on the outer layers, like OTX2 that we see here. So it does seem like this fluorescent reporter is a reliable indicator of our retinal ganglion cells. But as we heard before, there are several obstacles to effective replacement of retinal ganglion cells. So to replace retinal ganglion cells in an effective fashion, it's going to require, well, probably many things. I'm putting 3 of the most broad topics I can think of. Each one of these can probably be broken down into a number of other topics. But first, of course, we'll have to have effective integration and efficient integration of these transplanted cells into the retina. And as we heard earlier, this is not necessarily the easiest task, particularly with the inner limiting membrane. If we are to achieve effective integration of these cells into the retina, we will then need to establish long-distance axonal pathfinding so the axons of these retinal ganglion cells can find their target tissue rather deep back into the brain. And once they reach that target, we have to make sure that the formation of synaptic connections with the target tissue not only occurs, but as we heard before, it has to occur in a very specific fashion in order for effective vision to actually occur. So one thing we noticed, I'm going to focus mostly on this second one over here, this long-distance axonal pathfinding, because we think this is one of the things we can be kind of testing a lot of hypotheses in an in vitro assay right now, and then applying them to an in vivo situation. And one of the things we noticed is that when we take these retinal ganglion cells that we can isolate based on their red fluorescence and plate them down onto an adhesive substrate, they extend very elaborate neurites in all directions here in a very short period of time. All these neurites here have extended within the first 48 hours of growth. So these retinal ganglion cells don't seem to have any problem extending lengthy neurites in a dish. They seem to really want to do so. And these neurites are not only extending like this, but if we look at the leading edge of any one of these neurites that's extending outwards, we'll see features that seem to indicate that they have a functional growth cone leading the way. So here's our red fluorescence indicating this is an axon from a retinal ganglion cell with an accumulation of filamentous actin in the growth cone, as we would expect. It seems like they are extending these growth cones at the leading edge of these neurites to sense their environment and trying to find their way. So we've set up a number of assays to try to optimize the situation. One of the things we've looked at is taking these isolated retinal ganglion cells that we've derived from the iPS cells and plated them on a number of different substrates. And what we find is that some of these substrates, within the first 24 hours of differentiation, extend pretty lengthy neurites, whereas others down here, well, really do a pretty poor job. And we've quantified this data to indicate things like laminin seem to be the best substrate to allow for the outgrowth of these neurites. This is also important because laminin is known to play an important role in axonal pathfinding of retinal ganglion cells out of the retina and into the optic nerve. We're also testing a number of different media to optimize this axonal outgrowth assay in our lab. And we find that some of these media components, especially these top two, seem to be very conducive to axonal outgrowth in our in vitro assay, whereas others do a pretty poor job. And what we find is that there are some media that are certainly more significantly leading to enhanced axonal outgrowth compared to others. So taking this data, now we're looking to see, now we've tried to essentially optimize this assay to encourage axonal outgrowth from these retinal ganglion cells. Can we then adopt this to try to really encourage these cells to extend long axons, not just over the period of 24 hours like these assays were looking for, but long term? What happens if we go several days or weeks? What happens to the outgrowth of these axons? And what we find is that under the right conditions, these axons indicated here in red, stained for their tau protein, are not so haphazard in the way they're extending. They start to organize, start to fasciculate as we see here, and all these axons seem to shoot out in the same direction. These are axons that seem to be deriving from the retinal ganglion cells indicated in green based on BRIN3, and the cell bodies and dendrites of these cells in blue. But what's really intriguing is the ability of these axons to coalesce together and all shoot off in the same direction. And this is this outgrowth, I really can't emphasize this enough. It is so extensive that we're actually having trouble quantifying it because we're often running out of space and we're not sure that the axon's actually reaching their capacity of growth, or they're just running to the edge of the cover slip, or they're running into some other cells on there, but it certainly seems to be going at least for several millimeters. So we think there's a lot of applications for these iPSL-derived retinal ganglion cells. We find that our retinal differentiation from these pluripotent stem cells to retinal lineages, including retinal ganglion cells, is both effective and highly efficient. And these approaches have a lot of power, can be exploited as a system to study diseases that affect the retinal ganglion cells, as well as potentially developing drug screening approaches to these diseases. And especially at the later stages of the diseases, when there has been profound cell loss, we believe the use of these cells is going to allow for the potential to replace these ganglion cells that have already been lost by the disease. And then I will finish now with my acknowledgement slides. Some of the people that have done this work, especially Sarah Olemacher and Clarice Flygor up front there, some of our collaborators and our sources of funding. And I thank you for the opportunity to speak here today.
I think we have a couple minutes in case there's any burning questions in the audience. Yep.
I have a patient in my practice in Southern California who woke up blind in his right eye, and he found out that a piece of calcium had broken off from his carotid artery and lodged into one of the blood vessels leading to his retina. Do you have a referral for someone? I'm not sure if the obstruction needs to be removed first or if stem cells should be efficacious. I'm wondering if you could refer him to someone. Someone said Duke University. Do you have a physician in Southern California where between Los Angeles and Santa Barbara is where my practice is? We're not too far from UCLA.
Yeah, no, I'm not really familiar with anybody in that region. No, sorry. Yeah. All right. Well, thank you very much.
There was a.
Oh yeah.
There was a study in which they used a special AAV a vector to help transport, let's say, retina pigmented epithelial cells to the retina, and these cells were put in the vitreous because apparently retina doctors often attempt to do it in the subretina space, and there was a complication possibility of a detached retina. And I wondered if you're familiar with that. I think it was the AAV 7MB that was used as the vector. And I thought because in the comments that were made, it seemed like you were implying that most of the treatments were put in the vitreous, and I wondered.
Well, they are when they're addressing the inner retina. For the outer retina or anything that's outside of the neural retina, like the retinal pigmented epithelium, subretinal injection is the most common way to go. Even for some of the electronic retinal transplants that are done today are injected subretinally fairly routinely with not many complications, yeah.
Okay, thank you.
Okay, thank you. All right, thank you again.
I invite some of our attendees that are in the back. There are seats in the front, and it always gives a great visual when we have some of those seats filled. So please feel free to move a little forward. Are we good to go? Yes, Mr. Todd? Okay, good. So welcome, everyone. My name is Bernard Siegel. I'm the Executive Director of the Regenerative Medicine Foundation, which is the producer of this, the 12th World Stem Cell Summit from the Palm Beach County Convention Center in West Palm Beach, Florida. This marks the end of day 3's sessions, and we have a plenary session with two wonderful presentations, one from Yuzo Toda of Fujifilm, and immediately after, a title, The Future of Human Aging, Implications of Induced Tissue Regeneration, with one of the iconic figures in our field, who I'll introduce later, Michael West, the founder of Geron, Advanced Cell Technology, and BioTime, among other accomplishments. As I've mentioned earlier in our meeting, this is the Japanese day, we like to say, of the World Stem Cell Summit. We recognize the accomplishments of Japan and in advancing cellular therapies. In an amazing way, the government, industry, education, great academic centers, regulatory system working in lockstep to try to find a way to accelerate potentially life-saving and curative technologies. It's not easy to be a path-finding nation, very challenging, but I have found our Japanese colleagues so open to collaboration for the sake not of just Japan, but humanity. It's very welcoming to have this very large delegation from Japan, including our co-chair of our meeting, Dr. Norio Nakasushi, former director of Kyoto iCEMS, and now leader of the Nakasushi Foresight Foundation. When I was in Japan last summer, I had the opportunity to tour the massive facilities at Kyoto University, probably the largest single collection of research scientists, some 600 under one roof at Kyoto, that are researching iPS cells, reprogrammed stem cells. It was a beehive of activity. I hope to see Nobel Prize winner Shinya Yamanaka in person, but he was in a big meeting, but I saw him through the window and I waved. One of the other very interesting things that I did is I went to the headquarters of the Forum for Innovative Regenerative Medicine. Yuzo Toda is the chairman of that august group of industry leaders, the industry organization of Japan that is moving forward regenerative medicine in an assertive and positive way. And all of these companies, and I assume some are even rivals, yet they are working together to try to advance the field. And indeed, one of the highlights was a trip down to Fujifilm, where I had a chance to meet Mr. Toda. And what was so fascinating about that, and if I just take a minute to say, Fujifilm, for those of you who don't know, we think of the time when you would go to Disney World and grab a box of film. It would be Fujifilm, or sometimes if you go to the drugstore, it would be Kodak. And the story of Fujifilm is an incredible story of decades, many decades, this company, a leading technology company, and built the great consumer brand, Fujifilm, and actually vanquished its rival in the end, Kodak, only to see the digital age sweep across and change the way folks and consumers took pictures. But Fujifilm was a very nimble organization and steeped in science, chemistry, and now life sciences. And the journey of Fujifilm to becoming a leading life science companies, yet proud of the brand that they had built up so over the years, keeping the name Fujifilm, which is very interesting, but now actually acquiring a United States company based in the United States, Cellular Dynamics, which of course was based on Jamie Thompson's technology and the acquisition of these valuable patents and working with the California Institute of Regenerative Medicine and banking. Fujifilm has become a dominant leader, not only in Japan and the world, guiding this field forward. Under the able leadership of their Chief Technical Officer, Director Yuzo Toda, who's the Senior Executive Vice President and Chief Technical Officer of Fujifilm Corporation, he is a phenomenal ambassador himself for his company and for Japan. Most notably, and what I think he's going to speak to us today about, is actually FIRM, the Forum for Innovative Regenerative Medicine, and how industry and all other elements of Japanese society are advancing our field. So please welcome Toda-san, Yuzo Toda.
Thank you very much.
Thank you.
Good afternoon, ladies and gentlemen. Thanks, Bernie. Thanks to Bernie's invitation here. I'm very honored to be here and to share with you our idea. Also, I'm chairman of the Regenerative Medicine Industry Association in Japan. So today I'd like to share with you regenerative medicine revolution. But also Bernie stimulated me. His introduction is Fujifilm, which he stimulated. When I was a freshman to join Fuji, my senior, a little bit older than me, asked us, "Hey, young guy, do you know what is Fujifilm's products?" No one could answer. He said, "Trust." That is so impressed for me. Still, I think whatever product, whatever service we offer, whoever customers, most important idea is what we have to give them. That is trust. Also, Fujifilm is now, our film is almost gone, nothing, almost nothing, but we change our business, but such type of core concept, we sell trust, is also applicable in this area. Anyhow, thank you, Bernie. You stimulated me. Also, you reminded me again, trust is most important product for us. Okay, thank you. Today's my presentation is here, environmental change in medical treatment. Keyword is aging country. Second one is definition of regenerative medicine. Keyword is cell science. And also revolution, medical treatment by cell science, role of industry, and conclusion from the industry. First, environmental change. Anyhow, due to super-aged society, we have two major obstacles and problems: increased national health expenditure, and also very low satisfaction with drug and treatment. Therefore, we need new approach. One is really we need reactive but radical treatment result is needed. Also, proactive treatment. Both reactive and proactive approach is really necessary. That is one of the key solution is regenerative medicine. I need regenerative of this one. Almost no right, sometimes okay. Anyway, so this is first idea to share with you. Second one is I also shared with you kind of aging country. Here is a population rate over 65 years old, which is, if that ratio is more than 21%, super aged country. Japan is 26.7, super aging country. Italy, Germany, France will follow. And also in Asia, China, Korea, still percentage itself is lower, but their steep curve is so much higher than Japan. So anyhow, by the way, US is still okay. But anyhow, aging is really major cause for our quality of life. This is national health expenditure in Japan now, more than 400 billion. That is 60 years ago, just 2.1. 180 times more. This is so astonishing. We have to do something. This is kind of health expenditure per person at each age range. So you can see tremendously increase from 65 or 69. That means, of course, a very serious expenditure, but not only expenditure. If you pay more and more money, that means your health condition is so serious, so-called double punch. Then this is our problem. So here, this is satisfaction with treatment. Horizontal line and vertical line is contribution of drug to treatment. This area is so-called unmet medical needs. Here I pointed 3 domains. One is cancer, diabetes, dementia. This is major problem, but still we don't have real key to solve, so-called unmet medical needs area. But this, do you know why? This is all aging-related disease, not simple disease. So in that sense, we have to change kind of concept. Until so far, our approach to disease is kind of attacking single target. So here is low molecule, the medicine and the biologics, and here cell. I think this is better. No. Okay, so this is single target case is single molecules and single target. This is one-way approach. Also, biologics, antibody, also single target, but single polymer and single target. But most of the case, especially in cancer, this is multi-target, so not applicable. So we have to change paradigm or basic concept to utilizing self-healing ability. We have our body have this kind of ability we have. Then cell will appear as an essential solution. Single cell, even single cell, can adapt to a normalized problem. This is kind of idea. Okay, so still I'd like to identify 3 major areas. We need novel drug for new infections. Low molecule is still we need because infection disease such as very toxic influenza, HIV, HCV, HPV, still very serious problem. So in that sense, a new drug, new approach to infection is still very important area. Also, second one is second generation antibody is needed. So local battle single target antibody is not valid anymore. So for instance, armed antibody or PD-1 like immune system control antibody is second generation. Third one is, I mentioned already, biocontrol system utilizing cell. So immune system and gene and tissue, etc. This is so-called microenvironmental control, utilizing self-healing ability. Here is a cell therapy and also target of regenerative medicine. So this is 1, 2, 3 is so-called new target, new area. So I'd like to summarize first part. First one is new drug, and second one is utilizing immune system for multiple mobile target, and third one is interactive treatment to control abnormal environment. Then cure completely, and the new options, preemptive medicine. This is first paragraph. And second one is definition of regenerative medicine. In the first part, I shared with you, we need kind of new approach. One of the most important key factors is regenerative medicine. But also here, I'd like to define what is the new regenerative medicine. Here, regenerative medicine is one of the application of cell science-based medicine. Also cell therapy, also tissue engineering, also more and more complex kind of human body reproduction kind of system or device or tool is needed. But also only cell cannot reach these kind of value chain. Also we need engineering to support cell performance, also to optimize cell performance, also to maximize cell performance. We need different type of science. In one word, engineering. Cell science and cell engineering, cell science and engineering can perform what we needed. I'd like to skip here. Also, medical treatment. Thank you very much. Good. Now I'm completely recovered. Here is kind of statistic data. Here is healthy people in Japan. Healthy people is 53%, but half health is 20, and a little bit sicky is 20, real sick person is 7%. So here, from here, we start kind of cure, but it is sometimes too late. For instance, using regenerative medicine from here, this area, regenerative medicine is kind of essential treatment method. Therefore, our cure area is expanded. So therefore, our lifetime become longer. But also, this area, preventive area, regenerative medicine can contribute a lot. For instance, from here, preemptive medicine using regenerative medicine expand healthy lifespan. So to balance with preventive and reactive approach utilizing regenerative medicine, our life become more and more successful. Not anti-aging, but we can establish successful aging using this one. Of course, we have a big obstacle or problem to be tackled. One is early detection. To do some preventive action, we need early detection, for instance, biomarker, and also gene diagnosis. Also, a lot of scientific thema are lying in front of us. Also, early intervention, of course, drug and vaccine, and also IT, kind of big data of our total life is also very, very important. So this is a kind of basic idea. Third one is, I'd like to share with you some example of regenerative medicine. Here is ocular program for RP and PRD. This cell is already delivered by CDI, our group company, but also these two should be designed like engineering, like underlayer and overlayers, these two. This program already started together with NIH. So also Japanese our subsidiary company. JTEC is a professional company for making cell under GMP condition of Japan. So here is final kind of structure of ocular program. This is one of the example. Also, this is diabetes program. The example here, the diabetes mouse here, the blood concentration, glucose here, even implantation of beta cell itself is no difference between non-treatment diabetes mouse and implant islet. And implant islet and mesenchymal stem cell combination works very well, decreasing significantly glucose level of the blood. But this is still not good enough. Utilizing our kind of three-dimensional cell aggregation technology here, utilizing special recombinant peptide and special structure like this, then completely same glucose level of normal mouse. So this is really revolutionary treatment utilizing cell science and engineering technology. This is from Fujifilm data. So this is kind of summary or overview of the cell therapy product under pipeline and already in the market. For instance, autologous and allogenic, and somatic stem cell, and iPS and ES cell. This is overview. So now, really, regenerative medicine and cell therapy is just started, and in front of us there are so many kind of promising results will come soon. So next agenda is drug discovery and development system, not only for therapy, also this is very important outcome from regenerative medicine. Here is from preclinical to the end, We utilize animal cell and small mouse and chimpanzee and phase 1, phase 2, phase 3 monkey. Sorry, not chimpanzee, monkey. And here, if we utilize cell, like I mentioned here, this is just estimation, but not far from the real data, real result, I presume. For instance, success ratio is now is only 11% from preclinical, and also development cost in average is $900 million, and also total period is 10 to 15 years. If we utilize cell system, human cell, not small animal, success ratio become 55% from 11, 5 times more successful, and also cost is 5 times more effective, and also time-wise, 1/3. This is really, really revolution for even drug discovery and development. Please remind, we pay a lot, too much, for medical treatment nowadays. So this is some example what I'd like to skip for medical drug discovery. So almost end of my presentation is role of industry I'd like to share. Industry's role is very, very important. For instance, here is the research or scientific result. And here is the market, patient here. In between us, there are very big and long and deep kind of gap. For instance, what type of result we have to search. Also, business strategy is very important. Also, regulatory approval. Also, manufacturing, high quality, high trustfulness, reliability, cost, and also marketing, how to bring. So here is a lot of brick to fill. So anyhow, this is, I don't want to mention in detail, but for instance, business model and the clinical trial and money, funds, regulatory application, et cetera, is very, very important. For instance, I want to introduce Japanese new regulatory framework, one of the most important part. This is kind of drug regulatory framework, phase 1, phase 2, phase 3. This is regenerative medicine. We, Japanese, together with academia and the industry and the government, we implemented a so-called conditional term-limited approval system. Because this is not compromisation, this is due to science. So regenerative medicine is, in principle, personalized medicine. Depend upon the patient, the recovering condition is so different. Therefore, we implemented conditional approval system here. If we show confirmation of safety and also some possibility of the efficacy, we can get conditional approval. And after conditional approval, we have to collect all patient data, and if that is successful, here is kind of final approval we can get. This is a very unique and flexible system. Of course, safety is no kind of flexibility. We have to show the safety is same as drug approval system. So in this sense, I want to mention, in principle, most likely we can get from first clinical trial to the end before a conditional approval, in average, 2 years or 3 years are expected. This is, industrial association members nowadays in Japan, more than 200. Among those, so there are a lot of different variety of the industry. One is regenerative medicine and cell therapy, biotechnology, pharmaceutical company, 47. Also device and machine company, 37. Chemicals and material company, also 47. And also logistic, insurance, consulting, etc. is 73. This is, we have a very good relationship with ARM in the US. Same type of organization. Here, one of our activity I'd like to share with you. One is due to a Japanese new regulatory system, a lot of foreign company and academia and venture are interested in to start in Japan regenerative medicine. Therefore, we organize kind of crossroad meeting chance as a firm. So we are kind of consult and to realize the business in Japan, etc. We support here. And also we implement a new showcase very close to Haneda Airport. This is Life Innovation Center here. This is part of the building here. Here is a gathering member of the firm and represent and small experiment, also as example, also even a customer can try some experiment here. So this is kind of showcase of Japanese regenerative medicine industry. Both these two is newly opened. Here is also kind of worldwide collaboration from North America and Europe and Asian Pacific. We have globally the same types of organization in worldwide, but regenerative medicine is, we have a lot of problem to be tackled, to bring to the patient. So therefore, we have to collaborate worldwide. This is one of the example of collaboration worldwide association. This is my final conclusion. Between science and market, we have a lot of big barrier, gap. Also, academias, together with academia, industry, and also hospital, kind of real place for patient. Nevertheless, the role and responsibility is different, but target or kind of mission is the same. So we need open innovation to bridge here. Also, finance support is very important as a business model. Also, suitable, optimal regulatory is also very important. This is just, I'd like to share just in case. Thank you very much for your attention.
Yes, thank you. Thank you, Mr. Toda, and it was a very informative talk, and we all appreciate it. So for our final keynote presentation of today, I'm going to call up in a minute a good friend of the Regenerative Medicine Foundation, and I would call him one of the great pioneers of stem cells in regenerative medicine. And of course, that's Dr. Mike West. Mike, you know, is one of these iconic guys. One of the first books I read when I became an advocate was a book called Merchants of Immortality. And sure enough, Mike was described with his work at Geron. He founded Geron, Advanced Cell Technology, and now BioTime, that is doing pathfinding work. Mike, it's hard to start and be a pathfinder in a field as new as cell therapies. And Mike wrote a book that explained the core of his passion. And it's one of my favorite books that I've read in the field. It's called The Immortal Cell. And there was one particular point or passage in the book when Michael, his company funded some of the work of Jamie Thompson at the University of Wisconsin, and he was working in non-human primate embryonic stem cells. And Mike, when he saw that through the microscope and saw the dynamics of the cell in a dish, and he said, this is like nuclear fission in a dish. It was so incredible, such a powerful technology, that he knew at that point in time that this was going to change the course of medicine and indeed of human history, possibly, and impact everyone because of the power of cells. And that's one of the reasons we're all here today. Mike's resume speaks for itself, but I want to welcome him to the podium, where he's going to talk about the future of human aging, implications of induced tissue regeneration. Let's welcome Dr. Michael West.
Thank you. Thanks for the nice intro. Well, welcome everyone to the future of aging. What I'm going to try to do is talk about the prospects of life extension in the 21st century, and some concrete ways at which we might get there. In doing so, I'll be making certain forward-looking statements, and you know, we're a public company, and so I refer all of you to our filings with the SEC, the Securities Exchange Commission, for more detailed information about the company. So let's start with a statement of the problem. Maybe it's obvious. We age and we die. Life doesn't start out so bad. So, you know, when we're growing up, and I have some adolescents at home, it's really a joy to watch them grow and develop. And, you know, we have all of our lives ahead of us. But then we reach this apex and then begin that slow descent and aging, that inevitable process that has, as we've been talking about during this conference, has the prospect of all of this degenerative, chronic disease that can rob life of quality of life and can cause such a burden on our family and on our society. One of the challenges we have as gerontologists has always been, and maybe it's starting to change a bit, a difficulty of perceiving that we can ever understand and intervene in such a profound problem of aging. But it shouldn't come as a surprise that, like all of biology, there are clockwork mechanisms at work in aging. Any of you who go, remember the old carnivals, you go and people can guess your age, and if they're wrong, you know, you win money. We age on cue. And so many age-related degenerative diseases are an inevitable process of aging itself. And so nature is giving us clues as to what is behind this biology. And we need to pay attention. to those clues. I'm reminded just now of the Michelson-Morley experiment, and Albert Einstein recognized nature's telling us something here, and it led to the theory of relativity. Well, if we don't look and pay attention to those lessons of nature and just look at aging very simplistically, we could say, well, it's like entropy. Things go toward disorder. So a complex biological machine like a human, it's very easy to imagine that we just fall apart. After all, that's a law of nature. It's the second law of thermodynamics. Things tend to go from ordered state to a disordered state. In fact, maybe it's not so much a question of why we age, but maybe it's more of a question of why do we live as long as we do, given all the damage and wear and tear that we experience during a lifetime. You can think, for instance, of the free radical theory of aging. Many of you have heard about, take antioxidants because they fight free radicals that rust the body the way a car rusts as it gets older. Well, I would argue that nature is telling us something very different. And so my challenge is to try to communicate where modern gerontology is at. Here is an experiment not done by humans, but done by nature itself. So on the left, we see Werner syndrome. This is a premature aging disease that causes many manifestations of age in the 30s or 40s. And on the right, you see Hutchinson-Gilford syndrome, which is a premature aging disease. You've probably seen these children on television. It's really tragic. I mean, they have, if you look at this young boy here, graying of hair, around the age of 10 or 12, profound loss of hair or alopecia. If you look closely, you can see liver spots on his skin, the age pigment, wrinkled skin, osteoporosis, cataracts, heart disease, coronary disease. They generally die of a heart attack in their teens. But it's not because of type A personality, it's not because of lack of exercise, it's not because of a high cholesterol diet. Indeed, they have normal lipoproteins in their blood. Both of these diseases, of course, because of modern genomic technologies, are completely understood now on a molecular level. We know exactly the genes that are involved in this, and I would argue that they have essentially nothing to do with entropy and wear and tear. Another lesson that nature is giving us about aging can be seen in the life cycle. So what we see is a constant succession of similar individuals, but the individuals themselves passing away. And hidden within this picture is a profound message. I don't even have words to express how profound this is, but I'll try to walk you through the science behind it right now. So in the 1800s, with Schleiden and Schwann's cell theory, scientists recognized that the human body is composed of cells. And then in the mid-1800s with Darwin, there came together this grand synthesis. And the idea was life began with single-cell organisms that didn't age or didn't have to age. Instead, they just replicated indefinitely. And so they were called immortal. It didn't mean that they couldn't die. It meant that they didn't have to die. They could continue an uninterrupted proliferation. And then in a sort of debate between Darwin and a German naturalist, August Weismann, a brilliant naturalist, really, saw that here we had, embedded within the cell theory and the discovery that there is a human mammalian egg cell, that there is a mechanism that could explain heredity. So Darwin's rather contorted ideas were wrong, and Weismann proposed, as I'm showing here, that there's a lineage of cells at the top that continue to replicate just like that original single-celled organism that we came from. And then some point early in evolution, the cells clung together in a simple multicellular organism like Volvox that I'm showing you here. And that for the first time in the history of evolution, we had the evolution of programmed aging and death. And so somatic cells, cells that branched off from that immortal lineage, made sort of a soma. And the somatic cells, Weissman predicted in around 1881 in an essay called The Duration of Life, he predicted that somatic cells had a finite capacity to divide. And so aging of humans and mammals was caused by a limited capacity of the human body to regenerate and repair damage. So now let's move forward in time. 1960s. Many of you know this story. Here you're seeing Dr. Leonard Hayflick. Hayflick was an early pioneer in cell culture. He was developing cultured human somatic cells from the lung as a means of making vaccines. Billions of people are, by the way, walking around with the products made from these cells. They actually came from an aborted human fetus. They're fetal fibroblasts. And billions of people have received vaccines from these cells. Dr. Hayflick, in culturing these cells, had a problem. They would proliferate and then stop. And he was puzzled. He thought he had to be doing something wrong. And one of his colleagues one day in the lunchroom said, "Len, maybe your cells are just growing old." And he developed that into what is today one of the leading models of, laboratory models of biological aging. So the thought of many aging researchers are, if you want to understand human aging, use the fibroblast model. Of course, some other people study fruit flies and yeast aging and different kinds of aging. The concern, of course, is whether that has anything to do with human aging. So here we have on the left young WI38 fibroblasts, and then on the right, after a finite number of doublings, there's a clockwork mechanism at work. These cells stop dividing. Many researchers thought for years, this is so complex, we'll never understand it. There were hundreds of changes that occur in aging cells in vitro. And the consensus in even the gerontology community was hopelessly complex, we'll never understand it, and certainly never been able to intervene in it. Well, as you can see here, in 1986, a researcher named Howard Cook was measuring telomere length, and he was interested in how it was changing, and he put this figure in a Cold Spring Harbor Symposium publication, and he said, you know what? If you look at the ends of the DNA strands, the telomere, they're constant with time in, for instance, sperm cells, male reproductive germline cells. But if you look in tissues of the human body, if people of different ages, they're shortening with time. Now, there was a Russian theoretician, Alexei Lovnikov, who predicted that the telomere, which I'm illuminating here on the end of the chromosome strands, was actually a clocking mechanism behind the Hayflick phenomenon. Lovnikov's theory, called marginotomy at the time, now the telomere hypothesis, predicted that as the cells replicated, there was a defect in replicating the very terminal strands of the DNA molecule. And so the way we now measure telomere length commonly is to use restriction enzymes, that digest DNA into little pieces. And then here you can see what it's called a TRF, or terminal restriction fragment, from the end of the DNA strand to where a restriction enzyme cut it closest to the telomere. And so telomere, a TRF is telomere plus some subtelomeric sequences. So it's not a precise measure of telomere length, typically. But what was seen was that as somatic cells aged, telomeres were indeed shortening. And so here on the right, you can see how we typically used to measure telomere length on a southern blot, and you can see the intensity as cells double, the intensity diminishing and the telomere length shortening, or on the left here, a graph of how telomeres start at the beginning of life around TRF length, starts at around 15,000 kilobase pairs, shortens until you reach the Hayflick limit. Now, something funny can happen. If you hit the cells with oncogenic viruses that can turn a normal cell into a cancer cell, the cells continue to replicate beyond what they normally would to really short telomere lengths. This was one of the clues that attracted my attention, because when you do that, there's a later onset senescence, and telomeres are hooking together. You get what are called dicentrics. And that was a clue that the telomere could indeed be the clock. Are there exceptions? Sure. Cancer cells somehow escape senescence and become immortal and immortalize. The famous HeLa cell line that you hear about, Henrietta Lacks cervical cancer cell line, HeLa, is an immortal cell. And so the telomere hypothesis predicted that telomerase, an enzyme that's present in the reproductive lineage, is absent in somatic cells, causing telomere shortening, causing Weissman to be right, that somatic cells have a finite lifespan, and it's because of absence of telomerase. But could it be that simple? Another important clue was, as we were studying cell aging in the dish, it became clear that when cells arrest in their replication in senescence, they turn ugly. So here I'm showing on the top, on the left, a normal cell in its resting state. If you wound a tissue, cells become activated, and you can see they make, for instance, increased levels of MMP1. That's collagenase. That, for instance, dissolves a tadpole tail and makes it resorb. And so we remodel tissue, and then cells go back and rest again in the quiescent state. But senescent cells are arrested not in a quiescent state, but in an activated state. They're sort of like a car with the accelerator and the brake both down at a traffic light. And the result is senescent cells can cause a lot of tissue damage if they would accumulate in tissue. So here what I've just laid out for you is sort of a time bomb of aging, a clocking mechanism linked to destructive activity, elevated metalloproteinases and things that can destroy tissues, thin the skin, such as you saw there in the progeric patient, and so on. Could it be that simple? Well, one of the tests was, could we transfer telomerase from the germline into somatic cells and find out? That was what the major goal in the early days of Geron. So we had to find the molecular components of telomerase to test the causality of this whole mechanism. In this diagram, you can see coming in the telomeric strand, and then the finger and thumb modality of the catalytic component of telomerase that actually makes the telomeric repeat. So it's actually extending out the fuse, so to speak. And then it carries, it's a very unusual enzyme, it carries with it an RNA template that allows it to make the TTA GGG repeat on the ends of the telomere. We cloned the RNA piece and saw that it was expressed both in the germline and somatic cells. But when we cloned the catalytic component and looked in mortal cells and immortal cells, bingo, it was not present in somatic cells. And so we got to have the fun of dropping this gene into somatic cells, one gene, to see if it would immortalize cells. As Bernie was saying, there's a fun story behind this. Len Hayflick's skin cells from his leg were the first cells ever to be transfected with telomerase. And as many of you know, this gene can immortalize cells without transforming them into cancer. So what this led to in the mid-90s was an interest in, well, how do we use this technology, and could we capture the germline in the dish? because we would predict that the cells would be naturally telomerase positive, and this could open the door to what we've been talking about this conference, of regenerative medicine. So if we look at how development proceeds, it recapitulates evolution. It's called ontogeny, recapitulates phylogeny. And the thought was that very early in human development, the cluster of cells that forms from the fertilized egg are sort of like that volvox I showed you before. And the cluster of cells inside that were the immortal germline lineage are shown here as the inner cell mass of a blastocyst embryo. If we could culture the inner cell mass of a human blastocyst, we will have captured the human germline. And the magic that Bernie referred to is when I was meeting with Jamie Thompson, I realized Jamie had filed a patent on primate embryonic stem cells. It potentially could extend to human, and you would have a patent on the human germline. These cells are capable, all of the cells in the human body have no dead ancestors, right? We're a continuation of a lineage of cells that go back to the dawn of life on Earth, despite what people say about entropy and wear and tear. But going forward, our cells normally don't have that potential, but these do. The cells you're seeing here have the potential to make humans for the indefinite future. They are the human germline in the dish. Now, what most people picked up on was right off, where these were the master stem cells of the human body. They're capable of branching off into all the cell types of the human body. And, it's just amazing that up to this point, we can repair a car and keep an antique car going, manufacture parts. We've never had a manufacturing modality for the cellular components of the human body. It's an amazing omission. But now we had an immortal master cell bank of human embryonic stem cells that can proliferate indefinitely and then be used for the directed differentiation to make young cells of all kinds, neuronal cells. There's a lot of talk about RPE cells, heart muscle cells, but this list of cell types in the clinic being developed will continue for many decades to come. 1997, this is when Bernie entered the field, the cloning of Dolly the sheep. You see another example of what people thought was physically impossible being quite simple. Who would have thought that you could find the clockwork of cell aging and one gene would stop cell aging? Who would have thought that a somatic cell, when transplanted back into an egg cell, could reprogram itself and make a whole new animal? Keith Campbell used to tell me, who cloned Dolly, said everyone thought I was bonkers. No one believed this would work. But it does work. So somatic cells, when transplanted into an egg cell, can reprogram cells back into pluripotent stem cells. This technique, of course, is really laborious and technically challenging. It typically takes weeks to months to train people to do this. Yeah, it looks easy on a video with a trained technician, but you remove the chromosomes from an oocyte. Here is actually an aged somatic cell, a cell at the end of its replicative lifespan, an entire cell. We call it nuclear transfer. It's really cell transfer. Put into the intravitelline space and then given a little electric shock to fuse the membranes and trick the oocyte into thinking that was a sperm cell that fertilized it. Then if you turn it back into, in this case, a bovine clone, a cow clone, you can see here in this figure from a published paper the normal replicative lifespan of the cells. If we took the cells at senescence and transferred them back into an egg cell, we restore, and indeed beyond the original lifespan of the cell, we rewind the clock back to the beginning of life. Amazing, but true. But of course, the implications for human medicine are staggering. What that means is this immortal lineage of cells that can keep making humans, generation after generation, could be used to make new and young heart muscle or new or young neurons for you and me forever and ever. Nuclear transfer is a challenging technology. Who would have thought that we could reduce this down to just a few genes? I remember a discussion with Mark Tashney back in the days of nuclear transfer, and we had a few glasses of wine, and I asked Mark, Mark, it's probably transcription factors. How many, you think? And he took a long sip of wine and said, 4. Who would have thought? So now defined molecular components in induced pluripotency can take a cell back. And here you can see effectively reset telomere length and of course restore pluripotency to the cells. And reset cell lifespan back. It's not just telomere length. Now, of course, what this opens up are all kinds of opportunities. One of the most difficult, I think, to fully envision is the profound synergy and mixing now of this revolution in next generation sequencing with cell biology. If we kind of put all the cards on the table, I'll put them all on the table. The top left are my skin cells, and then the middle top there are my pluripotent stem cells. Those could become a baby Michael West. And on the right is confidentially my genome. What does all this mean? It means we can go into an immortal substrate like my iPS cells and engineer anything because the cells will replicate indefinitely. We can expand genetically modified clones of cells indefinitely. I can make my cells engineered in any way imaginable and make cells of any kind. And so the possibilities really extend beyond any imagination of any person that I know on the planet. But put simply, in the context of aging, it means that whether using off-the-shelf pluripotent stem cells or personalized reprogrammed cells, we can make, as I said, genetically modified or just normal cells of all types that are young, are the cells that we had that when we were born. Now, some of the products that are in the clinic right now are, first-generation products, RPE cells. Why are people so excited about RPE? In the back of the retina, you can see here this layer of retinal pigment epithelium. It's a really critical cell type in maintaining the retina. It repels the vascular bed on one side and nourishes the neural retina on the other. All animal retinas that have a neural retina have RPE. They're critical cell types. We've never published this, but RPE age, they lose telomere length, and then with age you see these patches where there are senescent or a complete lack of RPE, and you get the invasion of the retina by neovascularization, which is the wet form of macular degeneration, or the dry form, which is 90% of the disease, for which there's currently no therapy. A massive problem, hugely debilitating, easy to make, solid mechanism of action, a low dose of cells required, a relatively immunoprivileged site. It's the hallmarks of a low-hanging fruit on that tree of human cellular development. The cells can be injected in solution, as you see here on the left, and can then reform the simple monolayer in the back of the eye. So it's really an ideal clinical indication. These cells are being developed by a subsidiary called CellCure Neurosciences in Israel. Another application we've heard about worldwide, really. We heard just the previous talk about Japan, but it's the worldwide problem is obesity and associated type 2 diabetes and hypertension and coronary disease, the whole metabolic syndrome. Everyone knows about we need to lose weight. The problem is we don't do it. I mean, it's a joke, right? I mean, every New Year's Eve, it's a resolution, we don't do it. It's a massive problem. What we've learned in the last few years is there's hope of a new kind. We know about fat, white adipose tissue, WAT. So you eat a lot, you store it up. what's been a puzzle for years is why the redistribution of fat? Why do we gain it as visceral fat, abdominal fat? Why does it build up around the coronaries? Well, now we've learned about another kind of fat called brown adipose tissue, or brown fat. It does the opposite of normal fat. It burns calories. It sops up glucose like crazy out of the blood, metabolizes triglycerides. It's a furnace that normally balances metabolism. The problem is, look at this on the left. That's the amount of brown fat in individuals as they age, a precipitous loss. When you're born, you have a lot of it behind your shoulder blades. My son is a teenager. He eats a quart of ice cream every night. He would never gain a pound if he ate a gallon a night. I would. With that loss of brown fat, then you throw off that balance. And fat can accumulate in the wrong place, especially, not just subcutaneously, but around the coronaries, visceral fat, as I mentioned, and it leads to this whole imbalance we call metabolic syndrome. The exciting news is the transplantation of brown fat into obese diabetic rodents can reverse the symptoms of diabetes. It can make animals insulin sensitive again, they can lose weight, and the dream come true is without exercise, without cutting back on the diet, because you restored this balance of the metabolism. You can make these cells from pluripotent stem cells. Look on the left is a fetal-derived brown fat. I'm staining here for UCP1, a marker brown fat. You can see the cells, but they're not homogeneous. What we need for clinical use is homogeneous, scalable, defined, potent brown fat progenitors, and you can do that using a technology we call PureStem. This is a really big opportunity in aging. It's just one example of what we can do in aging, but it's estimated 1 out of 3 Americans are going to have diabetes, most of it, of course, 90% being type 2 by 2050. It's a huge expenditure in the United States, over about $250 billion in annual costs. A lot of direct medical costs associated with it and a huge amount of reduced productivity, like a lot of these age-related diseases. A problem near and dear to me is my father died of a heart attack, and it always bothered me, why can't we rebuild the vasculature of the human body? During development, embryonic vasculature reaches out to ischemic tissues and vascularizes them robustly. It builds the tree, the vascular tree. You can make embryonic vascular endothelium with 100% purity as well, using clonal expansion of progenitors. Vascular diseases being similarly, well, $1 trillion market worldwide. Now I want to talk, last part of my talk, about, I've talked about several revolutions here of thought. Finding the mechanism of cell aging, isolation of the human germline, the application of that technology and actually making products that enter clinical trials, reprogramming. There's a new revolution breaking, and it's a skunkworks project, really. It's a stealth project. It's not being discussed widely, but there's some groups working on this around the world. And we believe this is the biggest revolution ever. We call it induced tissue regeneration. What is it? Well, I begin with a quote from a researcher that many of us know in the aging field, George Williams, who worked on the evolution of aging. And he made, again, this prescient observation that seems so obvious, but hidden within it is a clue to some very profound biology. He said, it's indeed remarkable that after a seemingly miraculous feat of morphogenesis, it really is, isn't it? The human body is built out of nothing, de novo, and is alive the whole time. Can you imagine assembling a jet airline from components and it flies the whole time? Every component can fly. It's just amazing. but that it cannot perform the much simpler task of simply maintaining that which it's already formed. Why? On the left, you can see the development of a building. It's like embryonic development. You construct it out of nothing. And then we have a functional building, and then you maintain that building. But in aging, you see this destruction. It can't maintain that which it's already built. Why? Is it possible we could institute into the developed human the regenerative potential that originally led to the developing human? And that's the key to ITR. Why do we think this is possible? I was talking to Arnie Kaplan. Isn't that you out there, Arnie? We were talking about this earlier, and he said he quizzed people in his classrooms. I lobbed off my finger, how many of you think that medicine will ever be able to make it regrow? And he said, no one raised their hand or something. Who would think? Well, we should think so, because if you look on the scale of life I'm showing you here, simple organisms can regenerate. The planaria near the bottom of the list is immortal under the edge of the knife. You can cut off its head, it'll regrow a new head, and it'll do that forever. It doesn't have any limit to the amount of regeneration it can do. Other animals more advanced animals tend to show it, but only at the embryonic stages of development. The Mexican salamander, the third on the list, is a well-known example of a profoundly regenerative vertebrate. If you go way up to human, it's also well known that we have regenerative potential early in development. It makes sense. You're forming the body, why not form it again? But in up to 8 weeks of human development, while the human being in all this development, organogenesis is occurring, if you cut the skin, it heals back in about 72 hours completely and scarlessly. Once you enter fetal development, and development's pretty much complete, you start seeing the scarring response as a reflection that the body can't do that anymore. What's changed? That's the question. Another clue comes, let's look at the Mexican salamander. At the top you see the animal in the larval state. It's like the polywog state. Most such amphibians would go into an adult metamorph on the bottom left, an adult amphibian. But the Mexican salamander is different. It has what's called a heterochronous state. It is arrested in an embryonic or larval state as an adult. And it's thought that it's missing some signaling from the thyroid hormone axis. So it does not mature into to full maturity, but spends out its whole adult life as a larval in a larval state. And what it can do then is this profound regeneration. And this is really amazing. I remember a medical student I talked to once, and I said, what is the most, you're doing gross anatomy, what's the most amazing part of the anatomy? And I was thinking the brain, He says, no, the arm. It's just so amazing. It's so complex. It's so intricate. You can lob off these animals at the elbow, and they regrow it from the elbow. And lob it off at the wrist, they regrow at the wrist. How does nature do this? It has to be. All of you guys are scientists. It has to be that's repeating development. What are the molecular switches that are turned off as the animal goes from the larval state to the adult state, or in the case of humans, from the embryonic to the fetal state? If we could understand that, hidden within our own cells could be the potential of a profound scarless regeneration unforeseen previously in medicine. How would we get at that? So what we did is we cloned out. We took a single cell from this developing tree that we calculated was still in the embryonic as opposed to fetal stages of development, starting with pluripotent stem cells. We called this pure stem for lack of a better designation. And then we collaborated with a group called In Silico Medicine, who's using advanced artificial intelligence algorithms, deep learning, So these are computer algorithms that you may have heard that can learn, for instance, image recognition, face recognition. You can train these computers to do very complex analysis, recognize a Renoir. Some of you have used these apps on your phone, and you can take a picture of your choice, and it'll paint it as a Renoir would have painted it. This recognition of complex patterns can be applied to microarray data from thousands of different human embryonic progenitors that still have this regenerative potential, and you can compare them then to adult-derived cells and ask, how do these mechanisms work? They might not be that complex. Now I'm showing you, this is a stealth project, so I'm not showing you all the things here today, but I'm showing you one marker to show you how interesting this biology is. So this COX7A1 is a component of the respiratory chain in mitochondria. And we've known for years that this changes in embryology. So you're more glycolytic when you're an embryo and oxidative when you're an adult, and in cancer it's the Warburg effect. You go back to this state. We've known about this. The biology has never really been unearthed. So on the left you can see mesenchymal stem cells, neural stem cells, all kinds of adult and fetal-derived cells. They all are telling us they've passed the embryonic fetal transition. They've lost that profound regenerative capacity. Look at the diverse clonal embryonic progenitors. They're still embryonic, all of them. If you look on the bottom right, you can actually see we've traced out during human development how when this gene is turning on, and it's coincident with the onset of scarring and loss of regenerative potential in humans. And then on the far right, you can see that it's effectively reset by transcriptional reprogramming. Here's an example of how this plays out. So this gene, again, an adult marker, you can see in MSCs and then differentiated MSCs, and then in some chondrogenic and osteogenic embryonic progenitors, you can see that they do not turn this gene on, but on the right there you can see they profoundly turn on collagen II and make cartilage. We think this is the reason why if you induce trauma into a bone and inject these cells into a damaged bone like you see here, they can recognize and become bone where they should become bone and cartilage where they should become that tissue. Where I believe this field's headed is that we're going to be able to combine this technology with telomerase to make humans sort of like planaria, immortal under the edge of the knife. An ability to regenerate tissues indefinitely using these mechanisms. We call it ITR, induced tissue regeneration. To sort of define it, it's the induction of the ability to recapitulate embryogenesis in the tissues of an adult mammal by means of the administration of exogenous reprogramming factors, either in vitro or in vivo. So what we're beginning to learn about aging is telomerase turns off very early, maybe as early as 2 weeks in a lot of tissues. We start to age in the womb, as some people have said. This is playing an important role in aging, certainly in the hematopoietic system, but also there's this switch, probably like telomerase, being turned off because it's a tumor suppression mechanism to prevent tissue regeneration. It also is adventitious to prevent cancer, but the consequence is you can only scar, you cannot regenerate tissues as profoundly as you would with it on. Now, we've seen some demographics, but it's really worth taking a couple minutes to refresh our memory on this. We're at this, look at this inflection point, 2016, we're right where this curve is really beginning to hit us. 80% of our $2.5 trillion healthcare expenditures in the US are due to these very diseases we've been talking about, chronic degenerative diseases where the body cannot heal itself. 92% of the elderly have one, 77% have two or more. And it's only going to get worse. 4 of them have 2/3 of our deaths in the United States, and many of them need cell regenerative approaches because drugs are increasingly demonstrably ineffective. The boomers are a big part of this, but then after this we have the echo boomers, and we're not going to see any real relief from this demographic until Generation Z grows old. So this is a long-standing problem for the United States. Arthritis being perhaps the #1 complaint of an aging population, a massive percentage of our population having that, requiring knee and hip replacement surgery and a lot of pain. But you know the list. You know the drill. When I see this curve, I think of a tsunami. 76 million strong, adding 1 million to our nursing homes in the next couple decades, sapping the strength of our GDP. David Walker, the ex-comptroller in the United States, talks about the tens of trillions of dollars of unfunded obligations under Medicare. I wish that policymakers would look to science. When I grew up in the Sputnik era, everyone said, hey, you scientists, can you help out? Everyone seems to think that science and technology and biotechnology would just add to the problem. I point out to people, smallpox vaccine, you know how much it costs us to have smallpox put in abeyance every year? 0. Medical innovation doesn't have, because it's gone, right? Because of science and technology. Science and technology doesn't necessarily mean increasingly expensive therapies. It could potentially reduce costs, and I think we all know how that could be accomplished. And we can do some remarkable things. This is a patient that one of our centers put online, a subsidiary of ours, of Asterias, continuing Geron's trial with OPC1 for the treatment of cervical complete spinal cord injury. This patient was like Christopher Reeve, had a cervical complete spinal cord injury. He was on a respirator, had no sensation or motor function from his neck down, and is now texting his girlfriend and hopes to be married soon. And really remarkable things can happen with a lot of determination and a lot of vision. It's been a long time getting here. I wanted to remind us that, back when our nation was digesting how NIH should fund pluripotent stem cell technologies, Harold Varmus, the Nobel laureate and ex-head of the NIH, said that the development of cell lines that can produce any tissue of the human body is an unprecedented scientific breakthrough. It's not too unrealistic to say that this research has the potential to revolutionize the practice of medicine and improve the quality and length of life. I'm a gerontologist. When I was in Strawberry Fields in New York City, I could have swore this said, I'm aging. Blink your eyes, look again. It says, imagine. That's how we see the world. I know I see the sea of humanity with the problems of aging, but this is near and dear to the heart of all of us. It's us, and it's our family members. It's our society. It's a major challenge for, as we heard, Japan, for the United States. And so I invite all of you to imagine with me, how we can turn these basic discoveries into therapies that will help all of us in the human condition. Thank you very much.
When I was a student in the 1970s, a profound book that I read was Future Shock. What you heard, ladies and gentlemen, is Future Shock. It was profound, a profound speech. Thank you. Let's give a hand to Michael West, Dr. Michael West. I want to thank you for joining this plenary session. What are you going to do next? You're going to go down to the exhibit hall now. You're going to get some refreshments. You're going to drink a glass of wine, have a beer. Speak to our exhibitors, our wonderful exhibitors and sponsors that are in the exhibit hall, and certainly walk through and look at every single poster that was submitted. We have them from around the world. We're going to be presenting tomorrow, I think about noon. I'll have to check my calendar. It's getting a little confusing now towards the end of the summit, but we're going to be announcing our poster winners sometime around lunchtime or after lunch tomorrow. So please join us for the refreshments and the chance to network some more. See you tomorrow morning.